Okay. Sorry first. Anyway, for the stage, I call Margarida Ataíde, por favor. Maria Margarida. Elsa Baltazar.
Thank you and good morning to you all. So we would like to start this session as soon as possible, and by welcoming and asking our keynote speaker to go to the stage. Let me introduce you Margarida Ribeiro. She's a full professor at the Escola Superior Agrária de Castelo Branco at the Instituto Politécnico de Castelo Branco, and she has a PhD in forest genetics obtained in the Swedish University of Agricultural Sciences. She has a vast experience in Mediterranean species, in biodiversity and its distribution in global climate changes context. So due to her expertise in population genetics and macroecology, we would really like to welcome her presentation at this team today. So Margarida, thank you for being here with us, and you can go and present your work. Thank you.
Good morning. I hope you all are listening me well. So I'm going to start to talk about wild endogenous genetic resources. Due to several studies, I'm trying to present 3. I don't know if I will have the time to present them all, that we have done during this project, Cultivar, that joined together not only a network of people working at Escola Superior Agrária de Castelo Branco, as several other institutions like Coimbra University and CATAA that organized this Congress. And I have to tell that this was really a pleasure for me to work with all these people, and I have to thank them. So here we have a first paper that was published this year, and I'm going to talk very quickly about the results of this paper, which were done in this species that we know so well, the Cistus ladanifer rock rose or steva. And this species, which I think is really beautiful, so the name rock rose is obviously due to the form of the flower before blooming, and it's a very dry resistant species and tolerant to high temperatures. And since we are having some future outcomes that we need to work with species that are more prone to drought resistant, we have thought that we have here a source of biomass, a source of secondary metabolites, and a source of other things like capsules, seeds, and pollen that might be important as a source of income. And we know that there are 3 subspecies. One is ladanifer, the other one is sulcatus, and the other one is africanus from northern Africa. And the objectives was to know what was the impact of global warming in this species that originally is so resistant to temperature and to drought, but also to look into the past. Because sometimes, as you know, when we look at the past, we can understand better the future, okay? Like historical events or personal events. And here what we have used what we call algorithms that can do some modeling. Some of them are machine learning, like artificial intelligence-like. And we have collected a lot of data. Some of them was, for instance, the presence of the species. We now have this big data, and we can grab this information in a rather easy way. And afterwards, we can have also the information from the climate. And for instance, we have seen that the explanation of the presence of the species is better with temperature-related variables. And also we have precipitation, and of course, all the models for the future, we have used 2 slices of time, like 250, which is quite close now, okay, and 270, and also 2 global warming scenarios, which is this like business as usual, so we will do anything, and the other one was complying with the Kyoto Protocol. So after that, we also had some information, and this is my subject of previous subject of study, which is population genetics. This study, I helped Celestine Quintela Sabaris to do this paper. And we have seen with IFLP, it doesn't matter, it's a molecular marker, and we have seen very clearly that there are 4 different clusters in the Iberian Peninsula, which is the area where we have these species. So we have the southern one, we have this one, which was kind of enclosed by the Baetic Mountains in the southern part of Spain, which is also like a hotspot of biodiversity from the last glaciations, and Portugal and inland Spain. And here we also collect a lot of data from fossils, meaning pollen fossils. It's not very easy because cistus, it's a genre which means that it's difficult to distinguish the different kind of pollens. Anyway, what you can see immediately is that the darker the spots, the older they are. So it seems that we had, during the last glaciation, these species in the Iberian Peninsula bordering or by the sea, which is natural because the sea is milding the climate. So these are the modeling. It's a lot of maps. So these are the results. So for the past, we have them here. So last glacial maximum. So we had, like here, not so much of the species, because as you can see here, there is the probability of the presence of the species. If you look at the present, we have the species mainly in the Iberian Peninsula and in the northern part of Africa. Okay, so what happens in the future? So this is the worst case scenario in the bottom, and in the top in the other scenario. And as you can see, in the worst one for 2070, the species is collapsing to Portugal, and it's not migrating to the north. We don't know really the reasons why the models took us this picture, but we can really see that the species is going to diminish the area where it could be present, because this is probability of absence. Okay, here it's the results for the past, and we can clearly see that there are 3 refugia, and these are the data that we have grabbed from the modeling, okay, which is matching the molecular markers and also the fossil evidences. Okay, the second study, we have done a common garden trial, And so we have chosen a set of populations to try to define an improvement population because we want to make use of these species. Okay, so these are the populations that were selected based on molecular marker studies, so the most diverse species. So we have selected 8 plus 2 species from the region, Penha Garcia and Vila Velha de Rodão. And we had the capsules, we have the seeds, we have made some breaking dormancy protocol, we have produced the plants or make them grow in a greenhouse for about 1 year, but in this year 2 months where they were acclimating to the weather in outside of the greenhouse with a protective, shadow protective thing. And afterwards it was planted in autumn 2021. And this is the experimental design. We have 5 blocks. Each block is a repetition of each of the same type of plants. And these are the hardworking people here. So this is the experimental design. I think it's quite easy to explain. We have 10 populations, so 8 that we selected all around the area of the species, and 2 that are local, that means 10, and 1 seed per family, so 5 families per population, and 2 additional rows for destructive analysis. So this block was repeated 5 times. So we can grasp the environmental influence in this trial. And here what we have done was the indirect measurements. So we have collected the cross diameters and 8 to compute the canopy volume, which is an inverted cone, okay? And moreover, we have the direct measurements. So this year in July, we have collected the plants, and we have done some distributive analysis. And with distributive analysis, we are going to produce allometric equations, meaning we are going to pick these measures and to relate them with fresh and dry biomass production. So here, this is the hard-working people when we were collecting the plants in the field. So they are quite big because they are like 2 years old, not so old. And as you can see, maybe there is some resin here in the top. And this is very important to be collected and to be used. And here we are cutting the plants and separating the biomass that is woody from non-woody biomass. So further on, we are going to do this biochemical analysis. They were not already done. And these are the results. So this is the mortality, and it's very easy to explain. The higher mortality is to the southern populations, Moroccan from this Africanus subspecies. And the other one is Ladanifer, but it is the southern population, okay? Probably behind the Betic Mountains. So they did not migrate north after the last glaciations. And here, the best, or with less mortality, is one provenance, this one. Here, it's a Spanish provenance, but it's nearby the trial. Here, it's the 8, so the last measurement, which is in July 23. As you can see, the best growing populations in 8, it's Vila Velha de Rodão, Penha Garcia, so the local populations, and also the nearby population in Spain, which is here, And the worst popular, of course, this one that had very low growing, it's from Ponta de São Vicente, which is this Sulcatus subspecies, which is a subspecies that adapted to live in nearby the sea, and it's very resistant but doesn't grow very tall. And here the volume stands for the same results, okay? Exactly the same thing, and the Sulcatus population, which is this light blue, Ponta de São Vicente, it's also that has less volume. And here we computed the heritabilities because we have families. We have half-sibs sharing the same mother, probably not the same father, okay? And when we have a sibling like this, we share 1/4 of the genome. So we can compute the heritability. And the heritability here, it's not very high, it's like around 30%, not achieving the 40%, but this means what we can gain in the future with these traits we have measured, okay? So we can gain probably 40% or around 40%. The last study we have done with the Professora Fernanda Delgado and other colleagues, it's about a very special plant that we should try to conserve. As Fodelos bente-rainho, subespécie bente-rainho, or Abrotia. I don't know how to say the species in English, I don't remember. Doesn't matter. And we wanted to see what was the impact of the use of the soil and soil cover and global warming in this species. What was going to happen? Is it going to be more endangered? The answer was yes for the land use, but it was no for the global warming. And it's a little bit strange. So a species that it's not going to be affected by global warming, but this species only happens in Serra da Gardunha, and mountains are special places where probably the species are going to be refugied in the future, okay? So more questions, I think Professora Fernanda Delgado can help me in replying. So this is the team involved in all these works, and it was really very nice to work with these people, and this can really tell us that it's very important to work in a net, because each one of us can do some special thing, okay? So I thank a lot all these people, and I'm talking on their behalf. And thank you very much for the attention, and of course thank you for the opportunity to have this grant from Cultivar to do this work.
Thank you, Margarida. There is space for questions, and thank you for keeping the time.
I have no idea.
Because this is great for people here to put your questions and discuss a bit on your excellent work. Anyone would like to start the day?
One question.
Yes, there, please.
Don't be shy. I'm not afraid of the questions.
Okay. Good morning, everyone. Thanks, Margarida, for the wonderful presentation. I was wondering whether among the analysis was being done, was somehow verify any difference in terms of soil properties among the different populations you were addressing over these studies?
Which study? in particular.
So the one that you mentioned before from the different populations and then the establishment of your
Trial.
Trial, yes.
Okay. Okay. That's an interesting question. Actually, the work that my colleagues Celestine Quintela Sabaris has done, he has made the PhD, in this species, this Cistus ladanifer steva, exactly for the proprieties in capturing toxic metals. So some of the populations he has studied were in serpentinic or old mines that were like having these toxicity problems. So actually we have differences in soils in the populations that we have selected, but that was not the factor I took into account when I selected the populations, actually. Because in general, this species is growing in regions where the soil is not very good. It's a very poor soil. And it's a pioneer species. But since one of the things I was worried about was selecting for growth and selecting for production of resin and some metabolites. What I wanted to have, and that's why I think your question, was a broad variability, okay? So I wanted to have a little bit of everything, like the different subspecies and the different populations from different places. But I really can't say that I was looking at the soil. But do you think this would be important?
Well, I don't have the right answer for that, but in other species, at least the ones that I'm working with in equivalent trials, yes, we had great influence from the soil. So the original populations, when they were reinserted in other areas of the country, did not behave exactly on the same way. And when we went back and analyzed the soil, in some of these cases we found quite big difference. I'm not sure if that explains the behavior or yes, but yes, that was our experience.
Thank you again. Since I have this forestry background, and in forestry it's quite common to have this kind of trials. Maybe that's why I thought about it. And I had a colleague, Nuno Borralho, and he has worked many years in a row to the forestry improvement of Eucalyptus globulus in Portugal, which is considered one of the best improvement programs in the world. And I've been told of that when I was in Sweden doing my PhD, because it's going on since the 60s. And what happened is that in general, when you do these provenance trials, you should have them in different places, because the place where the trial is matters, okay? So that's why it was not surprising for me that the nearby populations are behaving the best. But what is really important is to have a very big variability, because if we want to select, we have to have variability. Otherwise, you can choose something from a small spot instead of choosing from a big one. That's why probably nowadays when you have contests for scholarships or professors, it's a worldwide context to have also variability. It's the same thing.
Okay.
Okay.
So, Margarida, and thank you for this other level of discussion. Please continue it at the coffee break, please, so we can move with the session. And thanks for the answers. I will pass to Fernanda now. Thank you.
Thank Thank you, Margarida. We are going to begin the selected oral communication, and the first one is done about ex situ conservation of agri-food germplasm, case study from the Cultivar project in Portugal, and Elsa Baltazar is from the Center for Functional Ecology, Terra Associated Laboratory from the Coimbra University. Please, it's your time.
Okay, good morning, everyone. First of all, I want to thank the organization for the opportunity of being here presenting my work. Today I will talk about ex situ conservation of agri-food germplasm case of studies from Cultivar project in Portugal. The central region of Portugal is particularly rich in agrobiodiversity. This region joins local varieties with interaction with modern horticulture and fruticulture. During this work, we identified several issues, such as the high risks of genetic erosion, of the genetic diversity erosion, the abandonment of rural areas, aging of the farming population, extreme climate events, and lastly, emergency of new diseases. The fruit sector, this region is very strong in the fruit sector, and the sector invested during the last years in improved cultivars, mostly clonal based, in order to increase the production. The using of the improved cultivars, the cultures become more homogeneous and are susceptible to biotic and abiotic stresses. The use of these clonal-based cultivars lead to the abandonment of the old and traditional varieties, and we are in risk to lose this specific and these valuable characteristics. So here, germplasm conservation play a decisive role for its conservation. Germplasm conservation can be made by in situ or ex situ. The conservation in situ can be made in the natural environment or on farm, in the farmer's field, or ex situ. With seed storage, with the germplasm banks, or with in vitro approaches such as micropropagation, somatic embryogenesis, genetic transformation, cryopreservation, and so on. And in the last, in the field gen banks like national parks or botanic gardens. This work is part of the line 4 of the Cultivar project and aimed the valorization of selected natural endogenous resources. This objective have several points, but today I will talk about the identification of valuable germplasm and how to conserve it. Throughout this project, several species were conserved and tried to conserve in vitro, such as Prunus dulcis, Citrus sinensis, Corylus avellana, Sidonia oblonga, Prunus cerasus, Prunus avium, and lastly, Prunus persica. For ex situ conservation, we use the micropropagation method. For the establishment of the plant material in vitro, we count with the help of the farmers to select the plant material. We do it by 2 ways, by the introduction in vitro by the nodal segments or by seed germination. After the establishment in vitro, we multiply this material, then we root this material to have a complete plant for further tests. The The in vitro establishment by nodal segments, probably some of them knows this protocol, but we select the mother plant in the field, then we collect the branches, then we bring this material to the lab, then we cut in little pieces, then this material is submitted to disinfection and sterilization and put in vitro. After that, we have to reach the multiplication step. This is the most important one because we have to adapt the culture media to the different species, to adapt the growth regulators, and it's a very hard process in this specific method of conservation. We also do the in vitro establishment through seed germination. Here we use the traditional varieties. We have a protocol here, as you can see. We have an example of peach seeds. We remove the external part of the seed, then we washed. We have done several washes with ethanol and fungicide, and in hypochlorite was a hard disinfection to allow us to isolate the embryo in aseptic conditions and put this embryo in vitro. After the embryo germination, we just use for multiplication the aerial part of the seed. To talk about some results, we have here some of the results with the test with the germination of almond seeds. We compared traditional varieties with commercial ones, and we didn't saw any different statistical differences. Here we have some examples of in vitro establishment of almond tree. We just do this method. Here in the quince tree, we used the two different methods. We established it in vitro by nodal segments and by seed germination. For sweet and sour cherry, here we used two different ways for the in vitro establishment. One way, we used the annual branches collected in the spring, and the other way was the collection of dormant buds, the branches with dormant buds in the winter, then we forced to sport in the lab. And we use these different meristems in the different times of the year to establish in vitro. Then we do the multiplication. One more time, this was a hard part because we have to adapt, and in this way we lose a lot of material because we can adapt rapidly to the species that we are working. And we have some examples of the rooted explants, as you can see. We tried to do also other conservation of other species, the hazelnut and the orange tree, but we can't conserve it because one more time it was very hard and material do not have the perfect conditions to establish in vitro. Here we have some of the material that we conserved. We have almonds, we have sweet cherry, we have sour cherry, and we have quince tree. Here we conserve traditional varieties, but we also have commercial varieties. These commercial varieties are very important because for further studies we need to compare the performance of these commercial varieties against the traditional ones to evaluate the differences in several parameters that we further study. For conclusion, the recalcitrant of some species had an impact on their ability to multiply in vitro. Several endogenous resources with agri-food and conservation relevance were identified and conserved. Plant tissue culture approaches were optimized for the in vitro establishment, multiplication, rooting, and acclimatization of the selected germplasm. And thank you all for listening, and it's okay. I think I'm on time.
Thank you, Elsa. We have time to 1 or 2 questions. Yes? Okay. You can do it. Teresa.
Olá, eu sou Teresa Carita, do INIAB.
Sim.
Dar os parabéns pelo trabalho que faz, porque é fundamental valorizarmos os recursos que temos. O que lhe queria perguntar é se tem algum tipo de colaboração com o Banco Português de Dermoplasma Vegetal, em Braga.
Não temos.
E pensa vir a ter?
Pois, neste caso, este projeto foi desenvolvido no âmbito do projeto Cultivar, que infelizmente está no fim, e lá está. A continuação deste trabalho necessita de também, claro que, investimento, porque para que tudo isto aconteça são necessários recursos. E eu, infelizmente ou felizmente, irei acabar o meu projeto de doutoramento e não haverá tempo para poder... E lá está, e muitas das vezes o problema é os recursos e não temos recursos humanos que nos permitam continuar com este trabalho e, possivelmente, manter este que já foi feito. É algumas dificuldades do nosso sistema que todos nós estamos habituados.
Obrigada. Ficarem.
Passamos ao português.
Obrigado. Can I?
If you want,
in
yes,
English?
yes. Okay.
What, but in Portuguese?
No. It's
Well, thank you very much for your very interesting presentation. Here is Pedro Brau from Landratec. Well, I have to remark that we are losing a lot of biodiversity, not only in the forests, but also in our traditional cultivars. So it's a very meritable work that you are doing. My question is, did you assess the maintenance of phenotype in the propagated, in your germplasm bank? Because, for instance, this is something that happens in the in vitro culture, which is some epigenetic modifications on the organization of allelic expression, so, and it changes the phenotype. So if, in the cultivars, we are maintaining a phenotype, we want to keep a phenotype, so...
We did not do these studies yet. We can't say that they maintain the phenotype or they change, because we didn't these studies yet. I don't know if I answer your question. I don't know.
Yeah, Thank you very much.
Thank you, Elsa. Welcome. Now we are going to watch Tercia Lopes from Centre for Functional Ecology, also from the Coimbra University. And in the same line, we are going to, your communication is in vitro establishment and molecular analysis of the micrograph union formation in almond, Prunus dulcis. And you can begin.
Okay. So good morning, everyone. My name is Tercia, and I would like to start by thanking for the opportunity of for being here and to present my work. I will be talking about the in vitro establishment and molecular analysis of micrograph union formation in almond. For as long as we remember, biotechnology has been a part of our lives. All over the world, cultivation and selection of numerous varieties across multiple generations has been made, usually prioritizing commercial varieties with high productivity. But this may lead to a decay in variety and in variety of traits and diversity, which may be commonly known as genetic erosion. So, in a world of daily climate change, it is important to call attention to the fact that the answer to these problems caused by these changes may have already been or still is present in our world. Within that same diversity, we are at risk of losing. So, in order to counteract this alarming scenario, it is important to try and find ways to preserve and cultivate these traditional varieties, conserving this germplasm. So it is in this situation that biotechnology can be of great value once again, through processes like cryopreservation or micropropagation. it is possible to try and to establish in vitro material with various characteristics of interest. The conservation of germplasm is one of the most effective methods to preserve genetic characteristics, either from endangered species, species with medicinal properties, or with commercial value, and is being used to restore degraded land and to maintain ecosystems in landscape. With that in mind, the Cultivar project, particularly the Line 4, aims to valorize selected endogenous genetic resources through the conservation of germplasm with in vitro techniques. And in this case, particularly, we focused on the almond tree. This is a species that belongs to the Rosaceae family, and the increasing lifestyle and health awareness has been leading to a greater consumption of dried nuts, which includes almond. And this has contributed to an over 25% worldwide increase of almond production over the last decade. And Portugal is no exception, and it's predicted a 20% increase of almond production in the next 5 years. Moving to grafting, this is one of the oldest horticultural practices, and it's commonly used to propagate commercially prunus species. Major outcomes can be achieved, namely production increase and improvement of fruit quality. However, this traditional grafting is labor intensive and weather dependent, and also sometimes there are phytosanitary issues that impact the success of the graft. However, this may be overcome with in vitro micrografting. This is a technique that produces eco-friendly and pathogen-free plants. And besides its important application in crop improvement, it has been used to study the molecular mechanisms involved in cyan and rootstock interactions. Regarding the almond tree grafting propagation, GF677 is an hybrid and is one of the most used rootstocks in all over the world for the production of almond. Antoinette of Raduel and Lauren are some of the most used in Portugal as science to produce almonds. But in the scope of the cultivar and in collaboration with the Sebal Institute, we are trying to better characterize Portugal traditional varieties so that we can have a broader range of options to deal with the adverse conditions that we may face. So the aim of this work was to evaluate wound-responsive and oxide-related mechanisms involved in different combination of micrograft union from almond tree micropropagated shoots. Graft union formation has three main events: wounding, callus formation in response to the wounding, and vascular reconnection. Researchers have pointed out that the wound response is a key factor for the successful establishment of the graft union. And many candidate genes have already been identified, but their specific role in grafting, particularly in woody species, remains unclear. Regarding this work, different combinations were made between traditional and commercial varieties of both cyan and rootstocks. The sampling was done 0, 7, and 21 days after grafting, and gene expression and auxin quantification are being analyzed. To show you some results, these are how the micrografts looked. On the left picture, this was the day that the micrograft was performed. 7 days in, we can see in the middle picture a little callus formation on the union spot, and 21 days after, we can see a more solid formation of callus in between the cyan and the rootstock. To evaluate the micrograft success, this was made 21 days after grafting. I have some pictures to show you what we considered success and in success. Homographed between bitter almond and Ferraduel and GF677 and Lauren showed high success, above 80% in the micrografting. Bitter almond and Canhota also had some successful grafting, as well the combination with GF and Ferraduel. On the other hand, bitter almond and Lauren, they appear to be incompatible. Various times, I don't know if you can see, but the rootstock, it shows signs of necrosis. When we were trying to handle the graft, the union broke, and it showed much signs of hyperhydricity of both the rootstock and the scion. So in the micrographs that we considered successful, we applied the paraffination technique and to perform microscopic observations. The cuts were stained with a solution of Astra Blue and Safranina. And in the right picture, there is a yellow circle where we can observe some connection between the scion and the rootstock, which suggests that the vascular reconnection is starting to develop between the scion and the rootstock. So to conclude, we were able to successfully establish various almond tree genotypes. The response dynamics actually depends on the combination, as some showed incompatibility, and it was possible to observe the micrograft reconnection in those that we considered successful. So I cannot end this presentation without thanking to all the people that made this project, this work possible, to all the institutions, and lastly, to thank you all for your attention and try to answer.
Thank you very much, Tercia. And we have time to one question or two.
Just one question. Which is the culture medium? And just one. Don't you have problems with fungi?
In the establishment, a lot of trouble to try and establish the. That's the crucial step always. And the medium, we use the MES with some supplementation, but it's difficult. We are still trying to get it right, because if we put too much DAP, there is a big callus formation on the base of the shoot, and that compromises the uptake to the scion. So we are still dealing with it's a very difficult word, because we are still dealing with to assert the media. It's all new. So I'm on the first year, so we are still trying to assert all the things. Okay.
More questions? We have one more question from Margarida.
I don't know if you achieved the routing, the final step.
No, we still, because sometimes the hyperhydricity is a problem, and as we are still trying to get the media right, we are still trying to find the perfect solution for the hyperhydricity, because it compromises the rooting and the health of the plant. And almond is really difficult to root, and it actually depends a lot on the genotype. Sebal has already concluded that, so we still have to find the right genotype and go from there. Thank you.
Thank you, Tercia.
Yeah.
Thank you. So let's move to our next speaker, which is Joana Domingues from Centro Biotecnologia de Plantas da Beira Interior. Joana, she will present her work on lavandula section stoecas from Beira Interior, chemical and morphological approaches. I know that you will speak in Portuguese, so you have your slides in English. It's okay. We have the translation. So thank you.
Bom dia a todos. Vou dar início então à apresentação deste trabalho que faz parte de uma das linhas de investigação do projeto Cultivar, que se baseou na caracterização morfológica e química de espécies de lavândula da secção stoecas recolhidas na região da Beira Interior. Pronto, ok. Começar aqui por fazer uma breve introdução a este trabalho através da base de dados Global Biodiversity Information Facility. Nós conseguimos observar a ocorrência das espécies e a sua distribuição geográfica em termos globais. No entanto, estas três espécies que trago aqui, são elas a lavândula pedunculata, a lavândula stoecas subespécie Louisieri e a lavândula stoecas subespécie stoecas estão fortemente presentes na região da Península Ibérica e toda a região envolvente ao Mediterrâneo. E, portanto, salientar apenas aqui um endemismo restrito à Península Ibérica, que é a subespécie Louisieri. Ora bem, para caracterizar morfologicamente uma espécie, devemos consultar referenciais taxonómicos e morfológicos de referência, obviamente. De acordo com a informação transversal destes referenciais, há alguma controvérsia em relação à ocorrência geográfica nesta região das três espécies referenciadas, assim como alguma ambiguidade nos limites de alguns parâmetros morfológicos diferenciadores entre as duas subespécies. Como podemos ver aqui, também, as fotografias das três espécies que estamos a falar aqui. E selecionamos alguns parâmetros que são indicados como os mais diferenciadores entre as subespécies e, de facto, se olharmos para os limites, eles andam muito próximos ou por vezes chegam a ser iguais entre estas subespécies, o que torna difícil a sua inequívoca identificação através da morfologia. Bem, então se temos a tarefa dificultada em termos morfológicos, sob o ponto de vista químico dos óleos essenciais, parece que temos alguma sorte. Aqui estão apresentados os compostos maioritários dos óleos essenciais das diferentes espécies. Salientar então aqui, mais uma vez, a subespécie Louisieri, com a exclusividade praticamente no reino vegetal, da produção destes compostos que estão assombreados, que são compostos de necrodano, sendo o principal composto volátil presente no óleo essencial da subespécie Louisieri o acetato de transalfa-necrodilo, que por muitos autores tem vindo a ser considerado como um marcador quimiotaxonómico da subespécie, e isso facilita-nos aqui um bocadinho em termos de diferenciação de subespécies. Só chamar mais uma vez então, continuando aqui a chamar a atenção para esta subespécie e de facto para o seu potencial biológico, em estudos onde é comparado, o seu óleo essencial é comparado com diversas espécies, quer seja do género lavândula ou de outro género, de facto tem revelado um potencial biológico em termos de atividades antioxidante, antimicrobiana, anti-inflamatória, inseticida, e também revelou resultados promissores no tratamento da doença de Alzheimer. E, portanto, sendo o endemismo da nossa região e da Península Ibérica, acho que deve ser valorizado e temos que tentar tirar o máximo de partido desta mais-valia desta subespécie. Relativamente aos objetivos deste trabalho, como disse, faz parte então de um trabalho mais abrangente. Os objetivos foram essencialmente a caracterização morfológica das espécies recolhidas, a caracterização dos componentes voláteis do óleo essencial e tentar associar variáveis edafoclimáticas das populações. Relativamente à metodologia, foram recolhidas entre 10 a 20 espécies nos diferentes locais, são 8 locais nesta região da Beira Interior. Aqui na tabela temos a identificação que foi feita in situ das espécies. Temos então 6 populações de subespécie Louisieri, uma população de lavândula pedunculata e temos ali a população VR, que foi identificada inicialmente como sendo subespécie stoecas. Relativamente a cada espécie, foi herborizado e foram depositados no herbário do laboratório da Escola Superior Agrária de Castelo Branco. Foram avaliados 26 parâmetros morfológicos com base nos descritores da UPOV para o género lavândula. Como cada espécie recolhida no campo foi georreferenciada, posteriormente conseguimos obter informações relativamente às variáveis edafoclimáticas, que foram no total de 26. Relativamente à extração do óleo essencial, foi obtido por hidrodestilação e o seu perfil volátil obtido e identificado através de cromatografia gasosa acoplada à espectrometria de massas. Em termos de resultados dos parâmetros morfológicos, como seria de esperar, a população de lavândula pedunculata verificou-se, na maioria dos parâmetros, diferenciadora entre as outras subespécies. A subespécie que foi identificada como stoecas não apresentou diferenças significativas, ao contrário de uma população identificada como stoecas, a população VV, que pareceu diferir em termos estatísticos das restantes populações de Louisieri. Em termos de caracterização química, foram identificados 36 compostos fenólicos. Conseguimos perceber aqui, com esta análise de componentes principais, que a maioria das populações da subespécie Louisieri está muito associada então aos compostos de necrodano. Este grupo que temos aqui mais arroz. Pelo contrário, a pedunculata mais associada a compostos como a fenchona ou cânfora. Relativamente ao marcador quimiotaxonómico da Louisieri, fizemos então a quantificação relativa do acetato transalfa de necrodilo. Esteve ausente, como seria de esperar, na lavândula pedunculata e conseguimos identificar este composto na população identificada inicialmente como stoecas e a população VV, mais uma vez aqui, a diferenciar-se das demais relativamente ao teor médio deste composto. Ajustando então aqui, para terminar, uma análise de componentes principais relativamente às condições, às variáveis edafoclimáticas, só chamar aqui a atenção para a tal população da subespécie Louisieri que parecia estar ali meio perdida, não encaixava nas outras populações em termos dos parâmetros avaliados. E, de facto, ela está bastante associada a elevadas temperaturas, ou seja, o que nos leva a indicar que é uma população com maior termicidade e, por isso, um maior stress hídrico. Concluindo, então, fazendo aqui alguns pontos principais deste trabalho, tendo em conta a avaliação morfológica, de facto, existem parâmetros que são características morfológicas que são intermediárias entre a Louisieri e a subespécie stoecas, ou seja, mesmo ou de uma ou de outra, eles partilham esses limites. Confirmar então a presença da subespécie Louisieri nesta região e sugeria como perspectivas futuras abranger mais populações ao longo do território nacional, principalmente estudar mais populações da subespécie Louisieri e da subespécie stoecas. E por fim, agradecer à equipa, inicialmente aos nomes que estavam inicialmente na apresentação, e também às entidades que nos ajudaram também, de certa forma, a concluir este trabalho. Obrigada e peço desculpa pelo tempo.
Obrigada, Joana. Some questions? Alguma pergunta? I don't see hands raising. So, acho que vou fazer uma pergunta só. Porque o estudo é muito baseado em populações e
Sim.
essas populações acabam por ter algumas espécies e depois a amostragem e a recolha dos óleos é feita nessa mistura. Prevês algum impacto que essa mistura pode ter depois nos óleos que são analisados, nesse rendimento, ou como é que isso podia ser controlado de algum modo?
É assim, nós em cada população tentamos recolher sempre, lá está, a mesma espécie e de facto aqui a nossa principal dificuldade foi em diferenciar as subespécies, que de certa forma acabamos por perceber que aquela inicialmente identificada como stoecas, à partida será a Louisieri, tendo em conta a sua composição química. E a importância da identificação correta das espécies prende-se sobretudo com o seu potencial de aplicação e eventualmente também a sua toxicidade ser diferente em termos de constituintes químicos. Não sei.
Sim, era essa mesma a questão.
Posso dar aqui uma chega, já que estamos aqui neste trabalho. Pronto, um dos grandes problemas que tem acontecido é que o óleo essencial que está à venda na maior parte do nosso país é uma mistura das espécies e não é isso que nós queremos. Nós queremos efetivamente identificar e ter a nossa lavanda Louisieri, que tem um potencial de aplicação completamente diferente da lavanda pedunculata, que é ao fazer o rosmaninho maior mais conhecido e que existe em todo o país, e efetivamente nós temos aqui um potencial de uma espécie que tem um composto diferenciador, com características diferenciadoras também em termos da sua atividade e, portanto, é por aí que nós queremos passar esta mensagem e passar e mostrar que é uma realidade, porque as pessoas colhem tudo a eito e elas existem consociadas e, portanto, no mesmo espaço. Era só isto. Obrigada, Joana.
Okay, so now we move to a different stage of this session. I appreciate, yes, we appreciate that you've been here in the stage with us, but now we go to a cycle of 3 short communications. So we ask Daniela Duarte, Mariana Correia and Vanessa Vasconcelos to come to the stage. So this will go in a different way now. They will present each other a short communication. Sorry.
Daniela, Mariana e Vanessa.
Okay, so please join us. They will present. And now it works in a different way, so this will be short communications. Please save your questions for the time after the 3 communications. So to make it fluid, we will now start this part.
Start with Daniela Duarte from the Center for Functional Ecology of the Coimbra University, and she's talking about understanding drought tolerance in copy, vinha unguiculata, Walp, insight from 3 Portuguese land races.
Just a quick note before starting.
Okay.
Não estava a funcionar.
Agora. Só uma nota antes de começarmos. Eu reparei que há pessoas que se calhar não repararam que temos os headsets lá fora para tradução simultânea de português para inglês ou inglês para português. Portanto, se tiverem alguma dificuldade com a língua, portanto, estamos lá fora em frente ao registro. So if you haven't found the headsets, we have translation from Portuguese to English and English to Portuguese, so it depends. So it's in front of the registration counter. Sorry for the interruption. Thank you.
Good morning. Today I'm presenting the work that I've developed with the help of my colleagues, titled Understanding Drought Tolerance in Caupi, Insights from 3 Portuguese Land Races. Caupi belongs to the Fabaceae family and is considered a nutritionally rich but neglected crop. It is well adapted to drought and challenging climatic conditions. And in the Lourdosa region in central Portugal, there are 3 Caupi land races with interesting agronomical traits: cara verde, arroz, and cara preta. A lack of more detailed knowledge of the genetics and physiology of these land races weakens effective breeding programs. Therefore, our study basically focused on the phenotypic characterization of these 3 varieties in order to assess differences between them. And secondly, we subjected the plants to water stress conditions to better understand their phenotypic and biochemical response, as well as their gene expression patterns. From our results, when analyzing their phenotypic characterization, we saw no differences between varieties during vegetative phase. However, when we subjected the plants to water stress conditions and we analyzed the phenotypic and biochemical response, we did see that cara verde showed root growth reduction and an augmentation in total phenol content. This increase in phenol comes aligned with previous studies, because plant phenols have a natural antioxidant role, and therefore we can assume that maybe Caupi plants use this strategy as a way of reduce the negative effects caused by water-limited conditions. When we analyzed the gene expression patterns, we also saw a general increase in relative expression under stress conditions, again particularly in cara verde. The genes we analyzed were associated with the response to dehydration stress, as well as the protection of the small heat shock protein. Therefore, our main conclusions can be that cara verde may possess better drought tolerance compared to arroz and cara preta, and therefore be a promising candidate for breeding strategies aimed at developing drought-tolerant Caupi cultivars. Also, our study highlights this important matter that this is in the context of global climate change and food security concerns. Thank you very much for your attention.
Very much. Now, Mariana Correia are going to speak about physiological and biochemical responses of prunus roots during water stress recovery.
Good morning, everyone. I'm going to talk about the physiological and biochemical responses of 2 prunus rootstocks during water stress recovery. Rootstocks are used to help surpass a variety of biotic and abiotic stresses and are critical to for stone fruit production. And the rootstocks, I don't know what's that. Não sei porque isto não está a passar. Como isto é isso? Well, I'm just gonna move on. Rootstocks are used to help surpass some abiotic and embiotic stresses and are critical for stone fruit production in Portugal. The rootstocks ensures the viability of the root system, provides adaptability to the soil, and guarantees the support of the science, water, and nutrition absorption, and guarantees backup of compound storage. You guys, this is hard. Não
Estava tudo a correr tão bem.
Continuar a apresentação.
Ok.
Olha para ali. Isto aqui esquece. Olha para ali.
Certo.
Não olhes para aqui.
Sim. Okay, so we picked two hybrid rootstocks, GN15 and GF677, and we exposed them to stress during 18 days. We divided each variety in 3 groups, one control, well-watered, one exposed to stress by waterlogging, and one exposed to stress by drought. After those 18 days, we measured photosynthetic parameters, relative water content, and pigment quantification. Then for recovery, the plants were well watered for 30 days, and we did the same, analyzed the same parameters as before. Okay, after stress, we noticed that plants exposure to drought experienced some arrested development, and plants exposed to waterlogging experienced some hyperhidrosis of the foliation. These traits were kind of improved during the recovery process. We noticed that drought and waterlogging obviously induce some physiological impairments, especially in the drought stress. RWC was reduced by drought, and there was a decrease of chlorophyll content for both rootstocks and both stresses. For post-recovery, the assimilation of CO2 conductance, stomatal conductance, and transpiration in the post-drought period increased. For post-waterlogging, the gas exchange perimeters and chlorophyll A fluorescence perimeters show that the stress is still very present. The RWC achieved the control levels, and for post-drought period, chlorophyll content increased, meaning carotenoids worked as a protection, but for post-waterlogging, GF67 showed the lower contents of chlorophyll, which is not that good. After the stress recovery, some physiological
Trata da organização do centro que organiza aqui o nosso evento, e ela vai falar sobre physiochemical and nutritional characterization of queens from the Cova da Beira region. Você passar aonde?
Good morning. I'm here today to talk about queens from Cova da Beira region. The study was developed at CATAA in the scope of Cultivar project. Queens is a fruit of autumn with nutritional characteristics that can provide benefit health. In addition, queen trees are easy to grow and resist to adverse climatic conditions. However, queen produce is neglected and undervalued. So, to valorize queens for Portugal, these parameters, nutritional analysis and quality physical chemistry, was evaluated for 4 queens cultivars, maçã, pera, Portugal, and galega. Quality physical chemistry parameters showed different significance. So, maçã presented the highest weight and size, which is interest for consumers because it can bring more economic return. However, galega presented the lowest hardness and a high yellow color, which is interest for consumers because it's important parameters. In addition, galega is the sweetest and presented the lowest acidity. Nutritional analysis also showed significant difference, and queens is mainly composed with water, carbohydrate, and sugar. The sugar profile showed that fructose is the major one, followed by glucose and sucrose. Mineral analysis showed that Portugal is the cultivar which presents the high amount of minerals, except for sodium, zinc, and iron. So, overall, the physical, chemical, and nutritional parameters of queens depend on the cultivar because they show the significant difference, and the variability in the characterization data of queens can be explored to develop new products which can result in the valorization of their source, and consequently may encourage queens cultivation in Portugal. The future perspectives are to determine bioactive compounds and to develop new products. Thank you all for listening.
Thank you, Vanessa.
Thank you, Vanessa. Thank you all, and for keeping the time and enduring with the problems. So now we have time for questions. Any question to our speakers? I don't see any hands. Fernanda, do you want to?
I have one general question to Vanessa, and it's the future. What new products could be interesting to develop based on these variety studies?
We already started to do this with bread. Yes, use the
beans?
Yes, the dehydrated, the queens, and do bread with this. It's one of the products we started to develop at CATAA. And another idea is to do vinegar. So,
vinegar?
Yes, it's interest as well.
Okay, thank you. Another? More questions? To copy to Daniela. You know what is the scale of consumption of green beans compared to rice or to black beans? To the other the other varieties. You know here what is the really consumption of the people to the green bean or not?
We didn't make that study, but we did a parallel one on a survey about understanding which varieties people knew about,
yes.
Not the consumption. We didn't saw the consumption.
Okay, thank you.
I don't like that. Just a question to Mariana and concerning this issue with the rootstocks. I mean, why did you went and study the waterlogging? So since we are in a drought, climatic change context, or what was the main issue with the waterlogging in your study?
I think we have different stages of climate change, and the waterlogging is definitely a problem. And we see that waterlogging is even more stressful to the plants, and even after recovery, it's even harder for the plants to recover physiologically. So I do feel that waterlogging, maybe we're going through drought, but when it rains, it pours, and it's definitely a problem.
Okay, yeah, the extreme climatic events.
Exactly.
Okay, and so if we don't have any more questions, we finished with, I would say, an amazing punctuality. So thank you for our speakers for that, for you to being there, and now let's go for the coffee break. Thank you all. Yes. Thanks.
Inês, fala.
Here, the authors of the posters from the sessions of yesterday, from theme 1, theme 2 and 3, please be next to your posters, try to be next to them. In case there are some questions, you could talk about your posters. And also the sessions of today, so session 4, the one of this morning, and the one next to coffee break, like theme 5. So you please, authors, try to be in the area of the posters while getting the coffee break. It's just there. So because we will deliver the prizes for the best poster when we come back from the coffee break for yesterday's sessions. Vamos dar os prémios a seguir dos temas de ontem, portanto, para as melhores posters, que ontem já não houve tempo, pedimos desculpa, e portanto, tentem estar na área dos posters se houver questões. Obrigada.
From the University of Congresso.
Good morning, everyone. Let's see if we can have people coming from the coffee break with the bell ringing. But I think we can start. It is a pleasure to be here together with Marie for this theme 5 on biodiversity, ecosystem function and services. And it is with great pleasure that I asked our speakers to come to the stage. I asked Carmen Vasquez, Silvia Castro, Elsa Baltazar and Ricardo Leitão to come and join us here on the floor.
Where do you want?
We can keep the order. Okay. So we will start with our plenary speaker. It is with great pleasure that we welcome Carmen Vasquez from the University of Wageningen. She is a postdoc researcher on the University of Wageningen, doing much of her work on soil functional biology, as you will hear just now. Okay, thank you, Carmen, for coming.
Okay, well, thank you to the organizers for the invitation, and thank you all of you for your attention. I was just very nicely introduced. I'm Carmen Vasquez Martin, originally from Spain, so happy to be close to home. But I'm based in Wageningen University, where I do quite a bit of work trying to disentangle the role of soil biota in maintaining soil health and in sustaining sustainable food production. And the work I'm going to present to you today is a little bit different than some of the other presentations I've seen, because I'm going to present part of my own work, but also work of my colleagues, and of course for the contextualization, a bit of work from other researchers, because soil biota is such a broad world that it just takes a little bit of help to be able to give a good context. Let's see if it collaborates there. So if you ever happen to find yourself in a grassland such as this one, you may not really be thinking about it. This is in the Netherlands. You may see the pretty cow, you may see the very green grass, but in about a square meter of a typical soil, an average soil, you can find something like 100 earthworms per square meter, belonging to around 10 species. About 20,000 enkitrates, these are smaller worms, belonging to around 20 species. 10,000 nematodes, so individual animals, belonging to around 100 species. You can find like 50 kilometers of fungal hyphae under your feet in that square meter, and like billions of cells belonging to hundreds of thousands of species of bacteria. And what's really important and relevant about all of these organisms is that, of course, they take part in processes that in turn give rise to ecosystem services. They are part of processes that have to do with food production, of primary productivity, for water filtering, for the control of pests and diseases. So maintaining this diversity in the soil, or being aware of this diversity in the soil, is very, very important to be able to sustain production in the long term. And I'm going to give you a few examples now. So for example, when we think of microbes and we think of bacteria and fungi, we often, and I've heard a couple of these today, we think about the disease they bring, and that's fair enough. They are tricky to work with, but besides the disease they bring, they have huge impacts on the cycling of nutrients in the soil, and they have huge impact in, for example, pollutant reduction, because they can actually hold on to many pollutants. Fungi, we also think about often in terms of disease, and we've also heard one of the speakers talk about how difficult that is for them. But fungi can actually have many other functions in the soil. Here you see, and I'm sure many of you are very familiar with mycorrhizal fungi. They create a symbiosis with the roots, and thanks to that symbiosis they get carbon from the plant. But in exchange for that carbon, they provide often phosphorus and other nutrients, and water that is present in pores that are very difficult to reach. A little bit less known may be this picture, which I love and I didn't take and I couldn't find who to credit for. But here in this picture you can see a fungi that has trapped a nematode and is about to feed on it. And you can see the cells surrounding the nematode because it's created a lasso to trap it. So fungi can also control some of our pests. Same for nematodes. We're very used to seeing them as a pest, and here you can see some of those feeding on the roots of grapes. But in fact, through their feeding on bacteria, they can also help in mineralization and therefore create or increase the amount of nutrients that are available to plants. But they can also feed on other nematodes, and this is the picture you can see here on the right. And in that sense also provide some sort of pest control, although how big is one of the questions. I haven't mentioned them yet, but of course, let's see if this works, because I'm not sure if it will. Sorry. I may have ruined this for you guys. Oh, it's not working. Sorry, I kind of forgot that I had a video in it. If it doesn't work, it's okay, yeah? No, it's just that there's a video, but...
okay.
It's okay.
Image, no, not the video.
Yeah, in the original. It's okay. In any case... there we go. Yeah, so we will not be able to see this video, but soil organisms are also detritivores, so they feed on not just plant litter in forests, but also all sorts of crop residues. And through this feeding, they also bring a lot of the material that is on the top all the way down into the soil and make a lot of nutrients available for organisms that otherwise would not be able to reach them. So it is thanks to detritivores and bioturbators like earthworms or potworms that we actually get nutrients deeper into the soil. Sorry, I'm just gonna wing it. So most processes in the soil, I'm actually not sure why I can't see the presenter mode, but that's okay. So most processes in the soil cannot really be sustained without soil life, and this is an important thing to keep in mind. If you deprive a soil of what is alive, when we're thinking about how healthy is a soil, and when we talk about how healthy is a soil, we're trying to look at how much can a soil support ecosystem service for us. What we often forget is to actually measure the soil biodiversity. And in a research carried out by some of my colleagues, together with Elsa Bunnemann, they looked at, it's okay, you can take your time. They looked at how many times are people measuring different types of indicators. Yeah, I don't know what happened.
We have a technical problem. I don't know why. Okay. Sorry. I don't know why.
Let's see if it works with this one, because I think...
No, not work. Okay. Let me see. Okay. Click on okay. Thank you.
Oh, there we go. And actually, let me see one thing. Okay.
You need to, yes.
Yeah, it's okay.
Okay.
I think now I can find it. Thank you very much. No, I think I did it myself. So anyway, what I was saying is that some of our colleagues did a very thorough review, or relatively thorough review, to look at what is actually being measured when people are trying to understand soil quality or health. And what they realized, and you can see this in two ways. First of all, in blue you can see things that relate to soil physical parameters, in red soil chemical parameters, and in green soil biological parameters. And you can already see in number, soil biological parameters are way fewer. But you can also see in how long these bars are is how many studies actually looked at any of these biological parameters. And again, what you can see is that the bars are much longer in the soil chemical parameter measurement, which means many more studies included them. And yet soil biological parameters often get forgotten. And this is not just in scientific literature. Our colleagues at the EGP reviewed or looked into how member states are actually looking at soil health. And what they observed as well is that within all member states, these are all the indicators that are biological that are being studied. And then in different colors, you see different regions. And what you can see is, wow, 80% of the Western countries in member states are actually measuring quite a few of these biological indicators, but that's 4 countries. So, so all in all, I would say we have a lot to do to know and track the sustainability of our land practices, and to know really, are we losing this biodiversity, and is it going to lead to a loss of functions? And one of the reasons why we're not measuring these things too much is, well, on the one hand, they cost a bit more money than other measurements, but I think they say a lot. But on the other hand, it's because for many years we've perhaps been confounded by how diverse the soil is. There's too much. So our colleagues went about and really tried to look at who is doing what in the soil. And they put together this incredible amount of work, and I did very little of it. But basically what they did is they tried to think of soil-based ecosystem services that were important, particularly in agricultural soils. And here you have in blue water regulation and purification, nutrient cycling in purple, carbon and climate regulation in black or gray, and in green, disease and pest suppression. And for each of those, they started thinking about, okay, which processes are really relevant here, and what is the role of different organisms in those processes. So at the bottom here, you can see a really long list of processes, and above it, you can see sub-functions. So these processes sort of link together into common topics. And just to give you a very brief look into how this looks when you zoom in, here you can see what they put together for carbon and climate regulation. So you've got here the functions, the processes, and next to the processes you've got a really, really long list of different organisms and how they are related to these processes. In fact, if you take all the functions and the sub-functions, it ends up looking a little bit like this. So when we say soils are complex, they are. But basically here what you see in brown is all the different types of actors, and in beige you see the different processes. Now the size of the bubble has to do with how many times that organism came up in these models. So here really, really large you can see fungi and bacteria. That means that they are relevant to many, many, many processes in the soil. Now we know, and we've talked about this, and I think you heard a bit about this yesterday, that agricultural practices can have detrimental effects on soil life. But what's really important to keep in mind is that they can also be utilized to promote soil life. And I'll give you some examples. So these are all meta-analyses, which means that the trends found here are quite sturdy. And what we found, or what they found, I have nothing to do with these papers, is that no tillage or reduced tillage can have significantly positive effects to soil structure, which in turn can then continue to provide a better habitat for animals. Cover cropping can increase the soil organic carbon by an average of 15%, and it can also increase microbial abundance, activity, and diversity. And then, for example, organic fertilization increases bacterial and fungal abundances, but also the abundance of many other soil organisms. Now, if we know these things, the question is why aren't we doing them all around? And the answer is that these effects are not always so easy to quantify. So in the case of tillage, the effect increased quite strongly with latitude, meaning that the closer you get to the tropics, actually I think this should say decrease, the closer you got to the tropics, the less impactful stopping tillage was to soil organic carbon or any structure. In the case of cover cropping, the increase of soil organic carbon was higher in clay and temperate systems, meaning if you're in a sandy soil in Mediterranean climates, it may not be as impactful. And the difference there can be from 7 to 35%, so it's quite a large difference. And in the case of microbes, they observe lower effects in continental climates, not sure why, and in the case of conventional tillage. So if cover cropping was together with conventional tillage or chemical finishing of the cover crop, then these effects disappeared. And similarly, in the case of organic fertilization, the effect size depended on the crop to which fertilization was being applied. It depended on the texture and the climatic conditions of the field. So how do we move on from this, and how do we know whether the practices we apply make any sense at all? Through monitoring. If we monitor soil health, if we want to apply a new practice, we should be able to measure before and measure after. And of course the question is, what do we measure and how do we evaluate whether or not this has improved? An example of how we did this, and this is from a project that's already finished, called Landmark, is we looked, they built a set of models by which they could calculate 5 different soil functions. This model is available online, so you can check out what type of information is needed to use it. And this model requires information on soil properties, climatic information, and management information. And I did this for 52 sites, grasslands and arable crops. And what I was asking is, do these fields provide several functions simultaneously? And are there important, yeah, sorry, I see you now. And are there important trade-offs or synergies? But basically what we found is that there are important trade-offs and synergies, and that primary productivity is being provided at the expense of other functions. And this is true for grasslands and croplands. However, this is not so true when looking at the whole European region, where we could actually see that primary productivity is not always at the expense of something else. And here you can see positive and negative relationships between different soil functions being provided at high, medium, or low levels. Now, one last thing I will say is that what's also really relevant is that we don't always understand the role of climatic zone, and we think it has to do with what organisms are present where and how they carry out functions, but that these relationships between soil functions can change depending on where you are. And in this case, you're looking at the relationship between climate regulation and biodiversity and habitat provision, and you can see that in the Atlantic system, particularly in arable fields, you see a negative relationship. Climate regulation comes at the expense of biodiversity and habitat provision. But this is opposite when we look at Pannonian climatic zones. And I have to skip quite a bit because I assume and I hope partially because of all the management problems. But anyway, thank you very much for your attention, and I'd like to thank my colleagues as well for all the work they put in.
Thank you, Carmen. Thank you also for your patience with the technical issues.
No problem, they have to happen.
Thank you so much, and thank you for the good overview regarding soil health and biodiversity. So we will continue with our program, and I would like now to invite Silvia Castro. Dr. Castro, she is research at the University of Coimbra, and she will now present to us flowers, pollinators, and pollination, what we have learned through Cultivar about pollination service in Beira interior agro ecosystems. Sylvia, you have now the floor. You have around 10 minutes. And just to the others, keep in mind the two girls downstairs, they are giving us the time, okay?
So thank you very much for the introduction, and good morning to everyone. So my presentation will actually focus on the studies that we have been developing within the Cultivar project, in particular related with pollination services. Do I have to? So let's see. No enter? Okay. Aqui? Okay. So, just starting from the beginning, very simple. So, pollination is a very simple process where pollen grains are actually transferred from the male structures to the female structures, and for the plant, it implies the possibility to produce fruits and seeds, and for the animals, it implies food resource for themselves and for their brood. However, under our current scenarios, actually pollinators face major threats to their survival. From changes in the habitat structure, so with the habitat loss and fragmentation, climate change, increased use of agrochemicals, biological invasions, among others. And as we change the landscape structure and composition, actually the habitats suitable for pollinators decrease, and this impacts pollinator populations, reducing both their abundances and their diversity. More importantly, we also know that pollinator decline is linked and poses a menace to food security, as about 75% of our key crops used for human consumption actually depend on pollinators for production. And we also know that increased yield and quality of the crops are actually significantly correlated with pollinator abundance and pollinator diversity. However, the pollination is not so simple as just transferring the pollen grains from one side to the other. Actually, there are different components that impact pollination, and here we try to summarize it in this simple graphic. So first we have the intrinsic traits of the crop, which actually determine things such as the ability of the crop to self-pollinate. So we have a component that could be actually determined by that. Then we have a component that is linked with the available pollinator communities in the production site and determines the current production in a given site. And finally, we have the difference between this natural pollination, so what regularly occurs in the site, and an optimal pollination, which actually constitutes what we call a pollination deficit that is related with a deficit in pollination in pollinator communities that compromises crop production. Of course, these last two components are tightly linked with pollinator communities, obviously, and these in turn depend on local practices, as we have heard through the entire Congress, and on landscape structure as well. And within this scope, our aim within the Cultivar project was actually to produce knowledge, tools, and best practices to promote pollination services in the agro-ecosystems of this region. And for that, we structure the work in several steps, increasing the scale of the study at each step. So we first focused on sweet cherry as a key crop for the region, which actually depends on pollinators for production, and studied factors such as phenology, self-incompatibility, crossability, factors that are very important because they determine the quality of the pollen for successful pollination in a given production site. And with these, with the phenological observations, for example, we were able to study the behavior of many of all the cultivars from the cultivated in the area, their behavior in the regional edapoclimatic conditions, which is an information that is lacking for most of the producers that want to start new production sites. Additionally, with controlled pollinations, we were able to test their self-incompatibility. So sweet cherry is able to recognize the pollen and rejects pollen coming from the same clone. This means that if you have an orchard with only one cultivar, it won't produce. So you have to introduce pollen donors to guarantee if your cultivar is self-incompatible. So we test all these with self-pollinations, and we detected that most of the sweet cherry cultivars that are actually cultivated here in the region are self-incompatible, so they need to plant the orchard to have multiple cultivars for pollination. So we tested crossability between cultivars, and actually were able to identify multiple, let me see this, so good pollen donors and others that were better producers for sweet cherry. And all these together enable us to compile a set of indicators that are extremely important for farmers to plan and implement new sweet cherry orchards. In the next step, we try to scale up to the orchard and its surroundings, okay? And our first task was actually to identify who is the pollinators of sweet cherry. And then we characterize the pollinator communities in many production sites to understand how they vary. And at the same time, we try to relate it with the surroundings of the orchards, and found out that actually, and as it has been shown in the literature, both the abundance and richness of pollinators significantly increase as the percentage of natural and semi-natural areas in the surroundings increased, meaning that they work as sources of this functional group that then pays pollination services to the production site. Adding to that, we were not satisfied, so we also quantify local pollinator communities in 12 land cover areas around a sweet cherry orchard, mainly to understand the dynamics of pollinators related with local practices and also with existing habitats. Where are pollinators through an entire year? What habitats can enable us to protect them and have pollination services to our production site? And indeed, we were able to characterize both plant and pollinator communities, and here in these colors. And with these, we identify, for example, habitats like shrublands and waterlands that were very important for supporting pollinator communities, and at the same time we also identified management practices, for example in managed pastures, that create unfavorable habitats for pollinators throughout most of the entire year. Then we scaled up again, and we went to the Centro interior region. And the next step, we characterize pollinator communities in the same land cover, land use types, but scaling it to the 3 landscape units that you see here in the map, and to map pollination services in the landscape. And here you can see identified, represented as pollinator richness and pollinator abundance. And this enables us to identify hotspots of biodiversity in the region, as well as hotspots of where pollinator populations are actually depauperated and would need deeper practices to change it. Here, for example, so we identify habitats, and we also identify management practices, so general management practices that actually compromise pollinator communities in long term. The last step, we went globally, so and taking as consideration the previous graphic, so taking these 3 components of pollination, we actually performed the bibliographic search focused on crops worldwide. And with this, we intended to update a database that dates from 2007 that characterize what we call pollinator dependence value, which is the value that accounts for the contribution of pollinators to crop production. And this is very important because it's used worldwide to give economic value to pollinators. But it was outdated, it used categories, and it missed a lot of crops that are emerging and becoming important in our food system. So with this, we were able to compile a database with pollinator dependence values for 119 crops, including actually cultivars, so there are sessions that was completely neglected before. We had information for 35 new crops that was not listed previously, and with this we actually assess that 80% of animal pollinated crops depend heavily on pollinators, and actually more than 40% of our production is due to pollinators, okay? And this database definitely will enable us to accurately quantify pollinator contributions both at local and global scales. More accurately. And here you can just find some examples that are presented in the database. Finally, with this database, it's a huge resource, so we also compile, we wanted to quantify the extent of pollination deficits that we are actually facing today. So what is the percentage of production that we are losing because pollinators are not in health conditions? And we compiled the pollen crop database with this same bibliographic search. Goes almost 300 studies with more than 1,100 pollination experiments, and using 108 crops across 50 years and 62 countries. And these enable us to, again, calculate the production losses due to pollination services. And the main results, so very general, we know that crop production decreased by 27% due to pollen limitation, due to limitation in pollination services, and despite 28% of the entries show no pollen limitation, so no problems with the pollination services. The results indicate that over 20% of the entries actually reduced the production by half of its potential due to scarce pollinator services. And of course, all these bring us back to the need to develop local practices and manage the landscape in ways that are pollinator friendly, so that we can regain our pollinator populations and promote crop production to its maximum potential. So just to finish, I would like to thank to the Flower Lab team that was involved in the project, especially Katerina, which is doing the PhD in this subject and largely contributed to these results, and Hugo Gaspar, that it was a technician from the project, and not less important or even more important, to all the producers and landowners that enable us to work in their properties, producers associations and municipalities that were linked with this project. Thank you very much.
Okay, thank you so much, Sylvia. We'll give floor to the public after these two extra presentations that we have, if you don't mind. Okay, so we'll continue with Elsa Baltazar. She returns to the floor now for speaking about morphological, molecular, and genomic identification and characterization of Molinia fruticola in Prunus persica from Portugal, a different perspective from what she showed before. Okay.
Ok,
Okay, good morning everyone again. I'm here to present another work that I developed in the scope of Cultivar project. The title is Morphological, Molecular and Genomic Identification and Characterization of Monilinia fruticola in Prunus dispersia from Portugal. Stone fruit the stone fruits are distributed in all world, but as we can see in the figure, it's more representative in the North Hemisphere, being highly represented in Europe. According to the last data published by FAO, peach reached a production of around 4 million tons produced. In a national context, according to statistical Portugal, we can see that the central region it's the region that produces the most these fruits, namely peach and sweet cherry. Cover the Beira region, it's the region that contributes more for this data. The collaboration with local associations and farmers and producers, we found several issues such as decrease of peach production, increase of the affected areas, and increase of mortality in the orchards. This is some picture of the symptomatology observed in the field. We find different symptomatology according to different times of the year. We can see in the spring, in the blossom time, the floral abortion with the gummosis exudation. In the springtime, we saw another symptoms such as cankers, gummosis exudation, and the death of the plants. Several diseases were identified, diseases that affect branches and trunks, such as canker and dieback disease, leucostoma canker and bacterial canker. Other diseases that affect leaves, flowers and fruits, like brown root disease, peach leaf curl disease, powdery mildew and shot hole disease. But today I will talk about the brown root disease. This is a post-harvest disease. Is caused by three related pathogens such as Monilinia frutigena, Monilinia laxa, and Monilinia fruticola. The first and the second were already reported in our country, but Monilinia fruticola it's not reported until 2022, as we can see in the disease distribution map of the PPO. The disease is not present until 2022, but with this work it will change. The symptoms caused by these three related pathogens, it's very similar, so it's very difficult to identify which the pathogen is causing the symptoms, so we need molecular tests for do that. The questionnaires to the producers were very important. Here I have to mention the local associations were crucial to come to the farmers and do some questions to know about orchard characterization, disease reporting, the last disease, and disease characterization. These questionnaires were very important for the study area selection. The sampling was made in the north and in the south of Serra da Gardunha. The 13 orchards were sampled between 2021 and 2022, and a total of 46 samples were analyzed between these 2 years. For detection and confirmation of the presence of Monilinia fruticola, we followed the EPPO standards. And in this work, we have done two different approaches. One of them, the first one, the molecular identification of isolates based in culture-based methods, and the molecular identification from total DNA extracted from infected tissues. In the first approach, we have to come to the field and identify the symptomatic material, then we bring to the lab, we cut the symptomatic pieces, then we disinfect, and we put in the culture media, in specific culture media, the fungi comes out of the symptomatic material, then we culture this fungi until be pure, then we can do the morphological identification, and after that the confirmation by conventional PCR. Here we use the ETS region for the fungi identification, and after the isolates identification, we have done the whole genome sequencing, and we also did a pathogenicity test based in Koch's postulate to verify the infection of this pathogen. The other approach was similar like the other one. We cut the symptomatic pieces, then we macerate the whole wood, then we extract the total DNA from this wood extract. After that, we apply a real-time PCR with specific primers to detect Monilinia fruticula and discard the other Monilineas, and do the identification. Here we can see some examples of the symptomatic pieces after the disinfection, and in the other pictures we can see the maceration with the omics being prepared to do the total DNA extraction. And to give you some results, based on culture-based methods, we isolate 4 samples of Monilinia fruticula from 2 different orchards. We also do the morphological identification, as we can see in the picture. We can see the conidia formation and connected by chains. It's very easy to see in the microscope, and we have our fungi perfectly isolated. It's beautiful, but not so beautiful. We also did the phylogenetic tree with the ETS sequences. The ETS sequences belong to the same branch, but this was expected. Here in the pathogenicity test, we can see the infection. We did this pathogenicity test with 3 different fruits, with nectarine, peach, and sweet cherry, 3 fruits that are affected by this disease. And we can see the results after 7 days that the infection colonized the fruit. We also did the whole genome sequencing of one of the samples with the Oxford Nanopore Technologies. Here we collaborated with the CATAA institution for this work. And the Monilinia fruticula was also identified by real-time PCR in 9 samples from 4 different orchards. Here we can conclude that this method is more accurate to detect and identify Monilinia, because compared to the old culture-based methods, when we identified less samples with the presence of this pathogen. During this work, we have done several publications, such as in the 5th Symposium of National Fruticulture, and a publication in an international journal, Agronomy. And to conclude, Monilinia fruticula was detected for the first time in Portugal. The pathogen was detected in 4 orchards in Cova da Beira according to the EPPO standards. Real-time PCR detection confirmed the presence of Monilinia fruticula in a greater number of samples compared to the traditional culture-based methods, as I said later, and before. And the whole genome sequencing of the sample, one of the samples isolated, was made, and this work will certainly contribute to take appropriate measures to protect this sector from this pathogen. And the last, I have to thank to the institutions and to you all for listening to me again.
Thank you, Elsa. So we will go for the next presentation. Please, Ricardo Leitão. He is PhD student of the University of Coimbra from the Functional Ecology Center in the Terra Associated Laboratory, and he will present to us the Cultivar Soil project characterizing the health and the overlooked resource. Please.
Hi everybody. I'll switch to Portuguese since the translation is available. I think I'll be more effective with my message. So, estou aqui em nome da equipa do Solos, do projeto Solos, do programa Cultivar, e queria apresentar-vos um bocadinho o que é que foi este projeto, o que é que ele constou e de alguma forma perceber o que é que nós tentámos fazer aqui quando falamos em caracterização de um recurso que tantas vezes é esquecido e menosprezado e tanta importância tem no nosso dia-a-dia. Ok. O programa Cultivar, como toda a gente sabe, portanto, foi um programa integrado constituído por 22 projetos, se eu não estou em erro. O projeto Solo é o único projeto, é um dos projetos, portanto é o único projeto que eu saiba que foi totalmente, exclusivamente focado no solo, o que faz de alguma forma dele um projeto especial, digamos assim. Portanto, não menosprezando nenhum dos outros, mas de alguma forma acho que é um projeto que vale a pena ouvirem um pouco sobre ele. Antes de mais nada, vale a pena estudar solo? É uma das questões normalmente que se coloca. Será que justifica este investimento em algo tantas vezes tão sujo? Bem, de alguma forma é um recurso endógeno, o que está completamente dentro do âmbito do projeto Cultivar, até arriscaria mais, é um recurso endógeno genético, portanto, que claramente que está de acordo com tudo o que o Cultivar procurou fazer, está completamente dentro das prioridades da União Europeia. Portanto, nós temos, desde há 2 anos atrás, uma atenção cada vez maior da União Europeia e da Comissão Europeia na questão do solo, com esta Soil Strategy. Uma das 5 missões da Comissão Europeia é dedicada exclusivamente ao solo, e tivemos agora muito recentemente a publicação de uma proposta de uma lei de monitorização do solo. Portanto, estamos completamente dentro daquilo que é o objetivo e um tema quente para a União Europeia. E, por outro lado, a nossa saúde depende direta e indiretamente do solo. Portanto, isso é algo inequívoco e que se cada vez percebe mais, até numa linha desta abordagem de uma só saúde, e como as saúdes dos vários componentes do solo, dos animais, das plantas, do ser humano, estão tão interrelacionadas. Só que quando falamos em saúde do solo é preciso esclarecer o que é que estamos a falar, porque é um conceito relativamente novo. Até agora tem-se falado muito em contaminação do solo, portanto, a questão da qualidade do solo muito assente nos aspectos mais químicos e físicos, mas hoje em dia, portanto, o que se considera a saúde do solo é esta avaliação química, física e biológica da condição do solo, que determina a sua capacidade para funcionar e para funcionar como um sistema vivo e que produz serviços de ecossistema, serviços aos quais todos nós beneficiamos diretamente. E queria salientar aqui esta questão da funcionalidade, porque a funcionalidade e a saúde do solo estão interrelacionados, portanto são 2 pares que não podemos dissociar. E o que é esta funcionalidade, estas funções do solo? Elas estão bem identificadas, portanto falamos de composição de matéria orgânica, regulação do ciclo da água, portanto, produção de habitats, etc., portanto, que estão de alguma forma também identificadas com ameaças bem claras e que dão origem, estão no suporte do serviço de ecossistema da qual todos nós beneficiamos diretamente, como por exemplo a alimentação. Mas estas funções do solo, e nós sabemos quais elas são, na prática, e quando olhamos aqui uma perspectiva até um bocadinho mais científica, nós ainda estamos perante uma caixa negra em relação ao solo. Portanto, porque nós sabemos que o solo realiza certas funções, sabemos o que é que entra, o que é que sai, quem são os seus atores, mas não sabemos bem como é que os vários atores biológicos se relacionam entre si, como é que eles se relacionam com as faculdades químicas e físicas. Portanto, estamos um bocadinho longe de perceber esta funcionalidade. E então, neste contexto e neste ecossistema, nós, de alguma forma, começámos a pensar o projeto Cultivar. E um dos primeiros objetivos que se pretendeu com este projeto, o projeto Solo Cultivar, foi avaliar a saúde do solo em diferentes modelos agrícolas na região Centro, que é a região onde o cultivar, portanto, foi implementado. Para além disso, e aqui já entrando um bocadinho num aspecto mais ambicioso, procurou-se de alguma forma gerar conhecimento. Conhecimento em que contexto? Um conhecimento, para já, que pudesse ser integrado no cultivar e integrado com os outros programas do projeto Cultivar, portanto, dando resposta e complementando este programa, mas, por outro lado, é muito importante dar uma resposta, e tem todo o contexto do projeto Cultivar, dar uma resposta local aos nossos agricultores e aos stakeholders envolvidos. Para além disso, um dos objetivos foi produzir ciência, publicar, portanto, fazer apresentações, mas não esquecer que, e na ciência temos um problema, que os projetos de investigação têm uma durabilidade de 3 a 4, 5 anos, ou seja, muitas vezes acaba o projeto e cai muita da informação que se conseguiu obter. Então o objetivo era também que este cultivar serviço de base para outros projetos na região poderem começar a ser construídos a partir daqui e subir em termos de conhecimento e de graus. Por outro lado, contribuir de alguma forma para a nível local e regional, portanto os governos mais locais possam de alguma forma ajudar na planificação e na gestão a nível local, mas também ambiciosamente tentar de alguma forma influenciar positivamente a legislação nacional, a legislação internacional, como por exemplo a política agrícola comum e o nosso plano estratégico nacional, cada vez mais procura de alguma forma financiar por via de resultados e não por via de práticas. Portanto, ou seja, é claramente que há aqui uma ambição grande e alguma complexidade que de alguma forma levou a que o desenho tentasse dar resposta a essa complexidade. Então como é que na verdade, o que é que foi o projeto, ou o que é que está a ser, porque ainda não acabou o projeto de solo cultivar, ainda há muitos dados a serem processados. Portanto, ele focou-se na região, portanto, do programa Cultivar, de alguma forma muito próximo da região de Idanha Nova, portanto, e da própria cidade de Castelo Branco, portanto, e que é uma região dominada pela agricultura, dominada por vários sistemas agrícolas, portanto, e nós procuramos de alguma forma tentar incluir o máximo possível de sistemas agrícolas de representatividade. Portanto, focámos em 3 grandes culturas, portanto, o olival, as pastagens e o amendoal, e dentro destes tentámos subdividir em vários tipos, portanto, tentando sistemas mais tradicionais e mais versus sistemas intensivos, trazer o montado também para dentro da equação, pastagens melhoradas, portanto, irrigadas, e mesmo nos amendoais tentámos procurar diferentes modelos de intensidade com diferentes abordagens ao solo. Medir a saúde do solo não é fácil, portanto, nós no nosso caso utilizamos mais ou menos estes 14 parâmetros. Será que são precisos todos para medir a saúde do solo? Neste momento sim, talvez daqui a alguns anos se perceba que serão precisos menos, mas é tão complexo, ainda estamos tão longe de perceber, portanto, que é preciso uma grande quantidade de parâmetros, que se dividem, portanto, então nas tais vertentes química, física e biológica. Eu não estou a incluir aqui a erosão, mas já ontem o colega falou sobre isso. E então, pegando nestes fatores, neste conjunto todo de parâmetros, o que é que nós fizemos? Nós fizemos um desenho base experimental, portanto, baseado em transetos e em pontos, onde aplicámos onde medimos todos estes parâmetros e replicámos este desenho por todas as nossas áreas, no total de 24 sites, portanto, apanhando todas estas diferentes modalidades, o que de alguma forma nos permitiu e permitirá, porque ainda estamos, como eu digo, numa fase de processamento, ter este tipo de informação por camadas, portanto, de camadas biológicas, camadas fisicoquímicas, que são sobreponíveis e que nos dão uma ortogonalidade, que é um aspecto muito interessante e que nos permite ir mais fundo, ou seja, permite-nos entrar um bocadinho nestes modelos que a Carmen já falou há pouco, e que permite perceber a relação entre os vários agentes biológicos, como é que elas relacionam, permite-nos de alguma forma, permite-nos perceber como é que está a saúde do solo, como é que as diferentes práticas estão a impactar o solo, portanto, e de alguma forma revelar um bocadinho desta caixa negra. Então, os resultados expectáveis, porque a Marie, entretanto, vai apresentar alguns resultados preliminares, o meu colega Eduardo e a Luísa também já trazem mais resultados preliminares, portanto, o objetivo aqui é perceber, de alguma forma, como é que se processou este projeto e o que é que nós pretendemos conseguir. Para já, muito importante é tentar estabelecer, então, esta saúde do solo a nível regional, perceber o nosso contexto, como é que está o solo na nossa realidade, tanto de região centro. E depois perceber como é que, de alguma forma, estas práticas agrícolas estão a impactar esta saúde do solo, muito na lógica de tentar perceber como é que podemos ajudar a melhorar estas práticas e que estas práticas melhorem a saúde do solo e com isso beneficiam diretamente. Portanto, isto é claramente ambicioso, ainda estamos a perceber como é que vamos conseguir dar esta resposta. Um resultado que é claríssimo, tanto e que eu quero deixar aqui como talvez dos mais importantes, é esta rede de parceiros locais que foi criada. Portanto, esta rede de parceiros locais de agricultores, produtores, stakeholders, permite-nos de alguma forma conhecer o território, ter acesso aos locais, conhecer as particularidades, os problemas e de alguma forma enquadrar de forma completamente diferente e mais completa os resultados que vamos tirar. Portanto, isto é sem dúvida uma mais-valia que traz um aspecto que não posso deixar aqui de esquecer, que são as expectativas por parte dos produtores, que de alguma forma colocam uma pressão sobre quem está a estudar e que nos obriga de alguma forma a dar respostas e a ser até de alguma forma imaginativo e tentar perceber que somos parceiros do mesmo barco. Um resultado muito concreto, quero aqui deixar também, que acho extremamente relevante, que é a questão dos 2 horizontes da Europa que já foram aprovados, que vão trabalhar na zona, vão usar não só esta rede a cultivar, como dados que o cultivar já gerou e que de alguma forma vão permitir então elevar a fasquia, portanto, e passar para uma etapa acima em termos de estudos e trabalho. Deixar aqui um agradecimento muito especial a todos os produtores envolvidos neste projeto Solo Cultivar, a Igitânia, o Sr. Zé Sequeira, o Sérgio Torrão, a Generândia, a Vera Cruz, a Fundação Pina Ferraz e o Monte Silveira Biro. Um grande obrigado por todo o tempo, toda a dedicação, portanto, que eles tiveram para connosco, toda a paciência e espero que continuemos a trabalhar juntos durante mais tempo. E pronto, um agradecimento especial em nome pessoal a toda a equipa envolvida no SOL e a todos vós que estiveram aqui a ouvir esta apresentação. Obrigado.
Okay, thank you, Ricardo. We have quite a good amount of time for questions for the public, so I pass the word to you for questions for all our speakers, to whom we congratulate for the very nice presentations and for keeping the time. We are perfectly on time, okay? We have already 2 persons interested in making questions.
Thank you. Well, my research is more focused on food systems than specifically on soil, but I was a little bit curious because I've been also following this more agronomic approach, and none of you talked of one thing that I have seen in recent literature, which is disease in plants are many times related to nutritional deficiencies on the plant itself. And that comes from the quality of the soil, which you have all been talking about. So this is not a question for people A or B. I'm quite positive Marie is going to answer me afterwards. But just to bring a point that for having a healthy agronomic system, you really need a very, very healthy soil. And so if you have to focus on your practices, you should be using all the principles from regenerative agriculture and all the related principles from agroecology. And so this is the point I would like to try to make here, and I would like to have a comment of some of you regarding this situation. We have to change our agronomical practices in order to be regenerative, in order to improve our soil health, create biodiversified farming systems, and then the soil and the ecosystem will take care of the crop for us. And this is what I would like to hear from you. Thank you.
I can say a bit. No, I generally agree. I think we need to start treating our soils better. But I think what we need to keep in mind is that not every regenerative practice will have the same effect everywhere. And we cannot expect farmers to take in all the costs of trying, because some of these practices may not actually yield the same results in southern or center Portugal than they do in Northern Europe. So we really need to get better at understanding which. So cover cropping is really great. Which crops, which covers, or keeping the soil covered for as much of the year as you can. That's great, but with what and where, and where does it make sense for which crops? So there's quite a lot that's not as easily applicable, and we need to help farmers make those decisions better, and we ourselves need to understand the nuances of those decisions, because there are also quite a few trade-offs in those practices, where increasing, having a lot of input on your, or mulching on your soil can actually decrease other functions. So we need to understand a bit better as well. I'm not saying we shouldn't start, I'm saying we should monitor as we go. Anyone else? Do you want to add?
Can you just add? just to add a small commentary to what you said. Our focus and thinking now regionally, and in the scope that we work in Cultivar, I think one of the most important things is to improve the practices that are already implemented in order to improve the soil. Because it's talking now, I know ideally we should search for more ecological based agroecosystems, and that's obviously what we all work for. But socially, it's a difficult context, and economically and socially, and it's sometimes difficult to change, to make a big change. So I think working with everybody and trying to change the systems they are improving the soils and the systems that they are implemented now, I think would help in the future, and also working back with legislation and improving this funding, for example, the funding and the direction of the funds can help to do also to give a contribution for the transition. So that's because it's locally and with the socio-economical context we have, it's not so easy for farmers and for the systems, for local governments, and to do that change. So I think everybody should be on board. That's my opinion.
Yeah, can continue with more questions, Margarida?
Elsa, this is a question for you. You made the sequencing of the microbe was made, right? The sequencing of the microbe was made, right?
The whole genome sequencing of the isolates that we was made.
But what was the idea of this sequencing? What are you planning to do with this afterwards?
For now, nothing. But we sequence because it's never been made. So we have win some information with that work.
Because I don't know if there is some in these databases, some information about the virulence, because this is not my area, but that would be very interesting.
Yeah, it's the first isolate sequenced in Portugal, so we can compare it with the others in the other countries.
Thank you very much.
Não pode voltar. Posso fazer? I will do 2 questions, small questions, regarding because you said that the DNA analysis is more effective than the traditional cultivation in laboratory. Looking for the producers' views and what's more suitable. How is the balance on the time taken for both methods and the price of it? Is it available for?
Yeah, it's more cheaper the molecular one, because the culture-based methods we spend more material and it's costs more. So the molecular, it's the best one, and it's the more accurate one, because we detecting more samples compared to the other methods, and we have done the 2 approaches to the same samples. Yeah.
So we have time for one more question, or I don't know if Joana wants to add something. And José Paulo Souza there. And I want questions from people who I don't know the name.
Desculpem, é só ajudar a Elsa a clarificar. A monilinia fruticola é um organismo de quarentena, regulado de quarentena. Portanto, obriga-nos a ter condições especiais nos laboratórios, sermos reconhecidos pela Direção-Geral da Alimentação e Veterinária para podermos trabalhar com este organismo. E a detecção molecular que ela fala é porque nós temos que fazer a validação do método no laboratório. É possível fazer das 2 formas, são as 2 validamente descritas. O que nós sabemos hoje é que, em termos de detecção, a sensibilidade do método de RT-PCR é mais elevado. A isso juntamos o facto de ser mais barato e juntamos um tempo de resposta muito mais rápido. Portanto, o método já está completamente disponível para já está implementado, portanto, qualquer produtor que tenha este problema pode rapidamente consultar-nos, que nós ajudamos a fazer detecção. Qual é a relevância disto para a região e contexto nacional? É que a monilinia fruticola tem impacto nas exportações, sendo um organismo regulado de quarentena e, portanto, em termos de exportações, há países que não aceitam zonas em que a doença esteja presente. Portanto, isto em termos de segurança alimentar e em termos de exportações para a região vai ter impacto, até em termos de regulação e de restrições de áreas demarcadas, etc. Daí nós termos falado na monilinia como um caso importante na região, porque se nós encontrarmos depressa, rápido, conseguimos limitar o foco, diminuir a propagação. Era só um esclarecimento. Obrigada. Parabéns, Elsa.
If you can pass the micro to... And there is another question from a person who I don't know the name.
Thank you. So it's a question for Carmen. Thank you, Carmen, for your presentation. So my question goes, so you showed in one of your slides when you compare the different types of indicators, physical, chemical, and biological, that in biological we are far behind. So my question goes to the future. So with the new soil monitoring law, so what is your opinion? So what could be our expectations that we fight a little bit for biological parameters to be included? How optimistic could we be?
Well, I think we can be optimistic that some biological parameters will be included. I think we are going to have to fight to make sure that they are indicators that mean something. And I think that's going to be the fight is, or that what we're going to have to ask for is to make sure that we ask for indicators of function and less so indicators of biomass and diversity, and make sure that the indicators of function relate to the climatic and pedoclimatic zones that we're working with. So perhaps instead of measuring earthworms everywhere, we can start thinking about measuring the bioturbator community, and there try to look at the impact of those on the functions that they relate to. But I think we're going to have to make noise to make sure that happens. Yeah. Absolutely. Yeah.
Just a very final question before we move on to the 3 short presentations.
Yes, hello. I would like to thank all of you for such great presentations. I have a lot of questions for Elsa, because I'm in love with fungi, but I will speak with you on the next coffee break or during lunchtime. So my question is a very short question to Carmen also, about soil probiotics. I mean, do you think that we are close to commercializing soil probiotics, and what would be the relevance of using these products? Thank you.
I think they're already being commercialized without us really knowing how much of an actual effect they have. I'm not sure. So it's not my expertise, but soils are quite complex. So how much of what we add to the soil is actually kept in the soil is a big question. And I think we still have a bit to go until we know for sure that what we're adding to the soil stays in the soil and actually aids in any way. We do have a bit of evidence in nature where they use natural soils to promote restoration of soils, so that can work as a sort of primer, but I think there's still quite a bit of work. But if someone knows more, please feel free.
Thanks. Okay, thank you, Carmen. I ask you a round of applause for our speakers. And we move on asking Hugo Gaspar, Eva Garcia, and Rafaela will be replaced by Marie, who is already on the floor with us, so that we continue with these 3 short presentations before lunch. Okay, we'll continue our session with the presentation Expanding the Knowledge on Pollinators and Homofauna in Beira Baixa through Ecology-Based and Taxonomy Studies from Hugo Gaspar, who comes from the Center for Functional Ecology of the University of Coimbra.
Acho que não. Acho que tem que ser na outra a seguir.
Vamos só deixar.
Ok, muito bom dia a todos. Eu venho falar um bocadinho sobre o contributo que nós fizemos no projeto Cultivar para a entomofauna de polinizadores na Beira Baixa, um bocadinho em ligação com o que a Sílvia acabou de apresentar. Vocês aqui podem... Vocês já têm noção de, como a Sílvia falou antes, da importância dos serviços de polinização. Os polinizadores, como os vetores de polinização, têm um papel bastante importante, e aqueles números que a Sílvia apresentou há pouco dos 90% das plantas selvagens e 75% das culturas, são estes os valores que são influenciados pelos polinizadores, os animais polinizadores. E quando falamos deste grupo, estamos a falar de 3 principais grupos, que são as abelhas, os sirfídeos ou moscas das flores e as borboletas diurnas. Em Portugal estamos a falar mais ou menos de 1000 espécies, um pouco mais de 1000 espécies. No entanto, o conhecimento que temos sobre estes grupos é bastante restrito. Estes são o nível de ameaça a nível europeu. Temos ali uma grande falha, principalmente no que toca às abelhas. Mas se formos olhar para o contexto português, não existe este tipo de dados. Ainda estamos numa fase de tentar perceber onde é que eles existem, quais é que são as espécies que existem e qual a sua distribuição. E neste sentido é necessário compreender quais é que são os habitats que eles precisam, não só para a fase adulta, mas para completar todo o seu ciclo de vida. E nesse sentido nós fomos para o campo e um dos principais métodos que utilizámos foi amostrar as comunidades de polinizadores nestes habitats. Um dos principais métodos é a amostragem com redes entomológicas em transetos aleatórios, que depois leva à preparação dos insetos em coleções onde podem ser estudados. E então nós olhamos para a paisagem da região do projeto Cultivar e começámos por selecionar um local, como a Sílvia falou há pouco, em que fizemos o tal trabalho à volta de uma região de produção, em várias tipologias de habitat diferentes, e fizemos cerca de 545 horas de amostragem durante um ano inteiro, o que é um esforço bastante grande, tendo em conta a comunidade de polinizadores, e depois estendemos isto para as três das principais biorregiões desta área, também com estas tipologias de habitat, em 50 horas de amostragem, em 2 momentos no ano, em 2022. A isto acrescentamos aqui então aquele local e mais 2 locais que acabamos por estudar noutros trabalhos, e acabamos por mostrar um pouco por toda a área os habitats que existem em toda a região. Para terem noção em termos de contributos que nós fizemos para este conhecimento básico dos polinizadores que sustentam toda esta função de serviços de polinização, nós contribuímos com 2.400 registros de abelhas. Antes existiam 771 registros na Beira Baixa e Beira Alta. Fez com que a Beira Baixa, por exemplo, se tornasse a segunda província com mais registros. Antigamente era a penúltima. Depois, em termos de sirfídeos, fizemos 172 registros. Não temos conhecimento dos registros anteriores, neste caso. Isto também se traduziu em 177 novas espécies de abelhas conhecidas para esta região. Antigamente conheciam-se 134, mais do que duplicámos as espécies que se conheciam. E para a Beira Alta, 73 novas espécies de abelhas, onde se conheciam 193. Em números gerais, nós contribuímos com 12 espécies novas para a fauna portuguesa, encontramos pela primeira vez, entre abelhas e sirfídeos, incluindo aqui uma espécie nova para a ciência e também as espécies novas para a Península Ibérica. Os principais outputs que nós tiramos deste trabalho foram a publicação de 2 listas de espécies, e depois estamos em preparação ainda dos restantes dados, não só aqueles que são tirados diretamente à análise da taxonomia destes insetos, mas também da parte de ecologia, e estamos a falar aqui da produção de uma base de dados de barcoding das abelhas, que vai ser publicada em breve, uma lista de espécies de sirfídeos e também 2 trabalhos de ecologia para o mapeamento dos serviços de polinização e também para a dinâmica anual dos polinizadores ao longo de um ano inteiro. Isto aqui serve então para mostrar que o grande potencial do investimento regional e nacional que pode ser feito para aumentar este conhecimento de base, que é o conhecimento sobre as espécies de polinizadores que temos, depois vai traduzir nos serviços de polinização e na produção agrícola. É isso. Obrigado.
Thank you, Hugo. Eva, please, you can have the floor. Eva is PhD student of the University of Coimbra, and she will present the work that's linked with the Pedro Nunes Institute Laboratory of Phytopathology.
Obrigada. Bom dia. Eu carrego aqui, acho. Ok. Então, com as alterações climáticas, o transporte de plantas e o modelo atual da agricultura, as doenças das plantas têm vindo a ter um impacto cada vez maior no caminho para uma agricultura mais sustentável, tanto do ponto de vista económico como também ecológico. Assim, o diagnóstico destas doenças é cada vez mais importante para que possam ser adotadas as medidas ideais para mitigar o impacto destas doenças. Assim, Desculpem. No entanto, este diagnóstico apresenta uma série de desafios aqui, como por exemplo estes que estou a enfrentar agora. Pronto, por um lado, é a emergência de novas doenças. Cada vez mais ouvimos falar disto, cada vez mais temos novas doenças a aparecer. E então isto faz com que a quantidade de doenças que nós temos são cada vez maiores. Também a grande diversidade de agentes patogénicos que podem causar estas mesmas doenças. Para além disso, a sintomatologia não é nada específica, portanto, o mesmo sintoma pode ser causado por uma diversidade grande de doenças. E também, uma doença pode ser causada por mais do que um organismo, o que isto pode dificultar muito num diagnóstico, porque nós podemos dizer, ok, esta doença é causada por este organismo, e afinal não, é um complexo de organismos. E também, claro, a urgência em termos destes diagnósticos, porque os produtores precisam de saber o mais rapidamente possível o que é que realmente se está a passar com as suas culturas. Para isto, na nossa proposta é a adoção de uma tecnologia recente, a sequenciação de cadeia longa, neste diagnóstico. Com esta técnica podemos, com a extração de DNA, amplificar genes marcadores universais para bactérias e fungos e sequenciar estes genes. Isto pode permitir a identificação de grupos de organismos potencialmente patogénicos e assim direcionar as técnicas para a detecção dos organismos responsáveis pela doença. É muito difícil no contexto de laboratório aplicarmos tentarmos encontrar todos os organismos potencialmente patogénicos pelos métodos clássicos. Isto pode fazer com que o diagnóstico seja mais sensível, mais rápido e mais eficiente. Nós atualmente já estamos a iniciar este teste, a testar esta metodologia com um projeto que é o Chill Out, que procura diagnosticar as doenças presentes em prunos na cova da beira. Se quiserem saber mais, podem também ir ver o nosso póster. Muito obrigada.
Thank you, Eva. We'll have our final presentation. It will be Marie Barthes who will speak with us about farmers' perception of soil fauna and their ecosystem services.
Então nós vamos apresentar, vou apresentar em português também, para facilidade e para pegar o gancho para todos os apresentadores, e a gente vai apresentar, porque entra um pouco no gancho do que a Carmen, do que o Ricardo apresentaram da importância da biodiversidade do solo. Então é um pouco da percepção dos agricultores em relação à fauna do solo e aos serviços ecossistêmicos. Esse trabalho que eu vou apresentar é um pedacinho de um trabalho maior de questionários e inquéritos que foram aplicados, que foi liderado pela professora Paula Castro, e a gente vai apresentar rapidamente porque o tempo é bastante curto para a gente conseguir apresentar. Eu não vou entrar em detalhes da complexidade do que é da biodiversidade, das funções que eles colocam, que também já foi colocado pelos outros apresentadores, mas é a importância, né, como os comentários também que tivemos, que a vida do solo é ela que move todos os processos químicos e físicos, e isso que move a nossa vida terrestre, né? Então tem uma importância muito grande, e é importante que a gente tenha essa percepção e consiga entender melhor. E, nesse sentido, a nossa ideia era conversar com os agricultores para entender como eles manejavam, né, e entendiam a biodiversidade do solo. Nesse pedacinho desse questionário foram, então, 13 questões aplicadas que foram respondidas por 65 agricultores da região da Beira Baixa. Nesse questionário, a gente chegou a listar, quando a gente fala de fauna do solo, a gente está se referindo à macrofauna, são organismos visíveis. Então, é um grupo de organismos que, teoricamente, todo mundo consegue perceber e avaliar no seu sistema. Então, essa é a lista dos 13 organismos que nós listamos para os agricultores, e daí tinha uma série, né, de opções deles marcarem o que consegue, o que não consegue observar, e o que é conhecido por eles. E, num primeiro momento, né, com mais de 90% desses questionários respondidos, os 3 grupos principais, 4, desculpa, que tiveram um outstanding, né, que tiveram a principal percepção, são as minhocas, que, em geral, são um indicador universal de saúde do solo, as formigas, as aranhas e também os grilos, como os 4 grupos que foram percebidos como presentes no ambiente deles e reconhecidos pelos agricultores. E como, né, como que eles percebiam também essa biodiversidade nas áreas deles, se ela era alta, se ela era baixa, se era mediana. Então, dos 65 entrevistados, 9% deles, eles consideraram que a biodiversidade do solo deles é baixa. 45% mediana e 34% que a biodiversidade é alta. Relacionada a cada um desses grupos, quem disse que era baixa biodiversidade, 100% deles eram produtores convencionais. Tá? No mediano, 58% tinham produção em policultura. Claro que tinham outras categorias, tá? Mas essas aqui são as principais que se destacaram. E com alta biodiversidade, eram agricultores que tinham sistemas integrados de produção e, né, um pouco contraditório, eram agricultores também que tinham monocultura. Daqueles 3 grupos, né, principais que foram os mais avaliados, e daí como eles percebiam que esses grupos poderiam contribuir para os serviços ecossistêmicos, que foram listados pela Carmen, pelo Ricardo. Então, as minhocas, como tradicionalmente conhecido ali, né, 54% identificou como a infiltração e estoque de água no solo, o processo de decomposição da matéria orgânica e formação do solo. As formigas estando relacionadas com a dispersão e consumo de sementes, e as aranhas com a proteção, ou seja, com a regulação ali contra as pragas e doenças. O que a gente percebeu, de fato, é que houve uma dificuldade dos agricultores relacionarem a função do organismo com o seu serviço ecossistêmico. Então, a gente tinha um quadro que tinha todos os serviços ecossistêmicos e os 13 grupos relacionados. E houve uma dificuldade, ou seja, o que a gente conseguiu perceber, que os agricultores, eles sabem que a biodiversidade é importante, mas para muitos dos grupos, né, considerando todos os outros grupos, eles não conseguem relacionar para que serviço ecossistêmico eles estão presentes no ambiente. De uma outra pergunta também que se destacou era quais as práticas que na percepção dos agricultores influenciava na variedade, na densidade dos organismos. Então, para a redução da biodiversidade, acima de 71% ali a aplicação de pesticidas, o uso de fertilizantes químicos e o preparo do solo como os 3 fatores que mais impactam a percepção deles para a redução da biodiversidade. E para o aumento, acima de 80% a adição de esterco e de compostagens, a cobertura vegetal também como um item importante e o uso de coberturas que ajudam a aumentar essa biodiversidade. Quando a gente perguntou também se eles, qual que era a preocupação deles com o ambiente, né? 66% disseram que sim, que era importante, que precisava ter um cuidado. E na sequência, então, a gente perguntou se eles tinham a intenção de mudar as suas práticas agrícolas para beneficiar a biodiversidade e ter esses serviços ecossistêmicos em benefício a eles. 77% disse que sim, eles, né, gostariam de mudar. No entanto, se tivessem recursos disponíveis, fundos ou subsídios para aportar isso, e que também fosse garantido a produtividade deles nos sistemas agrícolas, né? Então, tem um contrabalanço de eles tentarem fazer essa mudança No entanto, precisa ter algum suporte e precisa a garantia da manutenção da produção deles. Diante, né, de algumas dessas das percepções que nós tivemos, entre as soluções que a gente percebe, eu acho que essa é a nossa responsabilidade como pesquisador, é a gente traduzir essa informação que a gente traz científica para o agricultor e para a sociedade, né? Então, é a gente aproximar, conversar, e não são, aqui nesse caso, eu não fecho apenas para os agricultores, mas é para toda a nossa sociedade, mas principalmente o setor educacional, porque são as novas gerações que vão fazer o nosso futuro e vão trazer as mudanças. Então, a gente trabalhar esses conceitos que a gente está discutindo aqui, levar de uma forma simplificada e entendível para toda a sociedade, para a gente consiga fazer a transformação dessas mudanças que a gente espera. Basicamente era isso, né? Queria agradecer ao Programa Cultivar pela oportunidade, das entidades que eu sou vinculada também ao grupo, e depois também, claro, a gente pode estar disponível a ter mais, discutir um pouco mais sobre o assunto. Obrigada.
Okay, many thanks, Marie. I think, I don't know if we have time for just a quick question. 1, 2 questions, so please raise your hand if you have any questions for Marie, Hugo, and Heva. Or everybody wants to make a break. Okay, so I think the organizers are coming, so I think they are expelling us from the floor. Any questions? If not, we will be here. Okay, there's a quick question from a person who has a lot of questions. Yeah, thank you so much.
Hello, my question is for Elsa. No, it's not Elsa, I'm sorry. Eva. So it was a similar name, I'm sorry. I also have a lot of questions for you, but I will only make a small question. That is, if you are planning, or if in your study, do you think it's possible to identify both fungi and bacteria with your technique?
Both what?
Fungi and bacteria, yes.
Sorry. Yes, it's the plan. Yeah, and if you want, you can go to our, well, you can talk with me, of course, and we can go to our poster because we have already some results. Thank you.
So we are closing this session. I ask you for a round of applause for our 3 quick speakers. And the biodiversity survey and the ecosystem service will continue in the afternoon. Okay, with second round. Christophe.
Thank you, João. Thank you, Marie, and thank you, Hugo and Eva. I just have some small announcements for the program. I want to tell you that yesterday, since we had a bit of a delay, we didn't do the poster session as we should have done, so we will do it today with the other teams, selected teams, and we will also give the poster prizes on that time. I want to tell you to please go also to if you are interested, of course, to the other workshops. There will be a workshop of Centro and also a workshop for Innov Cluster. And I also wanted to tell you that you please also visit our sponsors. They have things to offer you, things that will help you, and please have some time to meet them and learn what they can help you with. So first of all, I would like to thank Fruit Control Equipment for the sponsor. They are going to give a workshop on fruit and vegetable conservation this afternoon in 2:30 in the other building next door at CCCCB. So it's this cultural center here in Castelo Branco. I would like also to thank Schreiber. They will be here in the main session this afternoon. Also the municipality of Fundão, that they have a small place upstairs, that they have a center for the technological center for agriculture. and they can tell you more about it. And we have other sponsors that are in this room next to us. Please visit them and know what they have to offer. And now I would like to invite Nanta, which is one of our first sponsors of this Congress. I would like to call, let me remember the names, José Belo and João Mateus. They will make a presentation on a project that they are working. You can see a project that it's done by a company that also it gives a sustainable future for our agriculture systems. So, thank you, José Belo and João Mateus. They will present themselves. After this, we will have a moment from ANIPLA that I will also announce them. They have a smart platform for agriculture that they will show us today. Okay, thank you.
Good morning all. If you may allow me to do the presentation in Portuguese to be faster, so we can go lunch. O meu nome é João Mateus, eu trabalho para uma empresa que se chama Nanta. A Nanta pertence a uma multinacional que se chama Nutreco. A Nutreco está presente nos 5 continentes e está muito envolvida com a parte da nutrição animal. E opera com a marca Nanta dentro da Península Ibérica, onde nós temos e trabalhamos com 22 fábricas de fabrico de alimentos compostos. Mas para além disso, a Nanta tenta ir um pouquinho mais longe e tenta se enquadrar neste novo mundo e nesta nova realidade que nos estão a pedir, que realmente será produzir... deixa eu ver para perceber como é que isto passa. Ok. Portanto, enquadrando aqui um bocadinho no tema deste Congresso, que desde já tenho que agradecer a toda a organização pela presença que aqui temos. Vou-vos tentar aqui transmitir um projeto que a Nanta tem, desenvolvido ao longo de vários anos, em que temos um projeto de produção sustentável. Dentro da apresentação, vamos fazer o porquê, explicar o porquê que aparece a necessidade de criar projetos como estes, quais é que são os pilares deste projeto, como é que ele se aplica numa forma prática dentro das explorações, dando um exemplo de uma exploração, quais os resultados que obtivemos e as conclusões a que conseguimos chegar. A Europa tem-nos dado vários guidelines para tentar tornar o mundo um pouco mais sustentável e mais saudável. E o que estamos aqui a debater neste Congresso realmente é o Farm to Fork, que não é mais do que fazer uma produção saudável, sustentável e amiga do ambiente. Dentro da estratégia do Farm to Fork há vários parâmetros. Nós vamos aqui principalmente envolver neste projeto em 2, que é na parte do bem-estar animal e no registro sustentável de dados para posterior utilização e partilha dessa mesma informação. Há aqui um outro aspecto que também podemos considerar, que será a parte dos apoios à produção, que o que nós acreditamos e que se preverá é que as explorações que estejam enquadradas dentro deste tipo de projetos terão mais apoios no futuro. Serão estes os 3 presentes. Este projeto está baseado em 4 pilares fundamentais. A sustentabilidade ambiental, que é disto que falamos muito e toda a gente fala, mas também queremos dar, também importância à sustentabilidade económica das explorações e à sustentabilidade social. Sendo que acrescentamos um 4º pilar, muito importante para nós e que já trabalhamos há cerca de 20 anos, onde temos muitos ensaios e muitos registros feitos, que é o bem-estar animal. No pilar da sustentabilidade económica, o que nós criámos foi 12 parâmetros, 12 critérios medíveis para poder identificar a rentabilidade e eficiência de todas as explorações, dependendo se são explorações de leite ou explorações de carne. E isto porquê? Porque há necessidade de enfocar os objetivos dos produtores da maneira mais eficiente, de modo a que possam ser rentáveis, trabalhando de uma forma sustentável, com a economia circular. Dentro da parte do pilar do meio ambiente, o que fazemos é medir e quantificar as emissões de gases de efeitos de estufa, para além de quantificar também o estrume e o fósforo libertado para o ambiente. Aqui tentamos focalizar bastante a parte genética, que é importante. Quanto mais eficiente for o animal, menos pegada vai deixar neste planeta. Dentro da sustentabilidade social, o que pretendemos é saber realmente o grau de conforto e satisfação dos funcionários numa atividade que é penosa, que é diária, dentro das explorações, em que medimos o tempo que o funcionário também trabalha, mas o tempo que também tem que descansar, dando principal relevância à formação contínua dos próprios funcionários, de modo a poder evoluir dentro da sua profissão e com melhores práticas de maneio. Também quantificamos a parte das instalações e de todos os equipamentos utilizados para facilitar o trabalho do dia-a-dia dos funcionários. Como é, por exemplo, um exemplo de um carro de distribuição de concentrado, que é um trabalho muito braçal, a utilização de videovigilância, para não ter as pessoas tanto tempo presentes dentro das explorações, podendo lançar alertas se quando necessário, fazendo instalações que permitam a distribuição de forragem, que é realmente um trabalho muito árduo e de difícil manuseamento, que seja mais simples no dia-a-dia para os funcionários. Outro pilar do bem-estar animal. Aqui estamos envolvidos há cerca de 20 anos, onde fazemos registros de várias magnitudes, dentro do aspecto do ambiente, do manejo da exploração, das instalações, dos equipamentos, da própria saúde dos animais. Aqui temos cerca de 500 auditorias feitas, as explorações de ovelhas de leite, ovelhas de carne, cabras de leite e engorda de borregos, que foram feitas e elaboradas em 260 explorações, das quais também já saíram alguns trabalhos científicos e aprovados. Queria-vos mostrar esta foto para poderem ver que é possível produzir com conforto animal, onde os animais estão confortáveis, com o espaço suficiente, com o arejamento e com todas as necessidades que necessitam para poder fazer a produção sustentável e com o bem-estar animal em consideração. Outros aspectos que é quantificado, e gosto muito desta fotografia, é que todos os animais deverão ter sempre acesso à comida. Depois desta avaliação dos 4 pilares fundamentais deste projeto, o que se criou foi um decálogo de boas práticas de trabalho, em que são avaliadas dentro das explorações em vários parâmetros, desde a nutrição, à saúde, ao maneio, ao controle, à gestão, são quantificáveis para poder comparar com a nossa base de dados e com aquilo que é dito de normal de se ter dentro da exploração. Apresento-vos uma fotografia do Centro de Ensaio e de Investigação, onde nós fazemos os testes com aditivos nutricionais, de modo a aumentar a rentabilidade e, inclusivamente, a baixar a própria pegada ambiental dos animais através da menor libertação de gases de efeito de estufa. Queria-vos mostrar também esta fotografia, que é possível climatizar os animais de modo a terem mais conforto, muitas vezes, do que nós na própria rua. Esta exploração é muito perto aqui, de Castelo Branco. Eu já tive a oportunidade de estar dentro dela com praticamente 50 graus na rua e dentro da exploração não chegava a 30 graus. E, para além de refrigerar o ar, também, como é forçado o ar, também acaba por fazer a limpeza do próprio ar. Este aqui é mais um exemplo da preocupação que se deve de ter dentro das explorações. São extratores de amoníaco que existem nas explorações, de modo a que os animais não estejam com tanta intensidade dentro das explorações com concentrações elevadas de amoníaco. Agora vou passar a palavra ao meu caro e amigo Dr. Chema Velho, a.k.a. Chema Velho para os amigos, que vos vai mostrar a parte prática deste projeto todo. Muito obrigado.
Okay, thank you very much, Joao. Because the idea is the goal of this project is to measure, to evaluate a farm from the point of view of the sustainability in practical conditions in a farm. What the way to work in this project is doing this, developing this action in 3 steps. The first one is the emission calculations calculated by the IPC equations 2006, nitrogen, phosphorus, methane, carbon footprint, manure management, and carbon sequestration. The second one is the evaluate and assess really the farm from the as sustainable point of view. The farm could be measuring the 4 pillars described by Joao of sustainability, economic, environmental, social, and animal welfare. It could be, depending on some criteria established by the model, the farm could be evaluated in firm A, B, or C. And the 3rd step is to assess and to measure the degree of improvement of several best practices disponible, BPD, classified by a decalogue. You can see here nutrition, animal health, animal welfare, zootechnia, environmental, biosafety, waste, circular economy, labor, and planification and control. So, in order to become certified, to get the certification, a farm should do the 3 steps. So I'm going to show you several information that you can obtain by the model. For example, this is a real farm in dairy goats. For example, this graph, this table, the table over there, shows the CO2 equivalence for the different categories of animals in the farm: the lactancy, the growing up, the fattening, the males, replacement, adults, and the summary of all categories. In the graph on the right, you can see here the split and the importance of each kind of animal depending on the emissions. And the graph below is at farm level. It means it keeps into account not only the emissions of each category, but also the amount of animals in each category. And then the table above, below, is an interesting information about the emissions by farm and year, and also per kilo of milk produced, or per kilo of meat produced, depending on the kind of farm, and also per kilo of protein produced. Okay. Concerning the qualification of sustainable farm, this is a real case. The farm qualification is A, because this farm accomplished the 4 pillars. You can see here the threshold is in blue and the score obtained in the farm is in orange. And you can see that all of them have been accomplished. Consequently, the farm qualification is E. On the table below is an example of how the model detects what are the outputs obtained in the evaluation, in the audit, to be improved. In this case, for example, in the social aspect, the job rotation and the labor load should be improved in order to obtain, in order to get an improvement. So, besides, the model has set up a set of more or less close to 60 best practices disponible, BPD, in order to improve the sustainability in the farm. And we have done some documents and some guides in order to help the farmer to improve to implement these good practices. For example, when you do the audit, we obtain a profile in the farm in sustainability, a profile depending of the degree of accomplishment of every aspect of the decalogue, and also the profile of the farm depending on sustainability, social, environmental, and economic. In this case, is really balanced, as you can see. Another topic we have developed in this project is the evaluation and the certification of our feed, our commercial feed, depending on through a life cycle assessment. We evaluate the footprint, the environmental footprint of our feed, keeping into account the origin of the raw materials, the transport of raw materials from the origin to the farm, and then the transport from the farm to the from the factory to the farm. So this life cycle assessment has been certified, And also we have certified the equations in order to calculate in our company, to calculate in all ruminants feed that we market, to calculate the footprint, the environmental footprint. I mean climate change, carbon footprint, also eutrophication, acidification of water, water footprint, and so on. Well, these are the results obtained in our work we published last year in an international congress in Spain involved in small ruminants. These are 78 audits in small ruminant farms, and these are the emissions, the results of the emission for dairy goat farms, fattening lambs, and dairy sheep ones. In this case, the most relevant issue is that we have obtained several values, several reference values, in order to compare the real audits and the real results obtained in a determined farm. We can compare the results versus the reference values. We have obtained not only reference values, as you can see here, but also the variation, in order to know if an emission obtained in a farm is inside the range of the variability set up by the study. So concerning sustainable farm qualification, as you can see here, these 78 audits, close to the 60% obtain or get the classification A. Okay, is a good news. As you can see here, for example, concerning economical pillar, the dairy farms are the best score. Concerning environmental pillar, the dairy goats ones are the better score. And concerning social pillar, the fattening lambs ones. Regarding animal welfare pillar, you can see we obtain really high standards in all cases. So, the idea is to evaluate and to look at the evolution of a farm. Our model shows if the farm is improving or not is improving, and what aspects or what pillars of sustainability should be improved. And concerning PPDs or sustainable good practices implementation, as you can see here, biosafety and circularity are the lowest are the aspects that it is necessary to be improved. So we have developed for several years this model in Spain and Portugal, and we are really proud on the results. For example, in this example, you can see here several farms, and we put pay really attention in the role of the women in the farms. The women are really in all cases are really involved and are really aware of the importance of the sustainability in order to do is farm better from this point of view. And this we are really proud also with this farm is a first Portuguese farm in dairy goats that have got the classification, the certification in Granja Circular. I think this evening there will be a presentation about this farm, and you can obtain more information. So, as a conclusion, the Granja Circular is a model to evaluate, to certificate, to assess farms from the point of view of sustainability, but also it means it allows the possibility to improve each parameter, each pillar of sustainability. The second one is it allows us also to obtain reference data from emissions in commercial and in practical conditions. Another conclusion is that the study of a sample of livestock company has allowed us to make an approach to obtain a sustainability profile of the three subsectors studied, but also the model is available for monogastric species, for ruminant, and we are developing right now in monogastric ones. And the degree of implementation of good sustainable practices is quite high in general, with biosafety and circular economy aspects being those that most need improvement. So, this model is using in a European project financed by European Union and developed in Spain by the CSIC, is the Spanish Research Council, and they are using our model to evaluate farms in sustainability, and you have all the information, additional information, in our website nanta.es, sorry. And I really say thank you to the organization of this event, and congrats them in order for the success of it. And we are really proud to be here sharing this time of you. Thank you very much.
Please don't leave. Probably somebody has questions. No questions for Nanta. So now we pass to our next sponsor, ANIPLA, and I call Dr. João Cardoso to make the presentation on the next platform. It's a smart farm platform that he will be the best person to explain.
Thank you very much. Bom dia a todos. Eu também vou falar em português para correr isto mais rápido. Portanto, o meu nome é João Cardoso. Primeiro de tudo, queria agradecer ao CATAA, portanto, toda esta organização deste magnífico evento. Rapidamente vou-vos passar a apresentar um projeto que para a ANIPLA, a Associação Nacional da Indústria para a Proteção das Plantas, tem no terreno. Portanto, já faz hoje precisamente um ano que lançámos esta plataforma, a Smart Farm Virtual. Portanto, está de parabéns, é o primeiro aniversário. E esta apresentação, desculpa, isto
dá para apontar.
Ok, pronto. Portanto, este projeto, o Smart Farm Virtual, está a ser apresentado um pouco por todo o país desde o verão, portanto, no âmbito do Smart Farm Hub, que foi um protocolo que a ANIPLA fez com estes 4 parceiros, o Smart Farm Collab, o CIP, o Inov Plant Protect e a Aposol, no âmbito destas 4 áreas que são fundamentais para uma agricultura moderna e sustentável, nomeadamente a agricultura digital e de precisão, a biotecnologia, os biopesticidas e também a agricultura de conservação. E no âmbito deste, e o grande mote deste projeto é divulgar uma agricultura inovadora, tecnológica e promotora da biodiversidade. Muitos destes objetivos nós vimos refletidos aqui hoje de manhã, por exemplo, a questão da biodiversidade do solo, da conservação do solo, a questão das novas pragas e doenças, como vimos o caso da colega que apresentou a moniliose, por exemplo, nos pêssegos. Portanto, todos estes aspectos são relevantes e têm desafios muito grandes para a nossa agricultura. E então aqui o nosso contributo, portanto, para a capacitação do setor em todas estas novas tecnologias e ferramentas está vertido na Smart Farm Virtual. Convido-vos a fazerem a visita através aqui do QR Code que está projetado e também através do nosso website. Portanto, podem fazer aqui a visita virtual à nossa plataforma, a Smart Farm Virtual. Vou então começar aqui a visita. Portanto, pedia só que ajudassem a projetar. Pronto, já está. Desculpa. Isso. Portanto, vou-me só virar aqui de lá, como não tenho aqui projetado no computador. Sim. Ótimo. Desculpem lá. Isto. Logística.
Para cá.
Já está. Portanto, aqui a nossa Smart Farm Virtual está localizada na Companhia das Lusírias, portanto, em pleno coração do Ribatejo, mesmo junto ao do Rio Tejo, que podem ver lá à frente. Ok, pronto. Portanto, isto é uma plataforma totalmente interativa, podem ver, dá para navegar a 360 graus, como se estivéssemos no Google Maps. Portanto, isto como estava a dizer, é um projeto que está implementado na Companhia das Ilhas, muito perto do Rio Tejo, uma zona de proteção especial devido às aves e também à questão do estuário do Tejo. E é um local onde se faz uma agricultura moderna e com aplicação de bastantes tecnologias e de muitas preocupações ambientais. E aqui a nossa Smart Farm pretende capacitar o setor para o uso seguro e sustentável dos produtos fitofarmacêuticos, por um lado, portanto essa é a base deste projeto, mas por outro também integrar aqui todas as técnicas de conservação do solo, de proteção e produção integradas, das novas tecnologias, nomeadamente da agricultura digital e da agricultura também de conservação. Portanto, aqui a nossa Smart Farm está dividida em várias estações para facilitar a sua navegação, começando logo aqui pela questão do armazenamento, da proteção do operador, da gestão das embalagens vazias, depois passando aqui para a questão da gestão dos efluentes e da preparação da calda, depois todas as boas práticas na utilização de produtos de proteção das plantas, e passando aqui depois para a questão da proteção e conservação da biodiversidade e da agricultura digital e de precisão. Então convido-vos a virem comigo nesta visita. Podemos começar aqui pela introdução. Todos estes elementos que estão contornados a verde são elementos interativos, têm desde vídeos, textos, também painéis interativos e os nossos folhetos. Portanto, podemos simplesmente clicar e fazer o zoom. Aqui já tem bilingue, portanto os nossos colegas que falam inglês podem acompanhar a visita através dos painéis cuja tradução está realizada. Portanto, só tirar. Este sou eu, como podem ver, e se carregarem em mim vai aparecer um vídeo de introdução ao projeto que vos convido depois a ver também. Portanto, vamos entrando aqui para a primeira estação, que é o armazenamento. É muito importante termos presente que estas regras que estão vertidas na Smart Farm são transversais a toda a agricultura, portanto, desde que se utilizem produtos para a proteção das plantas, quer em modo de produção biológica, quer em modo de proteção integrado, quer de outro modo qualquer, portanto, temos que cumprir estas regras. E são referências os que estão aqui, portanto é adaptável a todas as realidades e todas as dimensões agrícolas. Começando aqui pelo armazenamento dos produtos, não vou entrar em detalhe em todos os pontos, não estamos aqui 2 horas e todos nós queremos ir almoçar, mas temos aqui, por exemplo, um esquema de como fazer um armazenamento correto dos produtos fitofarmacêuticos. Portanto, tem aqui todos os guidelines técnicos para fazer este armazenamento corretamente e em segurança. Só dar aqui outro exemplo, por exemplo, aqui quando vem aquele ícone, aparece aqui uma caixa de texto com todos os detalhes técnicos que é necessário saber. Portanto, aqui vem, por exemplo, uma fotografia 3D de um outro exemplo de armazenamento. E aqui nos PDFs abre o link para os folhetos técnicos que a Anipla editou, que é possível imprimi-los, descarregá-los. Portanto, isto em contexto de formação dá uma flexibilidade muito grande, só para dar este exemplo de utilidade. Seguindo, temos aqui também o exemplo de um vídeo de como vestir e despir os EPIs, como armazená-los, como fazer a sua manutenção. Também tem aqui um exemplo deste painel interativo que eu pessoalmente gosto bastante, porque tem, no fundo, resumido todas as características e requisitos que um bom EPI deve ter, para não estarmos a comprar gato por lebre, por exemplo. Aqui a questão da gestão das embalagens vazias de produtos fitofarmacêuticos é uma questão de sustentabilidade cada vez mais importante. Para terem uma ideia, nós no setor agrícola temos uma taxa de recolha e reciclagem muito maior do que temos nos setores urbanos, portanto é aqui um sinal de responsabilidade dos nossos agricultores para com o meio ambiente. Passando cá para fora, temos aqui a zona de preparação da calda. Estas infraestruturas que vamos ver agora são muito importantes para mantermos tudo o que são os produtos de proteção das plantas em circuito fechado, para não haver fugas para o ambiente quando eles estão na sua forma mais concentrada. Portanto, aqui trata-se de uma bacia de retenção onde se faz o enchimento e lavagem de todas as máquinas de pulverização e também dos tratores. Isto é muito importante porque aqui recolhem-se todos os efluentes e não se libertam para o ambiente, porque depois vão ser tratados na fase seguinte. Chama a atenção aqui também para os vários vídeos e informação que temos, nomeadamente aqui para os cálculos de doses. A cálculo da dose é uma característica muito importante, porque permite ao agricultor ter aqui uma ferramenta de rápido acesso para tirar eventuais dúvidas que possa ter neste processo. Existe aqui também o sistema de transferência fechado, que permite fazer uma preparação da calda sem termos que abrir a embalagem. Portanto, deixo aqui esta curiosidade no ar e peço-vos que vão ver este vídeo de demonstração para perceberem como é que isto é possível. Como vos falei, aqui do lado direito recolhemos os efluentes e aqui à esquerda temos dois sistemas em que vamos fazer o tratamento destes mesmos efluentes sem os libertar para o ambiente. Portanto, isto é um ponto de sustentabilidade muito grande em termos de prevenção da poluição da água e do solo. Aqui temos o sistema heliosec. Depois tem aqui o vídeo que demonstra como é que ele funciona, e aqui o sistema Fitobac, mais uma vez também tem aqui o vídeo de funcionamento. Mas no fundo o heliosec funciona pela evaporação da água, por ação do sol e também do ar, e o Fitobac funciona por degradação biótica dos produtos químicos. Portanto, e no final temos no fundo os nossos efluentes tratados sem haver libertação para o ambiente. Avançando aqui para a questão da pulverização, aqui é muito importante em termos de formação para os agricultores, porque ensina como fazer o controle da deriva da pulverização, quais é que são os bicos adequados a cada tratamento que vamos fazer, no fundo, quais é que são as pressões de trabalho adequadas e também como é que mantemos o produto exatamente no seu alvo, que no fundo é isto que estamos aqui a falar, portanto, de otimização e de eficiências e de não haver perdas para fora do alvo, isto é, para o ambiente. E quando falo aqui o alvo, estou a falar da planta que queremos tratar ou da infestante que queremos controlar. Passando aqui para uma estação que engloba muitos dos aspectos que foram falados hoje de manhã, portanto, aqui a estação da biodiversidade. Podemos ver aqui o nosso folheto de biodiversidade, que dá muitas pistas de como é que os agricultores podem integrar, portanto, os elementos de ecologia no seu dia-a-dia, quer em qualquer tipo de exploração agrícola, quer convencional, de produção integrada, produção biológica. Portanto, temos aqui informação muito geral e conceptual que os agricultores podem adotar. Ao nível aqui de alguns elementos de biodiversidade que temos expostos, vou só percorrer aqui alguns. Temos, por exemplo, aqui os abrigos, neste caso os ninhos para as aves. E porque é que temos aqui os ninhos das aves? Pode parecer uma coisa banal, mas não é tão banal assim. Só para dar aqui três grandes exemplos, por exemplo, uma família de corujas pode comer até 15 mil ratos por ano e cobrir até 5 hectares de plantação. Por exemplo, aqui o nosso parceiro, a Companhia das Lesírias, implementou um projeto de recuperação da coruja das Torres, que era uma espécie ameaçada no estuário do Tejo, e conseguiu multiplicar grandemente esta população através só da colocação de ninhos em sítios estratégicos. Um insetívoro como o chapi azul pode comer mais de 200 insetos por dia, e os próprios morcegos, como sabem, comem até 2/3 do seu peso por noite em insetos. Portanto, temos aqui um controle ecológico e uma prevenção muito grande na questão das pragas. Passando aqui à imagem do nosso drone, para a charca que dá apoio ao Centro de Lavoura, temos os abrigos flutuantes, neste caso são feitos de palha de coco, mas podiam ser feitos de cortiça, por exemplo, em que estão povoados com vegetação. Aqui não está muito exuberante porque tinham sido acabados de plantar, e isto tem uma tripla função: melhorar a qualidade da água através da absorção de nitratos pelas plantas, servir de plataforma para os anfíbios, e como sabem, os anfíbios são ótimos controladores das populações de insetos, especialmente na água, que é um local onde os insetos se multiplicam bastante, e também podem servir de base para nidificação de aves aquáticas. Eu tenho fotografias disto a acontecer. Passando aqui para... Portanto, só dar aqui dois aspectos importantes. Aqui convido-vos também a ver este vídeo relativamente à conservação do solo. Portanto, temos aqui dois aspectos muito importantes: o controle do escorrimento superficial para a prevenção da erosão e também para a prevenção da poluição do meio aquático, quer por parte de fertilizantes, quer de produtos fitofarmacêuticos, e por outro temos aqui toda uma panóplia de soluções para a conservação do solo, como já foi falado aqui hoje de manhã, e eu não vou entrar em grandes detalhes, mas que é uma preocupação muito grande que nós temos e damos muito ênfase nas visitas que fazemos à Smart Farm física, neste caso. Temos aqui o nosso abrigo de insetos, portanto, serve para sensibilizar os agricultores para o uso correto de inseticidas, por um lado, e para dar pistas como é que podemos utilizar os insetos em nosso benefício, nomeadamente nas duas grandes funções, que são no controle de pragas e doenças por predação ou competição, e, por outro lado, portanto, o auxílio à polinização. Já estou mesmo a terminar, portanto, aqui temos os benefícios das bandas de arbustos aromáticos. Como podemos ver aqui, a falta de habitat para os polinizadores é o grande problema que essas espécies enfrentam, e aqui damos pistas como é que podemos integrar estes elementos numa exploração agrícola para resolver esse mesmo problema. Temos ainda, para terminar, os abrigos terrestres que promovem a presença de fauna réptil de maior porte, nomeadamente cobras e também outros lagartos, para o controle de pragas e para também a melhoria do equilíbrio ecológico das explorações. Termino aqui com a estação para a agricultura digital e precisão, em que tenho aqui um exemplo concreto de sensorização do nosso parceiro Smart Farm Collab. Este é o sistema SOFIS aplicado à cultura da oliveira, em que temos aqui os sensores atmosféricos, no fundo uma estação meteorológica aplicada e trabalhada para certos efeitos, os sensores fixados na planta e os sensores fixados no solo, que nos dão uma multiplicidade de informação que podemos usar em nosso proveito, desde logo na monitorização e prevenção de pragas, neste caso de doenças. Estamos a falar mais de doenças fúngicas, também da utilização correta e otimizada do recurso água, que é extremamente importante para o nosso contexto. Só para dar dois exemplos de como a tecnologia pode ajudar a agricultura a ser verdadeiramente sustentável, que já o é, portanto, mas que pretende, neste caso, ir ao encontro dos objetivos do Farm to Fork, o mote deste Congresso. Convido-vos mais uma vez a verem a plataforma com tempo e com interesse, e muito obrigado.
Muito obrigado. Vamos aguardar a ver se alguém tem questões. Any questions? But I have some questions. Eu tenho uma questão. É possível, para além de visitar online, visitar a quinta propriamente?
Sim, basta contactarem-nos. Portanto, ter os contactos da Anipla, o meu próprio contacto. Portanto,
os contactos estão
estão na apresentação, logo no início. Portanto, é só contactarem-nos e dizer, ok, queremos combinar uma visita à Smart Farm, e então combinamos e podemos fazê-lo.
E colaborações em projetos também estão sempre abertos, claro e a colaborar, porque já têm muitas colaborações, por isso.
Sim, é verdade. E estamos numa toada de colaborar. Por exemplo, desculpem, falámos aqui do projeto Benchmarks, que tivemos o prazer de participar no primeiro workshop. É apenas um exemplo que vimos aqui hoje de manhã, no qual também estamos a iniciar a colaboração. No Centro para a Apicultura e Biodiversidade, que também somos parceiros, que o CATAA também é parceiro, portanto também estamos presentes, por isso estamos abertos.
Ok, pronto.
Muito obrigado.
Muito obrigado. Se alguém tiver questões, any questions? Everybody's hungry. Vais lá? Obrigado então.
Muito obrigado.
Obrigado. Yesterday we had to cut the part of team 1 for the videos. We will show 2 videos before you go to lunch, so you can get a little bit more hungry, so we can eat more, in order that we don't have any food waste, like we talked yesterday. It's just 2 minutes, if we are able to put the video.
Olha ali o rato. Espera aí. Tem que ficar ali.
Posso? Espera aí, posso?
Sim.
Vais lá?
Porquê que tiraram o rato?
Eu não te consigo ouvir.
Just for you to actually understand what you will see, we actually, in the Facility Project, the Facility Project has 34 partners across Europe, involves 13 countries, and one of the objectives, I mean the main objective was to create living labs across these 12 cities. We have cities like Rome, Oslo, Athens, Differdange, Kharkiv in Ukraine. So lots of cities across Europe where living labs were made to promote the sustainable food systems. So lots of changes were made in these cities. In Portugal, we are the only city that belongs to the Facility Project, so Castelo Branco. We also had a video before showing here. And now we will just see like short videos along the day. So now we will just see 2 cities. And they are like short videos, so you can see some examples of what they have been doing so far. Thank you.
So we have the city of Rome.
So authentic, you can copy me. Ooh, yeah. Just keep on watching me. You see me walking to my own beat, like I'm on it. All the Main Street, shake, 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 shake. Oh, watch me shake my thing. There's nobody in this world like me. Make it look easy, like 1, 2, 3. Yeah, you know I'm about to steal the show. You never met no one like me before. Ooh, yeah, better get on me. I know you like what you see. I know you like what you see. One of a kind, just watch me shine. You wanna be like me? We don't waste your time. Can't copy me. There's nobody in this world like me. Make it look easy like 1, 2, 3. Yeah, you know I'm about to steal the show. You never met no one like me before. Ooh, yeah. Just watch me do my thing. There's nobody in this world like me. Make it look easy like 1, 2, 3. Yeah, you know I'm about to steal the show. You never met no one like me before. Ooh, yeah. Better get a hold of me. Ooh, yeah. Just keep on watching me. You see me.
So we continue in Italy, and now Torino. Okay, I think we have seen so much food that we are really hungry now. So I invite you to be back here at 2 o'clock sharp, and we will start at 2 o'clock, okay? I want to remember you that we will have 2 workshops at 2:30, one special for farmers. It's in the next building. At 2:30 also a workshop upstairs for food trends of 2024, and then we have another workshop after it from our directorate, regional directorate of agriculture and fisheries.
If you want to know some of Portuguese products, namely from this region, we will have at 3 o'clock codfish, typical codfish Portuguese style, dried and salted, and also we have fish from the Mondego River. And also we have some products from agriculture, namely honey and peras São Silvestre, if you want to know, and other products. So feel free to enter this room and go upstairs and join us, okay? Have a nice lunch.
Faltam só 3.
So we thank you.
Thank you all for coming. Since we have the translation services, we are going to present in Portuguese this session. So this is, esta sessão então é relacionada com os serviços dos ecossistemas como ferramentas para a resiliência e sustentabilidade territorial e chamamos para se juntarem a nós os oradores Anabela Paula, Fernando Mata, Teresa Carita, Luís Duarte e Eduardo Nascimento. Vamos deixar subir. Começar pelo nosso keynote speaker, é o professor Fernando Polido, catedrático da Universidade de Extremadura, e que nos vai fazer a apresentação. Tenho que pôr os óculos, não vejo.
A apresentação vai ser Mosaic Approach for the Implementation on Fire Smart Landscapes.
Okay, should I start?
Yes, please.
Okay. Can you see my screen?
Aguarda un poquito, que estamos poniéndolo.
Pois, mas não está escuro. Ups. Onde é que está a Silvia?
Então.
Sabes onde é que é? Não é cheiro.
Ajuda?
Não?
Ah, é o Eduardo.
Nossa Senhora.
Será que podem dar só aqui uma ajudinha a pôr novamente o Hugo? Obrigada.
Obrigada.
Tem que se pôr em duplicado.
Não sei, eu de manhã também já estava com uns problemas.
Ah, ok.
Não está a resultar. Acho que...
Já está.
Espera aí. Agora metes isto em ecrã inteiro, bem. Não, ainda falta. Ecrã no ecrã?
Não. Aqui, deixa estar assim. Tá?
É, já é melhor.
Se carregarmos... Não, se calhar é ele.
Não, ele é máximo. Força, Fernando.
Can you see the screen?
Sí, ponelo en modo de presentación.
Okay. Good afternoon, and thanks the organizers for the invitation, especially to Professora Paula Castro, and I have the pleasure to collaborate with her team in Coimbra, and I hope we will get interesting results in the future, linked also to the Proyecto Mosaico. This is a project we started 7 years ago in the University of Extremadura, in the northwestern part of Extremadura region, very close to the border with Portugal. And I will try to summarize the approach and the main results of the project, and also some of the connections we have also with with the work of food, the food linked to the rural landscapes we have in Extremadura region. Of course, this project has been developed by the University of Extremadura in collaboration with different stakeholders, especially the government of the region of Extremadura, but also with the help of many different European and national projects, which are the main source of funding for us.
Fernando.
As you all probably know already, and as I try to show in this graph, you can hear the evolution in the last decades.
We are not seeing your presentation. Can you please start your presentation? Sorry.
Put in presentation mode.
I hear a lot of noise.
Your presentation, it's not running. You have to...
Can I continue?
You have to press on the button to start your presentation.
The slides are not...
I try to show you the evolution in the last decades of the proportion of the burn areas
We cannot see it.
in wildfires across Europe. As you can see, basically in the Mediterranean region of Europe, we have around 80% of the burn areas in the last decades.
Fernando, excuse me. The presentation is... we are not seeing... We are not seeing the slides.
A lot of noise.
We cannot see your presentation. It's in the first slide.
No, it's not...
it's not in the running mode. Please press the button for presentation.
But before you saw it.
No, we just see the preview.
No. Okay, I will try to share it again. Okay?
Thank you.
Can you see it now?
We see the slide, perhaps the current one, but it's not in the presentation mode.
You don't see it?
We see the slide.
I'm sorry that I cannot hear you.
We can see the second slide, but it's not in the presentation mode. Can you change it to the presentation mode, Fernando?
Okay. It's okay now?
No.
No?
No, it's not working. But you can continue. Just click on the slides in the left side, so you can see it.
Well, my problem is that I cannot hear you very well. So, do you see the picture now?
Yes.
Fernando, ¿me estás oyendo?
This light blue line shows that the probability of extreme large fires, what we call mega fires, will increase in the future, and the speed of this increase is much higher than in the other regions in Europe. So the trend we have suffered in the last decades will increase in the next decades. No? Especially in Portugal and Spain, we know a lot about this problem. Well, normally everybody relates this mega fire problem with only with climate, but we have a lot of information showing that the problem is due to the combination of climate and the accumulation and the expansion of forest with no management in our rural landscapes. Here I try to show 4 main drivers which are not related to climate but to land uses that can...
Fernando,
¿Sí?
Perdona.
Yes.
Pero tienes que poner en modo de presentación, si no los slides no van cambiando.
Pero antes lo he puesto en modo presentación y no lo veíais.
Aún no lo vimos nunca, en modo de presentación.
¿Puedes verlo ahora?
No, sigue lo mismo. Tienes puesto el slide 2 y no cambia.
Bueno, voy a intentar otra vez, pero no sé realmente cuál es la solución.
Intenta presionar F5. A ver si... Después de ponerla, después de compartirla.
¿No lo veis?
Ahora intenta cargar en F5.
Sí, lo estoy haciendo.
Y en tu PowerPoint abajo, ponerlo en modo de presentación, ¿no?
Ahora está en modo presentación.
Seguimos lo mismo.
¿No lo veis?
Bueno, no, nos vamos siguiendo así, pero tienes que cambiar los slides en el lado izquierdo, ¿vale? No, quizá quitarlo de modo de presentación. Intenta cambiar de slides.
Os oigo muy mal.
Quita el modo de presentación e intenta cambiar los slides en el lado izquierdo, donde ponen los slides pequeños que van a seguir.
Ahora ves. ¿Puedes verlo?
No, ahora nada.
Casi siempre es un problema relacionado con Webex. Puedo continuar si quieres solo con la voz.
No final vão ver que se calhar até pode fazer sentido porque vamos ligar à apresentação do mosaico. E portanto eu vou falar também em português porque como está a ser transmitido para fora e eu acho que é muito importante nós conseguirmos chegar ao território, e aqui temos a tradução, vou fazê-la em português. Portanto, vou falar da cocriação de estratégias de desenvolvimento sustentável para a área de estudo do cultivar, tendo por base o framework dos serviços de ecossistemas. Portanto, nós, como sabemos, as paisagens rurais enfrentam vários desafios, alguns ligados à agricultura, quer pelo seu abandono, quer pela intensificação, e estes problemas requerem uma co-criação, uma criação conjunta com a comunidade de soluções sustentáveis. Sabemos também, pela literatura, que poderemos tentar suportar e dar o apoio nesta resolução, nesta procura de soluções, tendo por base o framework dos serviços de ecossistemas, e foi isso que fizemos para a área de estudo do cultivar, que abrange 7 municípios aqui da zona da Beira Interior. É uma zona predominantemente rural, como podemos ver neste mapa, se calhar com exceção ali mais da zona da Cova da Beira, e que tem uma grande diversidade de paisagem, desde a agricultura mais tradicional dos mosaicos, agro-silvopastoris, até zonas com maior intensidade agrícola, ou florestas de produção, agroflorestas, zonas de carvalhais. Portanto, temos num território relativamente curto uma grande diversidade de paisagens. Para falar-vos deste processo de cocriação, vou-vos falar do roadmap que nós definimos. Obviamente começamos com o envolvimento de stakeholders, não apenas a parte agrícola, mas também florestal, a parte do turismo, do desporto, da cultura, o poder local, as organizações governamentais, NGOs, eu não me lembro do nome, entre outros. E o primeiro grande passo foi identificarmos quais os serviços de ecossistemas-chave que o território considerava importantes. Isto porque quem conhece e trabalha com estas ferramentas, temos uma grande multiplicidade de serviços, não conseguimos avaliá-los todos e é importante perceber quais avaliar. Tivemos diferentes abordagens, desde os workshops a questionários, como vai falar a Paula, e chegámos a um conjunto de 12 serviços de ecossistemas para avaliar. Fizemos também uma análise SWOT, começámos por uma avaliação de percepção individual através de um questionário online e depois uma definição de estratégias também num segundo momento de um workshop participativo. Portanto, como falava, tivemos uma abordagem múltipla em termos de seleção dos serviços de ecossistemas, que nos permitiu selecionar estes 12, portanto, a nível de aprovisionamento, produção agrícola e pecuária, os cogumelos comestíveis, biomassa florestal, depois em termos de regulação e suporte, a qualidade do solo, o estoque de carbono, a regulação do risco de incêndio e a polinização, e a nível dos serviços culturais, a parte recreativa, paisagens culturais, elementos simbólicos e de legado, conservação ou legado. Mapeámos estes serviços, obviamente procurámos identificar indicadores que fossem representativos dos mesmos, que nem sempre é fácil, e depois de mapearmos os serviços, voltámos a discuti-los mostrámos e discutimos com os stakeholders no sentido de perceber se representavam também a sua percepção ou se poderíamos melhorar algum dos indicadores. Com base no feedback, alterámos e revimos alguns dos indicadores dos mapas, que para depois analisarmos as sinergias e as zonas onde eles não coincidem, para depois podermos fazer uma análise de trade-offs. Relativamente à SWOT, como vos disse, começámos por um questionário online a diversos stakeholders, com perguntas abertas para nos elencarem as 4 oportunidades, as ameaças de cada uma, e depois trabalhámos isto do ponto de vista participativo. Portanto, na análise SWOT, a informação que recolhemos foi enorme, muito dispar, tentámos sistematizá-la, organizá-la em grandes ideias, mas, como sempre, às vezes é difícil termos a certeza que estamos a interpretar bem. Portanto, o que fizemos primeiro foi uma validação desta SWOT, recolhemos as suas percepções, estavam de acordo, se havia algo a acrescentar, e depois pedimos para prioritizar e identificar quais as principais ameaças, as fraquezas, as oportunidades e as forças. Com base nisso, trabalhámos o que chama uma análise SWOT, que não é nada mais que uma SWOT invertida, em que discutimos com eles como é que podemos utilizar as forças para explorar as oportunidades ou para evitar e minimizar as ameaças, e como é que podemos ultrapassar as nossas fraquezas explorando as oportunidades e minimizando estas ameaças. Esta discussão conjunta permitiu-nos começar a trabalhar numa definição de estratégias. Falar-vos agora muito brevemente deste mapeamento dos serviços, sem entrar em grande detalhe, o tempo não é muito, podemos falar depois. No fundo, trabalhámos a questão do aprovisionamento, aqui a parte agrícola, quer com base nos dados do INE, do relatório dos censos agrícolas, associado à COS, quer a abordagem na agrícola e pecuária foi semelhante. No caso dos cogumelos, trabalhámos em termos de publicações que nos dê informação da produtividade e associada às diferentes classes da COS, para podermos depois mapear a questão do biomassa florestal. Utilizámos os dados do inventário florestal e a COS. Este foi um daqueles que nós revimos após o workshop. Em vez de usarmos a COS 18, usámos a COS 22, que tem maior detalhe. Aqui no caso dos dados de qualidade do solo, utilizámos os dados do Lucas, e foi muito com base na relação carbono-azoto. Aqui o estoque de carbono também, os dados do inventário florestal e a COS 22, a regulação de incêndios e a polinização. Peço desculpa que isto não estava. Portanto, são dados que vêm de outros colegas de cultivar, neste caso a regulação de incêndios aqui do colega Albano Figueiredo e da sua equipa, e da polinização. Vocês já ouviram hoje de manhã falar sobre o trabalho feito. Do ponto de vista do recreativo, utilizámos dados de redes sociais, o FLIRC, dados de fotografia de natureza, o GBIF e também todos os percursos, todas as áreas mapeadas. Em termos de paisagens culturais, usámos aqui um bocado o índice da diversidade de paisagem. Do ponto de vista simbólico, todos o que são os... como é que chama, a heráldica de cada povoação. Cada povoação então tem o seu símbolo, e usámos essa informação para perceber, a informação para percebermos qual a relação desta agro-silvopastoril nestes símbolos, e depois a área de legado de conservação com todas as camadas de qualquer tipo de proteção. Portanto, esta informação depois permitiu-nos identificar os hotspots e os cold spots. Aqui mantém a mesma lógica de cores: amarelo, provisionamento; verde, relação e suporte; e cultural, azul. E depois temos os hotspots a azul, os cold spots, e a vermelho, os hotspots dos serviços. Isso permite-nos visualizar de uma forma mais óbvia as zonas onde temos realmente esta provisão de serviços e onde temos uma menor provisão de serviços. Informação muito útil para então discutirmos posteriormente os trade-offs. Portanto, fizemos esta análise para todos estes 12 serviços. E depois a análise das sinergias e dos mismatches. Toda a informação foi convertida numa grelha de 1 por 1 km para que pudéssemos depois compará-la. Este foi aqui um trabalho, um esforço também e toda esta parte deste projeto foi uma grande colaboração entre a nossa equipa com a equipa do IPCB, e aqui um grande obrigado à Natália, que com todas as suas múltiplos conhecimentos de SIG nos ajudou a criar esta informação muito densa, que procurei aqui torná-la mais clara, mas no fundo é uma análise de correlação positivas, negativas entre os vários serviços. Aqui as letras no fundo estão na coluna da esquerda, tanto A, B, C, D são aqueles 12 serviços, e por exemplo, como é óbvio, temos aqui sinergias entre o estoque de carbono e a biomassa florestal, entre o estoque de carbono e a questão dos cogumelos, ou por exemplo aqui a relação entre a questão agrícola e a relação de redução do risco de incêndio no guia. Portanto, temos aqui o tamanho das cores, no fundo está relacionado de uma maior correlação entre os serviços e uma menor correlação. Da mesma forma, analisámos estes mismatches, que também são como é que eles, onde é que normalmente quando temos um não temos o outro, e esta informação é importante porque o trade-off no fundo é uma discussão conjunta de onde é que estamos dispostos a negociar com os vários stakeholders, do que é que vamos priorizar e como é que vamos conseguir trabalhar isto. Dizer-vos ainda que, em termos de grandes estratégias, obviamente que saíram muito mais de estratégias, mas resumindo, temos aqui 3 grandes grupos, portanto, promover o turismo da agricultura de natureza como forma de produzir e dar valor aos recursos endógenos, promover também, desta forma, cadeias curtas de mercado, sendo muito importante termos uma boa estratégia de marketing que seja coordenada entre ambos os setores, o setor turismo e produção. A valorização dos mosaicos agro-silvopastoris, que, como vimos, são, durante a análise, hotspots de biodiversidade, como forma também de promover as dinâmicas rurais, sendo muito importante cada vez mais trabalharmos na consciencialização da sociedade do importante papel que estes setores agrícolas, florestais, pecuários, somente estes extensivos e estes exemplos que temos no território têm na manutenção destas paisagens culturais e todos os serviços de ecossistema relacionados, nomeadamente a redução de risco, e depois a importância de termos um foco na inovação, quer seja na indústria do processamento dos produtos, quer seja nas técnicas de produção. Em termos de próximos passos, dizer-vos apenas que este projeto Cultivar permitiu-me também perceber o que é que eu gostaria de fazer a seguir. Comecei o doutoramento recentemente ligado à questão também deste novo programa de transformação de paisagem. A área de estudo parcialmente coincide com o Cultivar, aqui Fundão, Sabugal e Penamacor, onde foram definidas estas áreas integradas de gestão de paisagem, das primeiras que foram definidas, conjuntamente com o projeto Mosaico, que o Fernando vai apresentar em seguida, em que tem o grande foco é avaliar estes trade-offs de serviços de ecossistemas nestes territórios que sejam fire smart, que sejam resilientes aos incêndios, em que o grande objetivo é dar continuidade a toda esta estratégia colaborativa que iniciamos para co-criar soluções de base natural que promovam estas paisagens smart e que simultaneamente não só reduzam o risco de incêndio, mas também promovam outros serviços. Para concluir, e aqui como message, encontrar soluções para problemas complexos é um caminho longo, difícil, que deve ser baseado na partilha de conhecimentos e execução conjunta, pois só assim conseguimos soluções realistas e práticas.
Muito obrigado, Anabela. Eu chamaria agora então para fazer a sua apresentação o Fernando Mata, que vem do Politécnico de Viana do Castelo, e a comunicação será sublinhada ao tema Demographic Tendencies of EUA Citizens' Perception about the Greening Subsidies Given to Farmers.
A apresentação.
Qual é que é? Não é essa, pois não? Não. Qual é que é a sua apresentação? A que diz Fernando?
Hello, good afternoon, everybody. I'm going to do the presentation in English, and I'll start by introducing myself. My name is Fernando da Mata. I'm coming from northern Portugal, from the Centre for Research and Development in Agri-Food Systems and Sustainability, which is a research center belonging to the Polytechnic Institute of Viana do Castelo. So, introducing the topic, setting the scene. As we all know, the Common Agricultural Policy is a key policy in the European Union. It consumes about 1/3 of the total EU budget, so 57 billion euros, which is a figure with 9-11 figures, so a huge number. So this Common Agricultural Policy was set from the beginning, the creation of European Community, and has suffered several reforms throughout time. And the last one was made, and the focusing now is on trying to tackle climate change and on the sustainable management of natural resources. Obviously, being 1/3 of the European Union budget, it is very important for the policymakers to make sure that there is a positive perception by all European citizens regarding the implementation of these policies. So the Common Agricultural Policy is very important, as we all know, for the Green Deal strategies, for the farm-to-fork strategies, and for the biodiversity strategy. So the aim of this presentation is this is part of a major project, which is a project is trying to understand how the European citizens perceive the Common Agricultural Policy, and particularly this study is concentrating its attention on the greening subsidies being given to the farmers. So the greening subsidies are basically direct payments being made to the farmers to support them in maintaining farming practices that can contribute to the environment and climate goals in the European Union. So in a certain way, we are rewarding the farmers for preserving the natural resources, our environment, for providing a benefit that is beneficial for all European citizens. So in the end, the farmers are providing a service to the European society. So we use data from the Agro-Alimentar. So Agro-Alimentar collects data and produces reports that include only descriptive statistics, and then they make the data available for researchers to further explore. And particularly in this study we used this question. The EU currently making subsidy payments to farmers for carrying out agricultural practices beneficial to the climate and to the environment. Are you in favor or opposed to the EU continuing to do so? So the question was answered kind of in a Likert scale, total in favor, somewhat opposed, and totally opposed. And several independent variables were also present in the survey, and we used these in a statistical model to produce some results that could explain this question from the point of view of demography. So these are the results of this particular model. So individuals totally in favor and supporting the greening practices tend to be older, more educated, slightly to the with a lefty tendency, from higher classes of the society, from smaller communities, so from rural areas, and they also tend to be more optimistic about the future of the European Union. Also, these individuals tend to have stronger positions in the relationship between agriculture and the climate change. So in conclusion, the Common Agricultural Policy is a cornerstone of the European Union policy making, increasingly orientated towards climate change and promoting sustainable agricultural practices. So these insights can be used by policymakers in trying to align the European Union policies with the needs and the expectations, obviously, of the European citizens. So your attention for these results, as these are only general tendencies, and also it's important here to highlight that the vast majority of the European citizens, in fact, support the Common Agricultural Policy, and particularly the greening practices. I would like here to introduce a small discussion and to remember a case study was being reported and studied by scholars, particularly in the United States, which is the political division in the USA between Democrats and Republicans regarding the climate policies. So, as we all know, Donald Trump withdrew the United States from the Paris Agreement that was implemented in 2015. This happened in 2017. And meanwhile, the current president of the United States re-entered again the United States into the Paris Agreement. This divide is very important to be highlighted because big corporations and also some corporations with interests, for example, in oil production, try somehow to intoxicate people and they tend to be in denial regarding climate change. Fortunately, it's not the case in the European Union, and it has been observed that only a small community of conservative males tend to be rather in favor or in denial of climate change. So most of the Europeans tend to agree with the common agricultural policies. Just some links just to show you that what I'm saying is not an invention. For example, we have here a news from the Washington Post, where you can see, and if you explore further, you will see that you can explore this news. There's a lot of places where you can see this news. Also here from BBC you can see studies where journalists have investigated and have linked the interests of the oil industry with the creation of doubts about climate change. So my aim here is also somehow to make you all aware of these intoxications. Thank you very much for your attention.
Muito obrigado, Fernando. Agora chamaria a minha parceira, a Paula Castro, que vem do Centro de Ecologia Funcional da Universidade de Coimbra, e vai-nos apresentar uma comunicação subordinada ao tema Farmers' Perception of the Ecosystem Service Provided by Agriculture.
Muito obrigada. Boa tarde. nossas desculpas por estes momentos. O Fernando Polido falará a seguir a mim. O que eu venho aqui apresentar muito brevemente relaciona-se com um dos objetivos muito fortes do Programa Cultivar e dos projetos que levamos a cabo nesta rede, neste grande projeto, que é envolver stakeholders, envolver atores, envolver as entidades relevantes para estas questões da sustentabilidade agrícola e do setor agroalimentar. Um dos principais atores são, de facto, as produtoras, os agricultores, e foi nesse sentido que os convidamos a participarem connosco e partilharem connosco também as suas percepções, as suas expectativas, as suas opiniões sobre as atividades e aquilo que oferecem à sociedade, que tipo de benefícios, que tipo de serviços e ecossistemas percepcionam que nos oferecem. E, neste sentido, foi elaborado um questionário que foi feito presencialmente, portanto face to face, que a colega Marie já falou também um bocadinho de manhã, e foi um questionário que demorou hora e meia. Portanto, estas pessoas disponibilizaram-se, deram do seu tempo para estar connosco, falar um bocadinho connosco e nos darem os seus feedback sobre estas questões dos serviços de ecossistemas, e a eles o nosso muito obrigado. A área é a área do projeto Cultivar, portanto estes sete concelhos. A nossa amostra foi de 77 entrevistados, cerca de 25% foram mulheres. Cada bolinha destas representa 10%, para terem uma ideia, portanto isto é só uma comparação muito breve do tipo de amostra. Fizemos aqui ou acrescentámos aqui a dimensão do género, se haveria aqui diferenças entre homens e mulheres em como percepcionam, veem a sua atividade, veem as suas explorações como positivas ou negativas, por exemplo. E começámos por perguntar se conheciam o conceito do ecossistema. E houve aqui uma ligeira diferença entre homens e mulheres, mas a grande maioria sabe o que é um ecossistema, está por dentro, identifica-se com ou conhece o conceito de agricultura sustentável, mas quando chegamos à questão dos serviços dos ecossistemas, aqui o padrão mudou. E, portanto, como estava a dizer a colega há pouco, a comunicação, entendermos, falarmos uma linguagem que todos entendamos, se queremos arranjar soluções em conjunto, é essencial. E, portanto, é uma das primeiras dos primeiros conclusões ou ilações que tiramos aqui deste estudo. Relativamente às percepções que têm sobre os serviços ou benefícios que as suas explorações, a atividade agrícola fornece, dividindo isto em aprovisionamento, culturais, regulação e manutenção, percepcionam que os mais providenciados são de facto os alimentos. Relativamente aos serviços culturais, contribuem para a beleza da paisagem, a sensação de pertença a um lugar e também atribuir um grande valor simbólico e espiritual às suas explorações, à atividade agrícola em geral. Em termos de serviços de regulação e manutenção, os percepcionados como mais providenciados estavam relacionados precisamente com o solo, que já falámos aqui muito, que promove a estrutura do solo, a sua capacidade de reter água e contribuem para a sua fertilidade. Houve ligeiras diferenças, mas não muito, entre respondentes homens e respondentes senhoras, em que as senhoras tendem a percepcionar uma menor provisão destes serviços. Relativamente àquilo que consideravam como impactos, sejam positivos, negativos ou então neutros, das suas atividades nos ecossistemas, nas zonas envolventes, percepcionam, de uma maneira geral, que os seus impactos são positivos: saúde humana, qualidade do solo, qualidade do ar, e ligeiramente menor para as questões da biodiversidade. Mas, no geral, de facto, percepcionam como positiva a sua atividade. Uma outra questão que fizemos foi como é que vêm as zonas envolventes, os ecossistemas à volta, e que influência têm nas suas atividades e na sua produtividade. E tudo que se relacione com água, vegetação, florestas, de um modo geral, consideram positivo. Áreas artificiais, grandes áreas agrícolas, matos, não tendem a percepcionar como um grande contributo positivo para as suas explorações. Portanto, aqui, mais uma vez, a questão da educação, da comunicação, é importante entre os vários atores. Quanto à influência da biodiversidade para a sua atividade e para a produtividade, tudo o que seja invasor, claro que está aqui o nome invasor aqui a condicionar um pouco as respostas, mas isso é considerado, obviamente, como negativo, mas tudo o que seja polinização, tudo o que seja controle natural, por exemplo, de insetos e por aí fora, tudo é considerado bastante positivo para a sua exploração e atividade. De uma maneira geral, praticamente 100%, quando fizermos assim uma questão, no modo geral, a vossa atividade contribui positiva, negativamente, para a sociedade, para a natureza, e as suas respostas são, de facto, unânimes, ou praticamente unânimes. Estes agricultores e agricultoras são atores que valorizam muito a sua atividade. Depois de conversarmos um bocadinho com eles e de nos clarificarmos aqui os conceitos, de facto percepcionam a sua atividade como capaz de providenciar múltiplos benefícios, portanto, e tem grande valor aquilo que fazem, e também no seguimento que disse a colega de manhã, estão dispostos a mudar para práticas mais amigas do ambiente, que promovam a biodiversidade, que promovam os serviços de ecossistemas, desde que não percam a sua rentabilidade, a sua produtividade, que sejam reconhecidos por isso, que os seus produtos tenham valor, que tenham acompanhamento técnico, e não se focaram só na questão de que precisam de subsídios. Portanto, foram muito mais além disso. A procura de soluções passa não só por pagar, mas também por um conjunto de outras soluções. Próximos caminhos. É necessário continuarmos este diálogo, é necessário que aquilo que fazemos de projetos ou de academia, agora pedimos apoio e agora temos que dar, não é? Temos também que contribuir para o crescimento, temos que dar feedback dos nossos resultados, temos que procurar em conjunto como é que devemos proceder, o que é necessário para mudarmos, para fazermos a tal transição agroecológica tão necessária nos tempos que correm, e portanto temos que co-criar caminhos, temos que co-criar soluções. E pegando aqui roubando aqui parte do slide à minha colega, de facto este caminho é preciso fazer, mas é preciso incorporar também estes atores na procura destas soluções, das melhores estratégias para o território. E só terminar por dizer que isto é um trabalho de equipa, com muita gente, uma atividade que consome muito tempo, e quero agradecer a todos os colegas e em particular a quem respondeu e nos ajudou a obter estes resultados. Muito obrigada. Esperemos nós para o nosso colega Fernando Polido finalmente apresentar a sua comunicação.
Está online.
Já está online, Fernando. Vamos ver se é desta. Se não, expulsam-nos aqui.
Ok, ele pode apresentar e nós...
Queres passar, Paulo? Ou passo eu?
Consegue ouvir, Fernando? Não sei se eu consigo ler. Consegue nos ouvir, Fernando?
¿Está bien?
Sim, nós vamos pôr a sua apresentação, ok? E vai-nos só dizendo quando é que é para mudar, está bem?
Es que yo oigo muy mal.
Ok.
Fernando,
¿Sí?
vas presentando y me dices cuando quieres que cambie.
Não, ali naquele
Sí,
computador
mira,
não é.
es muy fácil. Cuando yo haga una pequeña pausa, pasas la siguiente.
Muy bien. Pero si no me doy cuenta, dímelo.
Ok. ¿Ya?
Sí, sí, puedes. Estamos viendo.
Es que yo no veo vuestra imagen.
Puedes seguir.
Well, sorry for this delay. As I told you earlier, I am working at the University of Extremadura in project Mosaico. We have been trying in the last 7 years to implement a fire smart landscape in the northwestern Extremadura region, which is an area that has been suffering from extreme wildfires in the last decades. Next one.
Aguarda, que no está cambiando aquí. Setas. Sí, vale.
Okay,
estamos en el primer slide, después de presentación.
Es que yo no veo lo que estáis presentando. ¿Paso a la segunda?
Sí, vale.
Okay. In Southern Europe, we have, I will just summarize the idea. We have, as you know, around 80% of the burned area in Europe. And next slide, please.
Sí.
And this trend will continue and will even more intense in the future. As you can see in the predicted lines for the next decades, we will have a really sharp increase in the probability not only of fires but mega fires. Mega fires are defined usually as fires with more than 5,000 hectares in size, and so the probability of these events will increase in the future at a higher rate than in Central or Northern Europe. Next one. Usually, we attribute this problem only to climatic variables, but in Southern Europe, it is even more critical than climate. The problem with land encroaching or land abandonment. Here I saw 4 main drivers of the increase of fuel which feed the fires in the landscape. In the last decades, both in Portugal, Spain, and other Mediterranean countries, we have seen an increase in tree planting, an increase in the abandonment of tree crops, for example, olive crops or cherry or chestnut, etc. in mountain areas. We have also seen a decline in the grazing activity, especially in the mountains, and also a decrease in the use of traditional wood fires. Wood fires are small fires with a clear goal, and we have lost this opportunity to eliminate small amounts of fuel with this traditional tool. The next one, please. We have been working, as you can see in the map, in an area close to central Portugal, in Sierra de Gata and Las Urdes, and we are, of course, still working with many people in Portugal because the fire issue is a common problem. Next one, please. And in our area, you can see in the picture the small red dots are fires of different size occurring in the last couple of decades, and in the small in the small map in the right corner, you can see that in our area many fires are reiterated. So the frequency of the fire in a given point is very high. We have some places where 2, 3, or 4 fires in just in very short periods. So this is the problem we are trying to address by trying to change the landscape. Next one, please. Well, this is a nice picture from the fire we had last year, and this is a very common situation in our area. After the fire, we can see that there are many green islands in which especially tree crops or different kind of crops have been very useful as fire breaks. Okay, so from this picture and our experience in the last years, we can conclude that this is one of the main tools that we can use for managing the landscapes in the future. No? We have to promote this kind of fire breaks that are maintained just by their productive or economic uses, and with no cost for the administration. Next one, please. Okay, so our main tool in the projects are called productive fuel breaks. Okay, you can see a couple of examples in this slide. In the left side, we can see the conventional fire break in the mountain, which usually are not very useful for stopping the fires. And in the right side, you can see a productive fuel break. In this case, it is an area planted with young chestnut trees, but of course could be with other species. In this way, we are facilitating the firefighters to stop fire, and at the same time we are generating food products. Next one, please. Well, in the project that we started 7 years ago, we have tried to engage a lot of local actors, and here I just saw the evolution in the number of people, in the number of initiatives conducted by local people through this long period. As you can see in the red line, we have a continuous increase in the participation of people and the number of projects. And we still, the graph is not updated, but we still in 2023, we still have an increase and a lot of proposals for creating new productive fuel breaks in the territory. Next one. Here I just saw some examples of what we are doing in our landscapes. We can use, for example, goats or sheep grazing both in the linear fuel breaks and in the forest to reduce the amount of fuel. We can also thin the forest, especially the pine forests, which are very flammable, and we try to make them forests with a lower density of trees, in which we also can introduce sheep or goats or cattle. Also in the lower part, in the left, we are trying to stimulate and to promote resin tapping by the local people. We have almost 1,000 hectares in which these local people are harvesting pine resins, which is a very profitable product. And this is, of course, another kind of productive fuel break, especially because these people working in the forest are very useful also for controlling vandals or these kind of people that can burn the forest intentionally. And the last picture corresponds to a chestnut crop. As I said before, we use different tree crops and also vineyards or other species that can be grown in this area in between the continuous pine forest. Next one, please. This is a summary of the results in the map. In this area, which is about 100,000 hectares, we have been able to implement more than 100 fire breaks that are maintained through agriculture, forestry, and livestock. We are now managing about 6,000 hectares in this area, and many of the areas you see in these photographs are located in strategic places for fighting the fires. No, they are not randomly distributed across the landscape, but of course they have to be profitable for the farmers. But many of them are located in good places from the point of view of fire prevention. Next one. Well, this is another chart showing the results. Basically, if you look at the photographs, we can see the kind of changes in the fuel that we are implementing through cultivation, resin harvesting or grazing, etc. We have also analyzed the kind of changes, the kind of transitions in the fuel models that we are provoking with these farming activities. Next one. And as a summary, I am just trying to finish already. We can conclude that through theoretical simulations of the potential burn area, we can conclude that, of course, the size of the fires, especially of the big fires, is being reduced as a result of our activities. No? There is a very significant relationship between the area that we are trading with different initiatives, local initiatives, and the theoretical mean burn area in the territory. So this is a very good conclusion, and this is prompting us to continue our work. Next one. And as the final slide, I just want to add the relationship we have with the local stakeholders that are also selling the products with the market label that you can see in the slide. We are using this label to communicate the consumers that all these products, cheese, meat, olive oil, or essential oils, or many other products that we are working on, these products come from different operations that are helping us to combat the fires. No? And we know that the consumer appreciate this kind of products, and we have a very strong demand from the producers to reshape this label from Mosaico project. Well, I think I can conclude with this slide. Thank you very much.
Gracias, Fernando. Estuvo bien.
If anybody wants to, I don't know if anybody wants to present some questions.
Se quiser falar aqui, tem aqui só.
Alguma pergunta, alguma questão? Any question to Fernando? Or so, if there isn't any questions. Obrigada, gracias Fernando por tu presentación. Foi uma experiência. Vamos. Obrigada, Fernando. Vamos já continuar. João Paulo.
Sim, e vamos continuar com a Teresa Carita, que vem do INIAV e que vem apresentar-nos a comunicação Production of Quality Forage using Technical Itineraries aiming at the conservation of the soil and reducing the application of phytopharmaceuticals e é do projeto PPR Project.
Olá, boa tarde a todos. Apesar dos percalços, acho que a organização está de parabéns porque chegámos aqui com muita eficiência. Nos dias sempre acontecem estas coisas. Pronto, eu venho do INIAV e obrigada pela oportunidade de estar aqui a apresentar este projeto que se intitula, como o nosso moderador já disse, a produção de qualidade, de forragem de qualidade, usando itinerários técnicos que prevêem aplicar técnicas de agricultura de conservação e a redução da aplicação de produtos fitofarmacêuticos. Em Portugal, os sistemas pecuários verdadeiramente extensivos são, de facto, um sistema produtivo muito importante, quer a nível da gestão do território, quer a nível económico para o nosso país. E são sistemas que prevêem, ou que têm como objetivo, a produção de ruminantes à base de pastagens e de forragens. De facto, em Portugal, e daí a importância deste sistema também, as pastagens permanentes ocupam mais de 50% da nossa superfície agrícola utilizada. E a produção de forragem e a sua conservação são de facto muito fundamentais para garantir uma alimentação equilibrada nos períodos de escassez de erva que, em função de vários aspectos, são cada vez maiores e mais prolongados. Acontecem mais vezes e são mais duradouros, é o que eu queria dizer. E é muito importante também conseguirmos incrementar a sustentabilidade, melhorar a sustentabilidade económica e ambiental deste sistema produtivo, porque temos que responder bem à procura de carne, portanto temos que produzir em quantidade e em qualidade carne para a qual ainda existe bastante procura. Tendo tudo isto em conta, justifica-se termos proposto e termos em execução este projeto que tem como acrónimo GEBOV-MIT, que é um projeto apoiado pelo PRR e é liderado pelo INIAV, a instituição a qual pertenço, mas só o conseguimos implementar e executar porque contamos com o apoio de várias instituições, onde, por exemplo, está o Instituto Politécnico de Castelo Branco e a Sociedade Portuguesa de Pastagens e Forragens, da qual o nosso moderador é o seu presidente. Os objetivos operacionais desta ação, deste projeto, é a redução de gases com efeito estufa e o aumento do sequestro de carbono nos nossos solos. Como objetivos muito específicos, e como já o próprio título do projeto o indica, queremos produzir erva de qualidade, erva para conservar de qualidade, aplicando menos fertilizantes, principalmente os azotados, e ao mesmo tempo que conseguimos contribuir para a conservação do solo. Para tal, implementamos várias atividades, começam pela sementeira direta, que é uma técnica de agricultura de conservação. Optamos por instalar e monitorizar diferentes misturas de forrageiras, misturas biodiversas, e a sua monitorização é feita aplicando algumas técnicas de agricultura de precisão. O que é que nós queremos no final do projeto? Queremos, ou pretendemos, e esperemos que assim o consigamos, reduzir 20% da aplicação de agroquímicos por ano e aumentar em 30% ao fim dos 3 anos no teor de matéria orgânica do nosso solo. E muito obrigada pela vossa atenção.
Obrigado, Teresa. Eu chamaria então agora o Luís Duarte, que vem do MED, da Universidade de Évora, um centro de investigação da Universidade de Évora, e que nos traz a comunicação subordinada ao tema How do Territorial Approaches Contribute to the Sustainability of Agri-Food Systems? A Systematic Review.
Well, I can go on and... Excellent. Well, good afternoon. I will do this in English, so we are like alternating languages. But I guess for me it will be definitely faster, so I don't have to be here on my Portuguese and translating the whole thing. Well, my approach is a little bit different because I'm more on food systems, and the Congress has been, and the Congress has been quite a lot related to more chemical and biological and laboratory issues, although this theme here is not that much. Starting by the beginning, I'm a PhD student in agribusiness and sustainability. That's my graduation. It's a consortium between 2 Portuguese universities, University of Évora and UTAD, which is in Vila Real, in northern part of Portugal. Although I am affiliated to the Med Institute, as you can see there, and my supervisors in the Med Institute are also co-authors of this article. This is basically a literature review, and it focuses on the relation between territorial approaches and the sustainability of agri-food systems. The title appeared by the end of the article, because at the beginning the title was not exactly like this, but then we realized it could really work well on this way. Is it moving? Okay, just to give a general view, what we are looking here is to understand territorial interrelations established within sustainable agri-ecological practices, and as a literature review to assemble the bulk of scientific production, examining agricultural practices that are based on biodiversity and integrated into non-conventional food networks. Oops, I guess I lost something there. Okay, literature review highlights the scientific trends. Well, I'm not going deep into this, but this was the search criteria that was used. And on one side we have the territorial approaches, on the other side basically what we understand as sustainable agriculture. As a short presentation, need to keep going. At the end, we got 349 papers, articles. You can see this was a bibliometric analysis, so lots of statistical stuff here, but I'll keep it real short. You can see the evolution of the scientific production through time, and I will just give this result here because it will link to what comes afterwards, which is a co-occurrence network of one indicator, which was Q-Earth Plus in those 349 documents. And the result here is interesting because it highlighted 3 clusters. And we can see on those studies, we have the bigger cluster, this red one, based on agroecology, and also centered on agricultural production, agricultural management as a sustainable practice, and based on biodiversity, knowledge, and innovation. Then we also have the blue cluster, more on the agro-ecosystem dimension regarding sustainable agriculture as a means of producing healthy nutrition, viable livelihoods, and environmental protection. And then the green cluster is more related to rural development and food sovereignty, and also approaching food supply and consumption as well as governance. Well, this is just one result, but we gather the bulk of bibliometric results we could find from our review, and from there we are proposing, and this is a critical review, we are proposing segmentation of 3 approaches that are focused, one, basically on agro-ecosystems, so at the field scale, regarding words, concepts like sustainable farming, nutritional food, environmental health, and farmers' livelihoods. Then a second one, agro-ecosystems, but from a holistic perspective, so more on a landscape and or on a regional scale, bioregions, for example. And in here we have the concepts of agroecology, of course, knowledge, innovation, biodiversity, sustainability, and finally a third scientific group in these studies focused on the food system, on the whole food system, and so with a more national scale regarding issues as sovereignty, governance, and the relation consumer-producer or alternative food networks, FN.
Luis.
As conclusions, yes?
Yes.
I'm getting to the conclusions, thank you. The transition to more sustainable agri-food system requires a holistic and transdisciplinary approach, supported by systems thinking, very important, and place-based approaches that are linking economic, social, environmental, and political issues are also getting very important. And in terms of governance, we need approaches at multi-level, multi-actor that can realize the potential for territorial approaches. And finally, just an idea for the future of research, a more broadly holistic and systematic understanding of what a sustainable agri-food system can bring to a community in the region is something that is still scarcely realized and needs research on that. And so, thank you very much.
Muito obrigada e vamos terminar com a apresentação de Eduardo Nascimento, muito obrigada, da Universidade de Coimbra, com o tema Influence of Land Use and Crop Types on Organic Matter Composition in Mediterranean Agroecosystems and Assessment using the TBAG Index Methodology. Força.
Ela dá sua. Tá por aqui? Aqui, onde que pode ir? Esta é sua. Espero que sim, mas se não for, eu falo também.
Well, hello, good afternoon. Thank you very much for being here and for your attention. My name is Eduardo, as said, and I'm here to present a work that we've been doing in Coimbra, in which we are assessing the composition of organic matter, and how the land use and land management can influence that. So, I start with a very random and maybe funny question, but can drinking tea help us understand soil health? We could talk about that, but I only have 3 minutes, so I'll just skip to the answer, which is yes, we can help to understand soil health by drinking tea. But how? The thing is, soil health plays a very important role in the sustainability and functionality of agro ecosystems, both in crop yield and in natural resources conservation. So in this case, understanding the composition of plant residues is very important to understand not only the whole ecosystem, but the nutrient cycling and everything that's involved within it. Based on that, in this study, we adapted a methodology called tea bag index, which is literally tea bag as in a bag of tea. And we have this methodology, which is well described. And this methodology uses 2 types of tea bags, one made of rooibos tea and the other one from green tea. And basically you just set that on the soil and you leave it to decompose. So basically you have the initial weight, you have the final weight, you have the difference, and then you can assess the decomposition rate in a very easy and affordable way. On our study, we adapted, and we didn't use both tea types, we only used the green tea. As you can see on the right graphic, the green tea has a faster decomposition rate, it drops faster than the rooibos tea. And it can be a very good indicator of decomposition because it decomposes faster. And we actually put 112 tea bags on the soil of 3 different land uses: olive groves, almond orchards, and pastures. And they varied within like the structure of their use, their management, the intensity of it. We found some very interesting results. At first, we definitely saw that, as you can see, indeed the land type, the crop type or the land use would influence. So the almond orchards had a lower mass loss percentage, which means that the composition was slower, and the olive groves was faster, so it had like more decomposition within the same time. On the other hand, we didn't find any significant difference between management or with that being, we had like olive groves and we had almond orchards, and they have like intensive, super intensive, traditional types of management, and they didn't vary, so the management wasn't ideal. But we did find something very interesting when we were checking the pastures. The decomposition rates within pastures, like they were significantly different, and the difference was between the pasture, mainly pasture number 1, which was a montado, a natural montado, dehesa in Spanish, cork oak forest. And in pasture number 3, which was an improved kind of natural or traditional pasture with cattle and everything. As you can see from the photo 1 and the photo 3, these are like the main differences between the pastures. Pasture 1 has like cork oaks, pasture 3 doesn't have any tree, but still they have like the same, more or less the same grasslands, and also the kind of the cattle is different, and also the soil treatment is different. The vegetation type, the irrigation type of the soil, you can have soil tillage, grazing without control on pasture 3, and we believe that that is the reason for those data here. So, in conclusion, we can find that land use type and management influence the composition of organic matter, and this methodology, the tea bag index, it can be applied to compare decomposition rates. It's a very fast, very viable, very easy way to assess, And it will test the biological activity. We can imagine as a snapshot, like a fingerprint of mass loss. We can see on that moment how the decomposition rates are going, and I think in this study we could do that. And basically, yeah, next time you are drinking tea, you can think that you can actually do science with it. So with that being said, thank you very much for your attention. Thank you very much for all the agriculturists and the agricultors, then the farmers that worked with us, and the team who helped with us with all the work. And that's it. Thank you very much, guys.
Muito obrigado, Eduardo.
Não,
Estamos
obrigado.
um pouquinho atrasados na hora, mas daria a oportunidade para se alguém quiser fazer alguma intervenção, algum pedido de esclarecimento. Está além já um colega atrás.
Thank you very much to you all for the very informative talks that we had this last hour. So I have the question to Eduardo. Eduardo, regarding did you evaluate the release of, for instance, tannic acids from the teas? Because, well, I will try to. Well, it's a very long question, and I will try to show the hypothesis that I'm trying to suggest. So, the teas, they have antioxidant compounds such as, well, some polyphenols, and these polyphenols, some of them, they have antimicrobial properties. And for instance, in the montado, the trees that are there, they release a lot of tannins. In fact, the oaks, they were used in the past for tanning because of their polyphenols. So did you analyze, what I'm suggesting is that perhaps you have a microbiota population in the soil that is more resilient to these polyphenolic compounds that are being released by the tea, so they will degrade faster because they are resilient to that. So, and when you have improved pastures, sometimes it's used, like, for instance, leguminosas that incorporate nitrate in the soil, so they can inactivate sort of the action of the polyphenols. So can you comment on these hypotheses that I'm suggesting?
Sure. Very good question, by the way. I don't know if you were earlier when Ricardo was talking about the Cultivar project. We actually have 14 factors that we are analyzing. We are analyzing biological, chemical, and physical factors of the soil. We are analyzing soil aggregates. We are analyzing pH, moisture. We are analyzing a lot of things, including microbiota. We are doing like fauna, mesofauna, which can actually help to dissolve, to decompose the litter. And like I said, this part here is like a snapshot. When you compare everything, you can have an idea on which system is being more biologically active. We are not trying to say who is there. We know that's something there. But based on the other experiments that we are currently doing, we're doing microfauna, mesofauna, macrofauna, studying fungi, bacteria. We are doing everything. We are studying DNA. Based on that, I think we can answer more clearly to your question. But it's an ongoing project. So we will have to probably wait a little bit and add these layers that you mentioned, which are very good suggestions, by the way. We can add these layers so we can have a bigger, a better vision of the picture we are painting. So yeah, based on the other factors that we are studying, I do believe that we can go there and study, because this specific methodology is well described, because like I said, like all the phenols, ergosterol, lignin, phosphorus, nitrogen, all that is like within the litter or like the vegetal part of the the plant, and yeah, it's well described. So yeah, along with everything, we can answer better later when we analyze everything else.
I hope it
Muito
answered your
obrigada.
question.
Nós não temos tempo para mais questões. Eu queria agradecer a todos por cumprirem o tempo e provavelmente o Fernando também cortou ali metade da sua apresentação. Muito obrigada a todos e perguntas no coffee break a seguir certamente estarão disponíveis para falarmos. E vamos seguir já para o próximo tema. Muito obrigada.
Thank you all for your presentations. I will call now to the stage Pedro Assuda from the Nova Delta.
Havia uma pessoa que faltava. Havia uma pessoa que faltava por aqui a apresentação. Acho que nunca vejo aqui.
Boa tarde a todos. Em primeiro lugar, agradecer o convite para estar aqui como moderador. Para mim é a primeira vez, já estive ali no outro lado, mas muitas vezes, mas também é um desafio e espero fazê-lo cumprir dentro daquilo que são os objetivos deste evento. É uma oportunidade única também para estarmos juntos, não é? Os vários stakeholders deste setor, do setor agroalimentar. Eu estou aqui em representação da Nova Delta, portanto, a unidade industrial do grupo Nabeiro Delta Cafés. Hoje não vou falar tanto da nossa área, mas vamos aqui introduzir a temática que a todos interessa, que é o agroalimentar. E para isso eu chamaria os intervenientes deste mesmo painel. Portanto, estamos a falar que o nosso painel, vamos dar início ao painel Food Disruption, Making Sustainable, Healthy, Delicious and Appealing Foods. Eu vou chamar Belém Blanco, que é Head of Food Area at Agri-Food and Process Division in Cardiff. O Rui Lopes, da Medronho e Canela. Pedro Babo, da Landratec. Susana Ramos e Ana Rodrigues, da Schreiber Foods, Portugal. Gonçalo Costa, da Cricket Farming. E acho que estão todos. Olá, boa tarde. Obrigado a todos pela vossa participação neste painel. Portanto, também vou precisar da vossa ajuda, porque para mim é a primeira vez aqui deste lado. Portanto, já estive aí muitas vezes, como eu tinha dito há pouco. Portanto, mas vai correr tudo bem. Estamos aqui, de facto, e vamos ter aqui a oportunidade de ouvir os diferentes intervenientes nas suas áreas, falarmos um bocadinho do agroalimentar, que é, de facto, reveste-se de grande importância para a economia nacional. E também, obviamente, apostando sempre na internacionalização, na inovação, e vamos aqui abordar algumas temáticas, com certeza ligadas à inovação, à transformação digital, à transição digital, que é, de facto, importantíssima nos dias que correm, mas não esquecendo que a questão da sustentabilidade, de facto, também para sermos mais competitivos, as nossas organizações serem mais competitivas. Eu só queria deixar aqui, antes de começar e passar a palavra à primeira intervenção, de facto, o setor agroalimentar em Portugal, esta indústria é a indústria transformadora que mais tem contribuído para a economia nacional. É um setor estratégico para se atingir aquilo que são os objetivos económicos e sociais, seja naquilo que seja o seu papel dinamizador na própria fileira, também pelo desenvolvimento rural, pela criação de emprego ou pela preservação da identidade cultural. Se não vejamos, tem um valor em termos de volume de negócios aproximado dos 18 mil milhões de euros, que é bastante significativo para a nossa economia, representa em termos de VAB 3,4 mil milhões de euros. Em termos de postos de trabalho, estamos a falar de 120 mil postos de trabalho diretos e 500 mil indiretos. E, a nível de exportações, qualquer coisa como 6 mil milhões de euros em exportações. Portanto, são números de grande peso, não é, para aquilo que é a nossa economia. Grandes desafios e oportunidades, e depois mais à frente vamos ter a oportunidade também de todos podermos falar um bocadinho sobre esta temática. Aqui, e como outros setores, correspondem inúmeros desafios e oportunidades, e que estão enquadrados no conceito da sustentabilidade, que abrange contextos ambiental, económico e social, não esquecendo cada uma destas vertentes. É com base nesta abordagem que deve ser refletido e desenhado e colocado em prática aquilo que é o futuro modelo dos sistemas alimentares. O debate deve ser nacional, mas não só. Por isso também temos convidados internacionais e dos quais agradecemos a sua presença neste mesmo painel e neste evento do Farm to Fork. Que é isso realmente que definiu a União Europeia como estratégia, portanto, estratégia do Prado ao Prato, ao Farm to Fork, da Comissão Europeia. E era deixar este enquadramento da importância que o setor agroalimentar tem, e passaria a palavra da primeira intervenção, Belém Blanco, que é responsável pela área alimentar da divisão agroalimentar e processos do CATIF. No seu trabalho procura melhorar os sistemas alimentares sustentáveis através da implementação de ações e também melhorar as propriedades nutricionais e tecnofuncionais de diferentes matérias-primas, como farinhas ou subprodutos agroalimentares, através de tecnologias de processamento que permitem acrescentar valor a estes ingredientes e subprodutos. Quando quieras, Belém.
Let me use the okay. I'm not able to move the presentation. I'm sorry. It's here. Okay, but is here also pizza?
Yes. No, in this.
Okay, thank you.
Thank you very much. Boas tardes a todos. I'm sorry I cannot speak in Portuguese, but I have to do it in English, so I hope everyone understands my Spanish English. So thank you to the organization for inviting me to this event. I just want to present a journey from agri-food to lab-driven innovations from our point of view as a technological center. Okay, I have some problems with it. Presentation, but I'm trying to solve with your help.
I don't know what happened.
I think it's not in complete screen, maybe?
Yes.
Okay. Now it's moving. Okay.
Sometimes it does not move in the computer when you move there.
Okay. So it's a little bit delay, maybe? Yeah. Okay. I'm sorry. Okay, I want to introduce what is CARTIF, the center where I work. CARTIF is a technological center, which our mission is to generate, to generate technological knowledge and to propose innovative ideas to the companies and also to the partners that we work with that can improve their competitiveness. That is our objective. So we are based in Valladolid, in Castilla y León, the region of Castilla y León. Some figures of our center are we are more than 200 people working at CARTIF, and we are working on more than 140 projects, ongoing projects. More than half of the projects, up to 50%, are European projects, mainly. So what are our business models? We develop R&D, research and development projects for companies, offering personalized technological service and also technological consulting on the highest level. We develop also R&D projects financed by public fundings, reaching national and international competitive goals also. Advise public administration, city councils and region governments in the planification and development of innovative projects with high economic return. Some of our clients of CARTIF, we work with all kind of industry, from automotive car, so energy side, and also, of course, food industry. I'm sorry because I see one thing here and one different in this part, so. Okay. It's being hard, but. No. I'm in here, so I can see the presentation here, but not.
Oh, okay.
I don't know what is happening in this. I'm sorry.
So. No, it's not changing actually. Can we close it and open?
Yeah.
Because sometimes computers are not.
Yeah. Close and open or just escape, maybe?
Maybe just close the whole program and.
Let me see. Okay.
Okay, let's close.
No. So it's quite weird. I'm explaining one thing and the slide is different.
Okay. Let me open again. Okay. Let me see. Sorry. Agora nem sequer vejo o rato. Não vejo o rato, ok. Ok. Mas tem que projetar. Ok.
Aqui.
Oh.
Sometimes the secret is just shutting down.
Thank you so much. Okay, what we work in Cardiff, we are 3 big divisions. One is energy, that works in smart grid, energy and climate policies, smart cities, and so on. One is industrial digital systems. They work in Industry 4.0, infrastructure, also health and well-being. And the other one is agri-food and processes. That is the big division where we work. Which areas we have? Natural resources and climate, circular economy, biotechnology and sustainable chemistry, and also food, and also, most important, the area that I lead, that is food area. What are our research line and also the work we do? We work with food cities, we work with urban food system transformation, food 2030 methodology, using the food 2030 methodology with the source supply chains, healthy and sustainable lifestyle, consumer citizen engagement and education. We are, as you know, coordinators of the project Fusili, and this is one of our areas of work. We also work on food and health. Target nutrition, functional product development, product substitution, reformulation, doing the products more healthy and also more sustainable. More sustainable in the line with the valorization of byproducts. We work with this, I'm going to explain later. And also we work with food security and food safety. So we are focused on the sustainable food system. That is the theme that brings us here. What is a food system? This is supposed to be the actors of the food system, but all we know that the food system is more complex. It's a set of complex interrelation between actors and factors of the amazing part of the food production, processing, waste recovery, and distribution, all the parts of the food system. Just a little bit of context to explain what we're trying to explain today. So we are in a rapid increase of the global population, as we all know. Food and health is very important for us. We have risen the importance of the conscious binomial of food and health. Also, food production and the way we produce the food is important for us. So we work on the side of healthy, safe and sustainable food. What initiatives or tools or regulations we have? We have talked about that yesterday, the Sustainable Development Goals. There are a set of global goals adopted by the United Nations as part of the 2030 Agenda for Sustainable Development Goals, covering a broad range of interconnected issues. Also, in the European Union, we have the European Green Deal. That is an ambitious initiative launched by the European Commission to make the European Union climate neutral by 2050. The Green Deal encompasses a set of policy initiatives, very important, aiming to transform the European Union economy to a more sustainable and circular model. So one of the strategies, 5-4, is a key for specifically focus on food system. That is the big question of this Congress. So how can we develop sustainable, healthy, delicious and appealing food within all this context? It's something really complicated, scary. So we are trying to bring innovation to the lab and create our own film that takes the name of Journey from Agri-Food to Lab-Driven Innovation. We present to you, taking into account some specific questions and align it with the research lines of our area. So we work in food and health and valorization of food byproducts, and we work with food security. So we try to align all of these and try to do something innovative. Innovation is not easy, unfortunately, we all know, so we have to create a new concept. And we decide to work with this technology and align the needs of the market and the needs of the world to provide a solution for the market. And we work with this technology, that is extrusion technology. Maybe most of you know this technology. Basically, it's a kind of reactor. You put the raw materials, some water, and specific parameters that are temperature, pressure, also the specific mechanical energy that you do as an input of the system with the screws. In our case, it's a core-rotating screws, so you have a complex activity inside the reactor. So the raw material is mixing, cooking, gelatinization in case of the starch, heating, evaporation, pressure, all these factors occurring at the same time till the raw material reach the die and get expansion at the end of the reactor. So for what we use extrusion technology? Mainly for food development, like expanded snacks, breakfast cereals, flatbread also, co-extruded snacks also is possible, meat analogs, because we have also a system after the extrusion system for texturization, and also aquaculture fish feed production is possible to make with the extrusion technology. But where is the idea? The idea is to create an innovation, so we work with raw materials, abundant raw materials coming
organizado que tem produção anual, cujo produtor faz a limpeza e esta faixa atua como uma faixa de contenção de fogo. Aqui embaixo uma imagem de uma exploração no concelho de Oleiros, aqui bem próximo, e como podemos ver, veio um incêndio e esta mancha verde é um medronhal que foi poupado e que permitiu, que em alguns casos, permite, e neste caso não se vê muito bem, mas a imagem completa, permitia perceber que ele atuou como uma barreira contra a propagação do fogo. Portanto, este arbusto tem uma importância não só nutricional, não só económica, mas também nos ecossistemas, porque queria dar aqui uma nota que não está aqui nos slides. Nesta altura do ano nós já estamos em plena campanha, um pouco por todo o país, e nós encontramos simultaneamente no arbusto flor, folha e fruto. Nesta altura do inverno, eu não percebo nada de polinização, estão aqui especialistas da matéria, depois se me quiserem acrescentar alguma coisa. Nós temos centenas, milhares de insetos polinizadores e que precisam de se alimentar neste período de inverno. O medronheiro é um fornecedor de alimento e, portanto, contribui ativamente durante todo o ano para a preservação, para o fortalecimento e resiliência dos ecossistemas onde ele está presente. Portanto, ele oferece um impacto positivo nos ecossistemas, mas também não só nos ecossistemas naturais, mas também nos ecossistemas humanos. E é dessa perspectiva que eu vou falar um bocadinho a seguir. O que está aqui já falei anteriormente, só para avançar um pouco. Só uma notazinha, uma coisa que eu descobri recentemente num trabalho de pesquisa, que não fazia ideia. Não sei explicar isto de forma científica, porque a minha área não é botânica, a minha área é de nutrição e de gastronomia, mas descobri um artigo muito interessante que o medronheiro, por estar há milhares de anos entre nós, desenvolveu uma capacidade de modulação quando está sob stress hídrico ou quando está sob stress de qualquer ordem. Modela a sua fotossíntese de modo a manter a mesma taxa de captura de carbono. O que quer dizer que numa altura em que se fala tanto das taxas de captura de carbono e do pagamento a essas explorações, o medronheiro pode também ser um importante contributo para estas economias de territórios de baixa densidade. Era aqui que eu queria chegar sobre a sustentabilidade para as economias rurais que podem viver da cultura do medronheiro. É uma comparação, é um case study feito pelo engenheiro João Dias, um amigo que é presidente da Cooperativa Portuguesa do Medronho. O João Dias trabalha há muitos anos, quase 20 anos, em pomares de maçã de alcobaça e pera rocha do Oeste. As práticas cultivares de cultura que o João aplica nas empresas para quem trabalha, eles trabalham em pomares organizados em modo integrado e, portanto, este case study que apresento de seguida, ele é com base em pomares de medronho. Uma comparação entre uma exploração de eucalipto e uma exploração de medronheiro, e o quadro é para 1 hectare e temos aqui. Depois podem ver com mais atenção esta apresentação. Mas eu queria que se centrassem aqui nestes resultados. 1 hectare de eucalipto pode ter uma rentabilidade de 4.800 euros hectare à vida. Os eucaliptos, no máximo, vão até aos 24 anos. Nós sabemos que o medronheiro, aos 24 anos, continua a produzir, embora seja recomendada uma renovação das plantas ao fim de 20 anos, uma substituição, e a rentabilidade estimada pelo João, pagando a um preço de 2 euros o quilo, que está por aqui assim, qualquer lado, esse valor, pagando a 2 euros o quilo para uma produção de 4 toneladas por hectare, estamos a falar de um pomar que depois já vou mostrar a seguir o estudo comparativo com diferentes colonos, e podemos ter ao fim de 24 anos uma exploração que rendeu em termos de faturação 139 mil euros. A maior parte das explorações de medronho organizadas neste momento tem uma produção média entre 1,5 toneladas e 2,2 toneladas, o que quer dizer que se eu dividir este valor ao meio, ainda ficávamos aqui com 70 mil euros. Portanto, é simples fazer as contas. O que é mais rentável? É produzir medronho ou eucalipto? A resposta eu deixo naturalmente para a plateia. Continuando, este é o quadro que deu resultado para o quadro anterior. Eles utilizaram diferentes clones e cada um tem diferentes resultados. A produção média desta exploração era, ao fim do 5º ano de plantação, de 5.500 quilos por hectare. Ora, estamos a falar de uma plantação organizada em pomar, que tem um conjunto de intervenções, nomeadamente preparação do solo, com sistema de ferto e rega, correção do solo, irrigação gota a gota, enfim, um conjunto de intervenções para que as explorações sejam efetivamente rentáveis e para entusiasmar os produtores. Portanto, o que estamos a falar é de intervenção no medronhal, diferente daquela que nós estamos habituados. Quando se fala em medronho, isso é uma planta selvagem. O medronheiro, temos hoje em Coimbra, que é na Escola Superior Agrária, onde nós temos provavelmente os campos experimentais e de investigação que têm feito que têm maior contributo para a fileira. Eles têm imensos exemplos de que as plantas colonais e os pomares podem ser altamente rentáveis. Bom, avançando um bocadinho mais, porque acho que já só tenho um minuto, falar um bocadinho do que é que nós estamos a fazer na Medronha e Canela e no Politécnico de Leiria, ou o que é que nós fizemos. O nosso objetivo no desenvolvimento de produtos sempre foi a preservação máxima dos compostos bioativos ou sua incrementação, em particular daqueles com atividade antioxidante, e nós encontramos muitas dificuldades neste trabalho. Para terem uma ideia, eu estou a trabalhar com medronho desde 2010, fomos para o mercado em 2020 e até essa altura tínhamos gasto cerca de 110 mil euros em investigação e desenvolvimento de produtos e não tínhamos faturado nada. Estamos no mercado, portanto, há 3 anos e o primeiro produto que nós colocámos no mercado foi o pão de medronho, um produto que está em linha com tudo aquilo que nós temos neste painel. É um produto clean label, sem qualquer aditivo. Utilizamos farinhas do Alentejo sem qualquer correção e é um pão que tem um conteúdo nutricional bastante diferenciador, tem cerca de 20% das nossas necessidades diárias de fibra por cada 100 gramas, 2 alegações nutricionais, fonte de fibra, fonte de proteína, tem cerca de 19% de atividade antioxidante, óleos de cadeia curta da família de ômega 3 e ômega 6, vitamina B, C e E, enfim, é um pão que tem uma longevidade até 6 dias e portanto, é porque nós alcançámos este sucesso com este produto, desenvolvemos muito conhecimento e bastante tecnologia, entretanto colocámos mais um produto no mercado no ano passado, os bombons e trufas de medronho, mas não paramos de desenvolver produto, terminámos agora recentemente também 2 formulações para gelado, numa linha natura, outro numa linha incrementada em termos de aroma. Terminamos também um projeto com produtos para incorporar em pastelaria quente e continuamos o nosso trabalho de investigação e desenvolvimento de produtos. Trabalhamos essencialmente com estes 3 grandes centros de investigação, todos hoje aqui amplamente representados com alguns colegas do Politécnico, o CATAA, aqui pelo Cristóvão e Inês, e com esta grande organização. Lá atrás o Marco Alves do Inove Linha, são centros de investigação que nos têm dado suporte ao desenvolvimento de alguns produtos. Neste momento já estamos a trabalhar há algum tempo, embora em fases distintas, com preparados para iogurte, sumos naturais e smoothies e pastelaria fria. Por último, e indo já para terminar, estes trabalhos de investigação e desenvolvimento só são possíveis porque nós trabalhamos com tecnologias emergentes. Algumas dessas tecnologias são PEF, o HPP, o UVC, a pasteurização e a liofilização, tecnologias que nós encontramos nos centros de investigação no CATAA, no Inove Cluster e também no Baré, com outros parceiros. Temos colaborado também com alguns projetos de investigação, fornecemos matéria-prima para um grande projeto de investigação cujos resultados vão de sair entretanto, que estamos muito entusiasmados com eles, com o CATAA e com a Universidade Nova. Estamos neste momento a colaborar também com a Universidade da Beira Interior e com o LabFit para a extração de compostos bioativos de folhas de medronheiro. Nada disto se faz sozinho, apesar da empresa ser uma microempresa, eu sou o CEO e tudo o resto que a empresa necessita, mas há uma equipa de investigação que está por detrás, a Susana Silva, Susana Cardoso, a Ana Augusto, eu próprio, e para este trabalho tem contribuído também o meu amigo João Dias, que é engenheiro agrónomo. Portanto, o que eu vos peço para terminar é que plantem medronheiros, porque nós temos um grande caminho para este fruto no futuro e nós estamos a fazer um trabalho, acho que bastante disruptivo. Muito obrigado, fico disponível para qualquer questão.
Obrigado, Rui. Foi muito importante ouvirmos falar aqui sobre o medronho e sobre a importância que ele tem a muitos níveis, não é? Portanto, pudemos ter essa oportunidade. Obrigado mais uma vez. Parabéns por todo o trabalho também realizado nesse sentido. Vamos passar ao Pedro Babo, que nos vai... Portanto, o Pedro desenvolveu o seu pós-doutoramento com o grupo de investigação 3B, onde procurou estabelecer uma linha de investigação única em terapias regenerativas endodônticas. Foi professor convidado na Universidade do Minho, onde lecionou cursos de biomateriais e laboratórios de biocompatibilidade. O Pedro foi também convidado para liderar palestras no programa doutoral de bioengenharia do programa do MIT Portugal. A apresentação que o Pedro nos vai trazer tem como título A Forest to Fork to Industry, a Short Value Chain, Circular Economy Solution for Acorns from Iberian Native Forests. Obrigado.
Okay, thank you very much. Good afternoon to everybody, and well, thank you to the organization for the invitation to present here our business model in which we aim to change the paradigm or help to change the paradigm of the forest management in Portugal and Iberian Peninsula and hopefully in Europe, using our forest to fork through industry strategy to value the acorns. So it was already talked here by Belen that we are nowadays we cannot feed the population that we have today, and the increasing of the population it's putting much more pressure in our food chains, and which is it's actually struggling to feed us. Because of the length, like we are using very long supply chains that are susceptible to wars, to climatic changes, and to produce the food, we have to contribute to increase the climatic changes by producing greenhouse gases and destroying endemic forests to produce new fields. So to have a figure of the size of endemic forests that we destroy to produce new fields, like every year it's almost the size of Spain. So our question was, can the solution be in our endemic forests? We already saw that Darbuto Zunedo can be one of the solutions. But the European forests are mostly oak forests, and worldwide in the northern hemisphere, the oak forests, they are the most prevalent forests. We have more than 400 oak species that span from the temperate areas until the tropical areas. These species, they are perfectly adapted to all the climates. They evolved in these regions for millions of years, and they do not compete with arable land. In fact, these trees, they grow in marginal lands, and as we know in Portugal, in the agro-silvo-pastoral landscapes, as in Montado or Dehesa, like the oaks, they are very important. So, and the fruit of these oaks, the acorns, it was a staple food in the past. Like it is known, for instance, that in ancient Greece, they believed that the acorns was the first food that the gods gave to the humans. And we have also, like Pliny the Elder says that in Spain, they always have a space in their dishes for the acorns. But I prefer this one, the one in that one that talks about Portugal. So when Strabo wrote the geography, he said that in Portugal, the people in the mountains, they used to eat, for 2/3 of the year, they used to eat acorns as a bread. So it was a staple food in the past. And actually in Portugal, we are a net producer of acorns. If we sum together all the oak species, they are still more than the eucalyptus. So we are still acorn producing forest. 36% of our forest produces acorns. It represents almost 300 or more than 300,000 tons of acorns per year. It's 3 times the amount of grain, pulses and seeds that Europe imports every year to supply the needs that we have for gluten-free for instance, food, and it's produced only in Portugal. So, and it could provide, like these trees, they have years of low production and years of must. In a must year, you can have like from 1,000 to 3,000 kilos per hectare, which could represent more than 2,000 years of revenue each year per acre to the land producers. And why are we wasting this very abundant resource? Well, because like when we started to figure out how to build our business model, we observed that there was a very fragmented value chain to value this product. Well, as I said, like it is very seasonal, and the production varies every year. It is a lot of tannins, polyphenols, that make this fruit astringent. Also, the cultural prejudice: it is feed for animals, which is not true, actually. And it is a very expensive raw material because of the technical limitations. So in 2020, we founded Landrotec, a startup which aim at valuing our acorns, our endemic forest acorns, through a proprietary process that we developed internally, aiming to give a new ingredient to agri-food industry, but also to explore, in a context of circular economy, to explore the byproducts to other industries. So our systematic solution for the acorns is to provide the landowners that don't know how to value these acorns a network. So we build up a network of stakeholders that enables us to bring these acorns to the industry, mainly to the agro-food industry, but also to other industrial verticals. But we also develop technology that helps us to process, and we can also provide to the industry to produce food from acorns. So currently we have several products, like from edible acorn products to technology and digital solutions. Today I will talk only about the food products, which are an acorn, the caffeinated coffee substitute. We are not going to substitute the coffee, but it can be a solution for people that don't like or that cannot take caffeine. A gluten-free acorn flour, and also acorn tisane solution. And it is particularly interesting because we didn't invent anything. We just rescued traditional ways to eat acorns from our Mediterranean cultures. So this is our acorn decaffeinated coffee substitute. Like, in fact, we have a full acorn and a mixture with caffeine, because we are all addicted in Portugal. It's very important. It's very difficult to put a coffee substitute without caffeine in Portugal. So, like, we had to create this one that won an honorable mention by the Ocean Future Taste in a contest in which we were awarded with an honorable mention by Food Innovation Product. So I also have some figures here, so you can have an idea of the nutritional profile. So, like, for us, the most important, and for the public, the most important numbers is the fat. Like, it has fat which is rich in omega 6 and 9. In fact, it is also it has proteins, but it is gluten-free, and it is a source of fibers. And we can also, like, regarding the nutritional allegations, we can say that it is rich in potassium, and potassium is very important for the cardiovascular system and the development of neuronal system. And also it is rich in vitamin E, which is a very important antioxidant, and contributes to the protection against cells oxidative stress. We also have our gluten-free acorn flour, which has a very similar profile with a nutritional profile with a coffee substitute. The thing is that it is richer in fat, because, like,
for the acorn flour, and this is an issue that I am going to refer afterwards, like we can only use Quercus rutulifolia, the olm oak flour, because it's the only species that is listed in EFSA. And also it is rich in potassium, rich in vitamin E. And we are launching the acorn tisane infusion. It is very interesting because the supplements of acorns, they are listed in EFSA. We can use it, and traditionally in the European traditional medicine, this was used as for the regulation of the digestion. It is also rich in antioxidants, and we know, like by the content of fibers that we can quantify, that it really can influence the digestion and help in the digestion. Other way. So we can, with acorns, we can address 4 very important markets. The organic food. The acorns are produced in a very healthy way. So most of our producers, they have the production in biological, already certified in biological production. So we can address a world market that is growing in almost 2 digits. And it is in 2020, it was like 77.8, almost 78 billion euros. Also the gluten-free market, the coffee substitute market. In Portugal, we are really addicted in coffee and coffee substitutes, and the polyphenols. So like the polyphenols that make the acorns bitter, they can be used to correct, for instance, wines, but it can be used also for tanning, and for other industrial verticals. So regarding the scalability, in Portugal, we already count with a very strong network of suppliers throughout all the country, and we are also working with suppliers from Spain. And regarding the feedback that we have towards our products, for instance, we have here an influencer with whom we are working with, and people really likes the technical properties, which are and the flavor. It is really distinctive, and as you can see, this is a fully acorn-based cake. Tasty. It looks tasty, and it tastes very good. So we also performed some surveys to understand what is the response of the potential consumers to the possibility of eating acorn, or if they enjoyed acorn after eating it. So, and so we conducted a survey throughout 4 countries, and we had the response of almost 240 people from 8 nationalities, and most of the people that didn't have eaten it would like to try, and those that had already tried acorn-based foods, they really liked it and wanted to incorporate it in their diet. Well, regarding the awareness, it is not very well known. Most of you perhaps didn't hear until now that acorns could be a food, but since 2015 in Portugal, some news have been talking about that, but also internationally. The Guardian, in this news from 2019, in the Guardian, they were suggesting that the acorns can be the next superfood. And also, like in Portugal, this dish, millefeuille of potatoes and chanterelle and acorns, won the category of vegan in the New Gastronomy Awards from the RTP. And we are trying also in Landers like Thai to create awareness about the properties of acorns and as a future food. So the biggest hurdles that we have found is that the early adopters, they are very easy to convince, but the pragmatics and conservatives, they are very resilient. We have to compete with reduced tax food products. Like we have to pay 34% 3% of IVA instead of the reduced tax of 6%, for instance, of the flowers, of the other flowers. We are trying to upscale. We need to, like the market is demanding a lot of acorn products, and we are raising a seed round to increase our production to 1,000 tons per year. The harvesting, there is no harvesting tradition, so we are trying to create this tradition and to reduce costs. And unfortunately, only the Quercus rotundifolia is listed to EFSA, and our Quercus sobor can be only used as an additive. So just to wrap up, so the acorn was a food from the past and is completely aligned with the market demands. It has a sustainable production, it has a reduced carbon footprint, it is an healthy food, and it is tasty. So thank you all for your attention and to the funding from the investors and the supporting of the project. Thank you very much.
Obrigado, Pedro. Também uma excelente visão e apresentação e o trabalho todo realizado na Landratec com todo este projeto da Blota e de todo o seu aproveitamento que é de facto vital e importante e que devemos explorar também. Bom, a seguir temos a Susana Ramos e a Ana Rodrigues da Schreiber Foods Portugal. A Susana é mestre em Ciências Gastronómicas pela FCT. Ao longo do seu percurso exerceu funções de responsável de investigação e desenvolvimento em queijaria e no âmbito de um projeto de investigação conjunta com o Instituto Superior de Agronomia. Foi também nomeada para o prémio na categoria de Desenvolvimento e Inovação no Food and Nutrition Award, com desenvolvimento de produtos de queijo com microalgas. A Ana Rodrigues é licenciada em Biotecnologia com especialização em Inovação Alimentar, onde se focou na melhoria do controle do crescimento microbiológico em queijos curados e não pasteurizados, utilizando conceitos de antifúngicos naturais em substituição da natamicina. A Susana e a Ana são investigadoras na Schreiber Foods Portugal, onde exercem funções no desenvolvimento de novos produtos. A apresentação que nos vão trazer tem como título Yogurt Making, Keeping Up with Demand for a Healthier Concept. Obrigado.
So good afternoon. So thank you for the invitation for us to be here today. So we're R&D scientists for Schreiber Foods, talking a little bit of the company that we work in. It's an international company based in the US where they mainly work on processed cheese, but in the Europe region we develop a lot of yogurt. So, talking a little bit about yogurt making and keeping up with the demands, Schreiber Foods is, we don't have brands, you know?
We work on private label.
Yeah, we work on a private label. We don't have brands. We work for our customers, and that's what we do. A little bit about what we're going to present today, so where we are and what we do, and then a little bit about where we're based, so here in Castelo Branco, and then a little bit about the developments that we've made throughout the years. Clean label regarding modification starch substitution and also artificial color elimination. Then sugar reduction and lactose-free range, and how we began and how it's going. And after that, a little bit of a high protein range and the trends. Here in the locations, so basically we have 38, 39 locations. It's not all plants. Some of them are distribution centers and also offices, where in the Americas they have a lot of processed cheese. Regarding Europe, starting with the Czech Republic, they have a lot of yogurt, a lot of cream cheese, and also a line for plant-based products. After that, France, where they do a lot of desserts, a lot of yogurt, also natural cheese. They do a lot of natural cheese there. In Germany, it's the only base in Europe that we have processed cheese. Also in Spain, we have 3 plants that are focused on yogurt, desserts, and also Burgos cheese. And finally, here in Portugal, we have, we're mainly yogurt. Here is our plant in Castelo Branco. So we're focused on a factory that produces a lot of drinking yogurt. So 45% of our volume is drinking yogurt and 37% stirred. And finally, some volume in set yogurt. So we have a capacity for 70,000 tons per year with a potential of 110,000. These are some of the formats that we do. Starting with the drinking yogurt, we can do small bottles of 100 grams and also a little bit higher, so 160 to 180. With a lot of formats, we can do low viscosity, high viscosity, with only flavor, with fruit and pulp, up to 8 millimeters of size of the fruit pieces. For Greek type, so it's not like a natural Greek, it's a Greek type that is fortified with milk powder, and we fill in the bowls. So it's a 95 millimeter bowl that we can do natural with mixed fruit, with bee layers. So that's a fruit on the bottom type yogurt with pieces up to 12 millimeters. In the next cup, we do 2 types of products, so stirred and set. Stirred product is creamy, so we can do natural with mixed fruit in it up to 10 millimeters. And then set yogurt is fermented in the cup, where we do natural with only one flavor and multi-flavors in the same pack. And finally, a pouch product that is only natural yogurt, but we can do drinkable and stirred. And now for a little bit of the interesting part of our developments. So clean label. Clean label is something that is very ambiguous. What is clean label? Some classify it as eliminating ingredients that the consumer doesn't understand in the label. Some is a substitution to other ingredients that are more consumer friendly, that they perceive as more of a healthy option. In this case, we're talking about the modified starch that we use in our in our products, and substituting it with native starch. So the challenge was to work with our fruit suppliers in changing and substituting it without impacting physical chemical properties in the finished product. So as you can see in the graph, there was an impact in our products, but what we have to manage with our clients is to them to understand that there is an impact. There is a reduction of texture using native starch, but we're also giving a product that is more healthy, with less changes, as the modified starch is a chemically changed or physically changed. So this is a more healthy option and opportunity to have a better product in the market. After that, artificial color elimination. So here we substituted the E numbers for the artificial colors for basically concentrated fruits to substitute and imitate what we had with the E numbers. So E120, that's a cadmium. We substituted it with a black carrot juice concentrate and a beet concentrate. That is, the black carrot juice is more of a purplish tone, but as we work with an acidic and yogurt base, it becomes more of a reddish pinkish tone. The E100, that is a curcumin, we used to substitute it with a saffron and turmeric. Also E160, it's the carotenes. We used concentrates for carrots and paprika and sweet potato. The 163, it's anthocyanins, and we substituted with black carrot concentrate and also purple sweet potato. And finally, the 150, that it's a simple caramel, that we have to declare it as an E number. So with this product, we don't use it a lot, but we have in the coffee, in the caramel, for example, and we substitute it with those ingredients to achieve the same color. What we can take from this is, it's not a complete substitution, because the E numbers are very stable in the product during shelf life. And what we find with these substitutions, as they are more natural, they come from natural sources, and they are more variable, and they change during shelf life. So just to keep in account, and the client also needs to keep into account that there will be changes, and it's for the better, but it won't be the same color.
So about another project that we started a few years ago, and we are working on as well, because exists a demand from our clients, and I think the consumer are prepared for that. We are talking about the sugar reduction. So in 2016, our DGS, the Government Healthy Authority, published a document justifying the need to reduce sugar on food. So the food industry and the retailers worked together and provide some demands and some tasks to start to reduce sugar on food. And I'm talking about, I will explain what we did in that moment and what are we doing. So explain a little bit the method that you use in our industry. I'm only want to ask you to see the table from the right side, because I would like to explain this briefly. When we use sucralose, our sugar in the recipes, the added sugar, we obtain a sweet level around 1. And when we saw the lactose, the molecule present, the natural sugar present in the milk, we saw 0.16 sweet level on these milk molecules. So when we hydrolyze this molecule into glucose and galactose, we obtain a high level of sweetness intensity in the product. So we apply this method in our yogurt base. We made some tests, and one of them was hydrolyze this lactose partially. So in some products, this small conversion was very, very good, because we obtain a sweet level that, according to the profile of the product, was okay. But in other cases, because of the demand of the clients to reduce the amount of sugar that's asked to us, we need to put a total conversion. So we hydrolyze all the lactose from the milk, and in fact, we obtain a high level of sweetness in the product. And this keeps us to reduce the added sugar on the product. So it's very natural, only hydrolyze the natural sugar that we have present on the milk. This is one the method that we working on, but exists others. When we talk about the taste modulators and sweet enhancers that exist on the market, we work together with our suppliers. And this type of ingredients have molecules of stevia glucosides. This type of molecules are extracted from stevia, so it's another type of option naturally that we can find on the market. We can put this type of ingredient in our bases and in our fruit preps. And another method that we use, very simple, because the consumer habits are very present nowadays, and they wanted to find products with low sugar, because we have a lot of products with high sugar that it's not okay for us according to the healthy and according to our tasting. So we reduce a little bit of added sugar in a product, and according to the quality of the products and the profile that we wanted to put on the market was very similar, and we only reduce a little bit of content in a product. So we reduced since 2019, 2,000 of tons of sugar. We have a lot of sugar in here, so it's a big moment and a big congrats for us to understand how we can achieve this number only using natural methods. We have another segment, a lactose-free range. We started in 2016 with 4 SKUs in 2 range, a drinkable, a set yogurt. And we can see growing impacts of products and range on the market, because, so please, yes, I will ending, okay? So I will finish. So we can see this growing in our market. We can see that the consumer are looking for this type of products, because exists a lot of people with intolerance of lactose. So as a supplier, we need to correspond as well of the demand of the clients, and we need to put and increase this range on the market. Another product that we launched this year, and this is a big trend on the market, the consumer are looking for them. We launched the hyperproteic drinkable yogurt in a small format, because we look on a market for high volumes. So this product doesn't have added sugar, has 0% of fat. Per portion have 11 grams of total protein, from 50% of whey protein, and lactose free. So it's a big product. And we sell in Spain for Mercadona, we sell for, and here in Portugal for stores of Mercadona. And this is our portfolio. Sorry, I'm running, but we have a big players in Portugal, a big players in Spain. So this is a little bit what we are doing in Castelo. Thank you.
Obrigado, Susana. Obrigado, Ana. Também muito interessante e um grupo grande com instalação também e com uma unidade aqui em Castelo Branco, que é também de louvar para a dimensão deste grupo que eu estive a consultar e que realmente é enorme, é enorme. Bom, não sendo o último, mas os últimos costumam ser os primeiros, não é, Gonçalo? Portanto, vamos agora ouvir o Gonçalo Costa, que é, portanto, que é o responsável, ele formou a Cricket Farming. O Gonçalo tem formação em biologia evolutiva e é entomologista, taxonomista e explorador da National Geographic. Atualmente, no âmbito do seu trabalho de doutoramento, encontra-se a desenvolver o tema sobre descoberta e conservação de novas espécies de cigarras marroquinas. O Gonçalo é fundador da The Cricket Farming CO, uma startup de criação de grilos para a alimentação humana e animal que promove o consumo de insetos para a nutrição humana. A sua apresentação tem o título de Insect Bioindustry as a Link to a Circular Economy.
Estamos aqui com um problema técnico outra vez.
Isto tem tempo aí?
Não, mas são 5 minutos. Não, 15 minutos. Como é que eu faço fazer isto? Aquilo é complicado.
O quê?
É que quero expandir, não quero publicar.
Vai meter ali.
Não é isso. Espera, há um bocado as senhoras das regras. Quiseram isto tudo complicado.
Ok, deu. So thank you all for coming to my TED Talk. So the first thing I would like to, how does this change? Doesn't it? No enter? No.
Está a duplicar. Ok, vamos lá ver se conseguimos. Ok, preciso de ajuda.
Então posso usar aqui o...
Não. Isto está a duplicar. Eu não pode duplicar, tenho que expandir. Como é que isso se faz, sabes?
Não faço ideia.
Supostamente fizer aqui.
Technical issues, please hold.
Ok. Espera aí. É isso, metes a expandir. Por favor, obrigada.
Não posso os slides?
Ok, já está resolvido o problema. Tem que saber fechar. Vamos ver se
É aqui, né?
Ok,
Neste.
agora está.
Neste.
Sim, obrigado.
Sim. Welcome again to my TED Talk. So, when we think about circular economy, we think about reusing our resources, reducing waste, and also working in this closed loop approach. And Farm to Fork has its tenets within its initiative. And I think the best way to illustrate this is by looking, for example, at an initiative developed by Delta with the mushrooms, where they give up their used coffee grounds to make mushrooms, and then the spent substrate that the mushroom used is then introduced into agriculture. And how can we add insects to the mix? So let me ask of you, just a show of hands to whoever saw an insect today, okay? An insect this past week, and an insect this past month. Okay, almost all of you. And they are nature's unsung heroes because nature is based upon them, our societies are based upon them, and without them, probably our society wouldn't be as cool as it is now. And just to give a small taste of what are insects, so they have been around since the Ordovician period, about 480 million years ago, and they come in all of this, in this myriad of colors, of textures, of flavors, of functions, and each single species has its own evolutionary history tracing millions of years ago. And they are imperfectly adapted to each of their niches, their ecological roles. And albeit in a slightly anthropocentric view, we can see insects with two types of uses for humans, and the best two that we can use are the ecosystem service provided by insects, are provisioning when we see insects as food, and then insects as nutrient recyclers, so basically the garbage men of nature. Unfortunately, even though we are very dependent on insects, on society, they are disappearing right before our eyes. General trends point to from 40 to 70% losses of insects in the past years. Some areas are much difficult, but there is a general tendency. Insects are disappearing, and this is way problematic than you just think. Some people actually call this event Insectageddon, which is a bit of media trick, but I guarantee you it's very sad. I've read this book called The Insect Crisis by David Millman, and if you want to get very depressed, you can just read the book because it's super depressing. And this is mainly caused by two main issues. First, climate change, of course. Climate change is the big evil in these years. And also changes in land use and also use of pesticides. And the way we can, oh, sorry, my teacher is calling. And the way that that is going to happen in the future is that if you look at this IPCC report graph, the areas where the land will be getting hotter, so you can see several types of sceneries, arable land will get smaller, the ground will get much more hardened, so when water comes, it will just flush away, so we'll be reducing our arable land. So about 70% of water goes to agriculture, a fourth of the arable land has been lost in the past 40 years, pollinators are declining. What does this mean for food security? And the answer are insects. So this was AI generated, this picture. And I think that this boost to the insect bioindustry came with the IPCC, the FAO report in 2013, where they hailed insects as a very good promise for the future, being as feed or for food. And just to give you a couple of statistics. When you look, for example, at greenhouse gases, so the gases, for example, are CO2 or methane, you can see that, for example, cows, they burp and fart a lot, they produce a lot of methane, which has a higher impact on the short and medium term than, for example, CO2. But the crickets, for example, the crickets here are general insects, excluding termites. Termites fart a lot, I'm sorry for the term. They actually produce very low amounts of residual gases. And this can help us if we look at insect farming in the future as an alternative to all these more pollutant animals. Even also, when we look at the use of land, so insects, and I'll explain further, can be created in a micro-livestock way, so we can pile them up in a very small space. And in the same space, for example, to breed a cow, you need about an hectare by legal means. You need 1 hectare to raise a single cow. For example, in a space like, I think like this big, you can create about, I was making the math during the presentations, about 3 tons of crickets per year in just this small part. So when we think about raising insects for feed and for food and for other applications, 2 main outcomes come. First, the frass, so imagine the feces, the exuvia, and they can be used again to put in the agricultural setups. And then the protein, so it's like the hoof of the insects, and that's why we are raising them. So everybody knows this little guy, it's a cockroach. And when you think about the bio-industry, we can think about using residues that usually are going to waste. So think about cow manures, I don't know the name in English, so I'm going to say it in Portuguese, churumes from pig stocks. So those very highly pollutant residues that should be go to incinerator or some other place can be used, for example, as feed for cockroaches and used as applications, for example, in biodiesel, in lubricants. Like when we do climatic transition, guys, when we go out of the fossil fuel stage, we need lubricants for our machines. And there are some people, some illuminates, that are actually thinking about raising insects to produce lubricants, industrial lubricants for those machines, which is awesome. And I need to stress something. So the species that are used as bioremediation agents should not and never should be used be the same as insects used for food or for feed in the legislative in the law. Sorry. And when we think about now as insects as feed, so now we are going to reduce a bit the scope of the residues that we can use. We can think, for example, of the great work done by Daniel Murta on the company Entogreen. So they have been using this very beautiful fly that kind of looks like a wasp, but it's not, I promise you. And the black soldier fly actually is raised on a feed that contains, I think, 70% of olive pomace. And for the farmers out there, they know that olive pomace is a very big problem. So we produce annually, just in Portugal, 80,000 tons of pomace per year. So usually that goes to waste and they need to pay to dispose of the waste. This small fly actually thrives on that very hard residue. And then we can use the fly, for example, to produce oils, to produce protein for animal feeds. And it's a very cool animal species to work with. And now you're looking at insects as food. So now we reduce the scope, but we actually optimize the amount of things that we can use them and we can introduce them in human food. And the species that I'm working with is Achates domesticus, so the common house cricket. And people look at the cricket and they think, I think about nature. I think my youth with my grandparents, and it's actually easier to market the crickets rather than other insect species, for example, mealworms or grasshoppers. So I know this is a bit of a cherry pick of statistics, but when we look at the protein content of crickets, they actually are on par with the other traditional animal proteins. But they have a bit more of calcium and also iron, which are very important for our health. So when we think about trying to introduce edible insects to our diet, we can think of two main ways. The first one are the hedonistics, so the guys that pick up a big cricket and they take like a photo for Instagram. You know those ones? Probably you know them. And they actually do a lot of great publicity. But then as the market matures, we can look at other ways. For example, picking up insects, grinding them to a fine powder and using them in applications, for example, pasta, flours, protein bars, whatever. And the thing about that I was saying before about insects is that the species that are actually contemplated for human foods and animal feed, they love to grow up together. So they are communal. And this way we can raise them stacked. So they love getting clumped up together. If they are well behaved, they won't bite each other that much. And we can stack them atop one another. So when we look, for example, at a cow farm, we only grow in comprimento e largura, sorry. But with insects, we can also grow in height. So we have a huge potential to optimize space use. And this optimized space use can actually reduce the value chain because we can also place our factories closest to the cities, so where our consumers are. And this is a very big change of paradigm. So at the Cricket Farming Co., so it's the company that I founded in 2020, and boy, it's been a ride being a biologist and also starting now to do the entrepreneurial part. Our pillars of the Cricket Farming Co. are actually based in science, zero waste, and sustainability. Our 3 S's. And we are a small team, so we are 4 now. We have in the first panel, we have José and Sandra, and that big monkey over there that I don't know of. Then we have Sandra, so this is our pilot production unit, because we have lots of requests from our partners about raising crickets, and we have them. And also we have now Rafael. And now we also have other 2 interns that are helping us raise crickets. And now but now we don't want to be the simple producer that just goes there, produces the crickets, and then raise them to a fine powder. No, we want to do better than that. We want to take up the ingredients of the insect and then separate it, be it, for example, the chitin, which can then be processed to chitosan, which has a very high market value for the industrial pharmaceutical companies. We have the fats, we can do like different types of margarines or whatever, and then we have the protein, so it's great for people that want to do like gym and stuff. And by separating this and offering a myriad of solutions for food, we actually can go a long way. And this long way is actually being helped by a very cool mobilizing agenda called Insectera. So it is a consortium of 42 companies, if I'm not mistaken. So they involve research institutions, collabs, INESIs, and all of them work with a single goal, which is to bolster the bio-industry of insects in Portugal. And these are loads and loads of new opportunities to come, but there are still some challenges. The challenge being, for example, biosecurity. So what happens when our crickets leave the installation? Like the next big plague? No, it's not. Outdated legislation. It's very, very hard to actually certify our facilities for human food. Diseases. So, for example, our world is about viruses. So we need to up the biosecurity of our own facilities. Our crickets, if they are well and fine, because epi chicken, epi eggs or something, how to say it. And it's still a very difficult area to study, because we need a lot of money to do like a really cool part and automatizing the processes, because that's all we want to. And then, of course, there is this market acceptance, which is still on its early stages, and of course, climatization of the company, of the facilities. So to end, join me in this journey. So the next time you pick an insect like you did, you just pick it and then eat it. Thank you so much.
Obrigado, Gonçalo. Também muito interessante a apresentação, apesar de a maior parte de nós ainda não está bem com o mindset virado para esta questão da produção alimentar a partir dos insectos, mas foi evidenciado o potencial que tem a bastantes níveis. Obrigado. Eu passaria agora, depois destas sessões, depois das apresentações, há alguma questão da plateia que queiram colocar aos nossos oradores? O Gonçalo tem uma questão. Então, ligar, Gonçalo.
I actually have 2 or 3. So the first one is regarding the legislation of the Quercus rotundifolia. Why hasn't it been already accepted? Because are there differences in the acorn of rotundifolia with other oaks that are, for example, are more poisonous, have a higher degree of polyphenols? I don't know.
No, the thing is that the rotundifolia, it was that there is 2 ways to register new foods. For the traditional use, there is a law that, like foods that approved consumption before 1997, that they can be certified through an easier way, which is like you have just to prove that it is a traditional food.
Regarding the other is the pure novel food that you need to prepare a dossier and send, which is very, very expensive.
Yeah.
So the queco rotundifolia, it was used throughout the Mediterranean region, like since the
But why not quercus suber? Because they hybridize like they're from Alabama, actually.
Yeah, the thing is that even the hybrids, like for instance, why can't we commercialize in Portugal like wine from vitis, vinho morangueiro?
Rupestres.
Vinifera. Sorry, the?
Rupestres?
Yeah, there is the vitis vinifera. The wine is just from vitis vinifera. The other is not certified, I think.
And it's cancerous.
If there's any regulator here that can help us. Yeah, but that is a problem. Like, so we have to prove, for instance, the certification is for the use. For instance, for the infusions, they were traditionally used in the European medicine throughout the ages. So, like for instance, for the infusion, we can use for all the species that we have in Portuguese territory. But for the flower, we can only use but all of them they are edible. The presence of the tannins that make the flavor more astringent, it's our innovation. So we developed a method that enables us to reduce the astringency, and it's edible. The thing is,
and the other thing that I have is you were talking about coffee analogs. Does the acorn have analogs from the coffee, the caffeine molecule, or from thein? Or is it just for the flavor?
No, it's just for the flavor.
Okay.
The thing is, in fact, we didn't find. It can have caffeic acid, but it's not the same thing. It has polyphenols that gives a flavor. For instance, like chocolate. It depends on the species. For instance, if you try the quercus sobur, it will smell like chocolate or dried figs.
Okay.
The olm oak, it smells like honey and hazelnut. It depends on the species, and also all of them, like because of the tannins, they smell like liquor. It doesn't smell like liquor. Like we are used to associate the tannins to the smell of the liquors.
Okay. So thank you. And the next question are for the yogurt, and. So you are undertaking a lot of work changing the additives, the yeast, and why are you undertaking this large of change? Because you're using different types of products, and are people afraid of yeast, like in the labels? And the other question is, for example, E100, so the carmine is extracted from the cochineal, which is a natural product, and E120 is turmeric, which is extracted from the turmeric plant, curcuma longa, and how do they differ from? How do these extracts from plants and from insects differ from the extracts that you are using?
So basically,
yeah.
Is it working? Yes. Now it's working. So, yes, the consumer is very, very afraid of the E numbers. So we have to declare them in the label, and they do not understand it, even if it is natural, because it's derived from natural ingredients. But we do have to declare them as an E number, and they don't understand that it's natural, basically.
Okay.
Yes.
Okay.
The common consumer think about that. So we are scientists. We know we are on the area of food area. So, but the regular consumer doesn't understand that. I think could be a good argument, and could be a good way to explain, and this Congress is very, very helpful for that, to explain that E numbers or technical methods, chemical from natural products are okay. It's natural again, because we only talk about the technology to extract.
Yeah, because when you are looking, for example, at the clean label, I think it's cleaner to have like a small number rather than just saying beet juice extract from blah blah blah.
Yes. The consumer wants to see vegetables and fruits on the label.
Rather
only.
than numbers.
And any number, yeah.
Okay, thank you.
Acho que o Rui também tinha aqui uma questão para colocar.
Na verdade, são três perguntas, uma para o Gonçalo e para o Pedro, e é a mesma pergunta, para o Gonçalo com um pouco mais de detalhe e também para os colegas da Schreiber. Gonçalo, como é que está a produção de insetos e de produtos derivados de insetos em Portugal, agora que já temos legislação, aguardámos algum tempo por ela, nós até já tivemos com a Crica de Farma um projeto e um produto que resultou muito bem, não voltámos a ele, mas já hoje falei de um teste que fizemos, uma prova com diferentes pães e havia 2 que se destacaram e para alguma infelicidade minha na altura no painel, destacaram-se em relação à nossa fórmula base de pão de medronho e utilizámos uma matéria-prima vossa, que não era vossa ou era vossa uma pequena quantidade, portanto gostaria de saber como é que está neste momento a produção e quais são as perspectivas de futuro para a Crica de Farming. Faço também já a pergunta ao Pedro, porque esta matéria-prima da bolota nós sabemos que temos imensa, não é? No montado no Alentejo sabemos que algumas explorações de porcos ao ar livre utilizam-na para consumo dos animais, mas nós temos muito mais produção do que aquela que é consumida pelos animais. É possível hoje encontrar-se com facilidade a farinha? E a pergunta para as colegas da Schreiber, que é, sim, vocês não têm uma marca própria, trabalham tudo para marcas dos vossos clientes. Como é que surgem os vossos projetos de inovação? São internamente ou são os vossos clientes que vos desafiam? É isto.
Obrigado.
Posso começar?
Temos pouco tempo,
Respondo em
mas...
português ou em inglês? Português, então pronto. Portanto, aquilo que nós estamos bastante informados relativamente ao panorama de produtores de insetos em Portugal, até porque nós fazemos parte de uma associação que é Portugal Insects, e agora também com a Insectera, e portanto existem 3 produtores. Para a alimentação animal é a Entogreen, portanto liderada pelo Daniel Murta, que tem já uma fábrica, nossa, 3 pinguinhas, e depois nós temos também o Vasco Esteves, não estou em erro, que é da Tecma Foods, da Tecma Bugs, que é lá de cima do Porto, em que estão a trabalhar com a larva do Tenebrio Molitor, e depois nós temos a Crica de Farming Co., a minha empresa, em que está unicamente dedicada a produzir grilos para a alimentação humana. Relativamente a prospecções do futuro, agora, uma das medidas que nós temos do Insectera é termos uma fábrica a produzir no fim de 2025, portanto, isso vai ter que acontecer. Então, essa vai ser a nossa meta para 2025. Pronto.
A segunda questão para o
nosso
Pedro.
caso. Pronto. Relativamente à bolota, existe já farinha de bolota no mercado. Há uma empresa que foi pioneira, que foi o Freixo Someio, muito conhecido, com o Alfredo Sendim, no Alto Alentejo. Existe também no Alto Alentejo outra empresa que é o Moinho de Pisões, que também produz, estamos a falar de quantidades relativamente pequenas, estamos a falar na ordem, não chega às 10 toneladas por cada um deles. Havia outras que infelizmente não conseguiram avançar precisamente por uma questão de escala. O mercado precisa sempre de quantidades, para testes de mercado precisamos de quantidades muito maiores. No caso da Landratec, e estes parceiros, que são players, mas também são nossos parceiros, eles trabalham a uma escala artesanal. Nós começamos logo o projeto, na altura do projeto, com uma ideia de industrializar e desenvolvemos todo o nosso processo com a ideia de industrialização, identificação de equipamentos que nos pudessem ajudar. Neste momento temos a linha montada, permite-nos produzir algumas dezenas de toneladas por ano, mas neste momento estamos a levantar uma ronda seed para, no espaço de 3 anos, conseguimos produzir 1000 toneladas. Relativamente ao mercado, o mercado chegou a tudo. Nós estamos a receber, nós não conseguimos, por exemplo, entramos na Auchan e estamos a trabalhar, não é exclusivamente, mas está a absorver muito da nossa capacidade, mas o mercado precisa de muito mais e nós precisamos de ajudar o mercado a conseguir.
Obrigado, Pedro. Por acaso, a questão viu uma reportagem há pouco tempo da herdade do Freixo.
No público, sim.
Exatamente. E na RTP, creio, também, sobre o potencial da bolota. Obrigado. E faltava só alguém responder da...
Muito rápido.
Muito rápido. Já não temos muito.
Então, o objetivo dos private label é realmente eles colocarem a maior mancha da sua marca nas suas lojas. Portanto, a ideia dos projetos vem por parte do cliente, porque querem acompanhar as marcas, porque querem também inovar, apesar de querem acompanhar, também querem inovar as suas categorias, isto em novos produtos, assim como melhoramento ou improvement dos produtos que têm no mercado, e também um pouco do nosso trabalho, porque é a nossa pesquisa e porque é também o nosso ideal como cientistas para desenvolvimento, apresentar melhorias quanto aos produtos. Portanto, vem um bocadinho parte a parte, mas muito do cliente.
Acho que obrigado. No sé si Belén quieres aportar algo a la conversación, alguna cuestión. Sim, vale. Venga, obrigado pelo excelente contributo de todos e obrigado, penso que não há mais questões. Damos por encerrado. Eu só queria dizer que há aqui potencial para o café também, não é? Tal como foi avançado pelo Pedro e não pelo Rui, mas também podemos pensar em alguma coisa, obviamente que sim, que há ali questões que podemos ligar. Vamos agora ouvir as três apresentações públicas a seguir, e obrigado pela vossa participação a todos. Excelente apresentação. Obrigado.
I only want to say that the site visit to CATAA has been moved from 5:30 to 6, and now we will have the presentations, the oral presentations, and next to the oral we'll give the awards from the teams that awards from last day and from today. So we'll proceed with presentations now. Yeah, Vasco Lima, talking about fruit preservation by hyperbaric storage, the effect of pH on inactivation of Listeria monocytogenes.
É necessário carregar aqui em alguma coisa?
É só carregar.
É aqui? Hello, my name is Vasco, and today I will present in a very short form some results about food preservation by hyperbaric storage. And so, hyperbaric storage is a methodology for storing foods under pressure conditions up to 100 Pascal. And the idea is to control the growth of microorganisms and some of the enzymatic and chemical degradation in order to extend the shelf life and with the consequence of substantially lowering the energy costs and the greenhouse gas emissions in comparison with refrigeration. And so the work that I will be very briefly presenting, I did it with watermelon juice that has a high pH and water activity, and so is very perishable. And so the pH of the juice was adjusted to 4 and 6.5, and the juice was inoculated with the pathogenic Listeria monocytogenes and stored at room temperature, in pressures up to 200 megapascal. And so the first result, it showed that by increasing the pressure, it is possible to reduce the time required to achieve the inactivation. In the black line is the refrigerated control, and in the blue line is the highest pressure. And we can see that by increasing the pressure, it was possible to achieve a 4.7 log reductions after 24 hours, instead of the 7 days at lower pressure. And also by changing the pH, it's possible also to achieve quicker inactivations. For example, in the red lines on the second, it's possible to see the reduction after 10 days or in 21 for the pH 4 or the 6.5. And based on these results, it was observed that depending on the initial microbial, told that it is possible to see this technology and its potential for pasteurization. And so, in a second, we approached this pasteurization in the new methodology that we call moderate pressure pasteurization, that it uses higher pressure for shorter periods. And so by using pressures from 160 up to 250, it was possible just to pasteurize the juice at the room temperatures in a matter of a few hours. And by increasing the pressure, the possible impact of the pH is not as relevant. And it was possible just to achieve the 5 log reductions after 35 minutes in the pH 6.5, which is very high pH. And so the conclusions are very shortly that by lowering the pH and by increasing the pressure, it's possible just to achieve a quicker inactivation rates, and also that by during this storage, it is possible to control the growth of the microorganisms, but also to pasteurize the foods. And this can be seen as a sustainable option with almost no energetic costs and with very low greenhouse gas emissions. And that's it. Thank you.
Thank you, Vasco. Now we'll have Gabriela Matos presenting hyperbaric storage as a new methodology for preservation of egg white and at room temperature.
Aqui? Sim, vamos ver se funciona com os setos.
Sim, ok. Aqui?
Não, se quisermos.
Aqui?
Os setos.
Ai, para trás. Aqui. Hi, good evening. As like my colleague Vasco, I will talk about hyperbaric storage, but now in other matrices, in egg white. Egg white play an important role in the food industry due to their rich protein content and also for its desirable functional properties such as foaming, emulsifying, gelling capacities, leading to their use in a vast variety of food products. Nevertheless, eggs are highly perishable and prone to microbial proliferation, giving attention also to Salmonella pathogenic microorganisms. So, normally, egg white goes through thermal treatment, also known as pasteurization, but this causes loss of nutrients and the functional properties are compromised. Also, it is normally placed under refrigeration, and as you know, it is reported to have economic and environmental impacts, and also the shelf life of egg white remains limited. Thus, there is the need of more efficient and sustainable methodologies of preserving egg white, guaranteeing its microbiological safety without compromising these functional properties and quality. So, again, hyperbaric storage is a pressure-based food preservation methodology. It is based on the application of moderate pressures, up to 200 megapascal. The food products can be stored during days or even months. It can operate at room temperature, which is a great advantage, and have low energetic costs. Due to this advantage, in the last decade, it was performed several studies assessing the application of hyperbaric storage on different food matrices, just like juices, cheese, soups, meat, and also in fish. Overall, compared to refrigeration, it was observed an effective inhibition or slowdown of microbial growth with extended shelf life and also the quality parameters maintained during this storage. In this work, egg white was inoculated with 4 pathogenic microorganisms, 2 gram-negative bacteria and 2 gram-positive bacteria. After the inoculation, then it was stored under pressure with pressure levels between 25 and 100 megapascal at room temperature, and it was stored up to 60 days. And also, for controls, it was put the samples in refrigeration. After the storage, after the hyperbaric storage, it was made an evaluation on the effect of this methodology on the microbial growth, and also it was evaluated some functional and physicochemical analysis. Microbiological results show that between 50 and 100 megapascal, the yellow line and the pink line between these pressures, it allowed to reduce the microbial population with levels with reduction of more than 5 log units, reaching here a pasteurization level, in this case for Salmonella enterica, maintaining below the detection limit up to 60 days of storage. Similar behavior was observed for other pathogenic microorganisms, such as Staphylococcus aureus, Listeria monocytogenes, and other strains of Salmonella enterica. Regarding functional and physicochemical analysis, no changes were observed, or no pronounced changes were observed in pH, in color of egg white, in soluble protein, and also in the gelling properties. Here, the water holding capacities. Consider this, hyperbaric storage have a great potential not just to preserve egg white, but also not just to preserve egg white, but also to inactivate microbial population to values normally achieved by pasteurization, and also have the potential to replace refrigeration with environmental and economic and sustainable benefits. Thank you.
Thank you so much, Gabriela. I will say that Luis is not here. Is he here? No? Luis Castro is not here. So I want to refer that, I don't know if I said so before, the site visit will be at 6 and the meeting point will be in the front of the building. Não em português. A visita ao CATAA será adiada para as 6 e o local de encontro será em frente ao edifício.